And then also the indiments, or the hairs and glands, are highly characteristic and very, very diverse. So the Thyleptera ace have what are referred to as hyaline acicular hairs, which are transparent needle-shaped hairs. And these are often simple, but they may be branched, they may be hook-shaped. They're highly diverse within the family, and there's also a lot of diversity of glands, which the position, the shape, and the color of the glands can be very important. So please feel free to interrupt if you're able to load my talk, and I can transition from there. Otherwise, I'll continue talking about the Thyleptera ace. So the spores are also highly diverse and can be very, very useful for diagnosing major lineages, and also in some cases can be really useful for identifying closely related species that are distinct. So they may be reticulate, they may have wings or ridges, they can be very spiny, or have a combination of macro characteristics and micro characteristics. And so that's something that there's been some other work by Syed Shah and others in northern Pakistan where they're able to look at Thyleptera ace spore morphology and identify them to species. So it's really a promising line of research for looking at palynology and digging into the fossil record to be able to use spore morphology. So this is a project looking at the evolution of pairing morphology in Thyleptera ace that I worked on with my lab mate at the University of Vermont, Nikisha Patel, who took the lead. And so we were trying to look at different patterns of diversity. We found that some characters, like reticulate spores versus non-reticulate spores, were highly characteristic of large lineages within the Thyleptera ace. But some of the other patterns were much, much less distinct and were much more variable throughout the family. So in the course of that work, I began exploring a lot of the data that were published to build a large molecular phylogeny using some original newly generated data, and also synthesizing all the data that had been published on the Thyleptera ace over the last 20 years. So this was work by Alan Smith and Ray Cranfill, He and Zhang working out of China, and then Thais Almeida led a large collaboration that involved Harold Schneider, who will be speaking next, working towards a phylogenetic generic classification of Thyleptera ace, and they really made a lot of wonderful progress, especially in the neotropics, where they really sorted out the taxonomy of a lot of these groups and followed it up with a lot of taxonomic changes. But then in the old world, in the paleotropics in Malaysia in particular, there has never been a very detailed or densely sampled molecular phylogenetic study. So Richard Eric Holtum, who was working at Kew, who lived from 1895 to 1990, and the latter part of his life, he began a lot of very detailed work on Thyleptera ace, in which he named 7 genera and made hundreds of combinations and provided really the first detailed system of classification that dealt with this part of the world, of tropical, the paleotropics, both Africa and especially Malaysia and the Pacific. So I was fortunate to be able to participate in an organization called the Teratophyte Phylogeny Group, who brought together 100 different researchers from all over the world to try to develop a consensus for classification of the Thyleptera ace. So I worked specifically on the Thyleptera ace classification, and this group also involved Thais Almeida and Harold Schneider, and we largely adopted their classification for the neotropics. But then for the paleotropics, we were working with Richard Eric Holtum's classification, which hadn't been updated in more than 50 years and really hadn't benefited from any molecular phylogenetic evidence. So when the PPG was published, 10 of the 30 genera of Thyleptera ace were non-monophyletic, as they were circumscribed or had never been tested, and many of the old world genera were not diagnosed by a morphological synapomorphy. And again, the taxonomy was a half a century old. So the goal for my dissertation was to revisit this taxonomy using a combination of molecular phylogenetic evidence, morphological evidence by studying herbarium specimens, and also incorporating biogeography to try to inform some of these patterns. And so some of the challenges with working with the Thylepterid ACE is that many centers of diversity are very poorly collected. Many species are narrow endemics and they just occur in one small part of the world. At the species level especially, there's a lot of incidence of polyploidy and reticulation, so cryptic species that can be very difficult to recognize without very detailed data sets. And then also very few specimens are complete. The typical herbarium specimen of a member of Thylepterid ACE is one small portion of a single leaf, which also makes it very difficult. And then also microscopic characters are usually necessary to identify genera. So being able to look at the hairs, at the spores, at the venation. So it takes a lot of detailed study even to classify members of this group to the genus level. So this is an example of this would be the genus Chingia, which is mostly in Malaysia. And of the 24 species that are known at the time of the PTG, only eight of these were known only from the type specimen, only from the type collection or one or two other collections. And only a single member of this genus had ever been included in a molecular phylogenetic study. And to get into a little more detail, some of them have foul-smelling, irritating hairs on the stipe, so collecting them can be challenging. Some have leaves that are 12 feet long, so I guess that would be 4 meters. And the populations are also ephemeral, so it's a pioneer species that may come in after a disturbance and then disappear as the forest grows in around it. So, and they also tend to grow in some places that can be very difficult to get to, so isolated islands and mountains and some places that have not been well collected. So an example of this is of a new species that was hiding in plain sight. When I was collecting on the islands of Fiji, there's one of these ferns that was 2 meters tall and growing alongside of a very heavily trafficked paved road. And this had been collected before. It existed in herbaria. Non-diversity. So it was just a matter of spending time and really studying these specimens and comparing them with material from other places to find that there are often new species are often discovered hiding in plain sight and within herbarium collections. So I'll move on to the heart of my talk, which is proposing a new classification system for the Thylepterid ACE. So I had the great fortune of being able to collaborate with an amazing group of people who had funding to generate phylogenetic sequence data, molecular phylogenetic sequence data for thousands of ferns, lycophytes, conifers, and mosses and liverworts. So I was able to be involved with their project, and they were using what's called targeted enrichment, and they developed a custom probe set of 400 probes. So it's called the GO-FLAG probes, and it's part of the genealogy of flagellate plants. So the idea with this approach is to target a highly conserved coding region of DNA, usually about 120 base pairs, that has 65% or more pairwise identities. In one case, with a endemic to the island of Kauai with a long creeping rhizome and very small fronds that grows in rivers and near waterfalls. And then on the island of Oahu, there's another case where it has evolved into a rheophytic habit, again with very small fronds. And so the next one is Pronephrium segregate is Gripothrics. And this was a case where we adopted the taxonomy of molecular evidence. So in this case, these hamate or hooked hairs are typical of every single member of this genus, and they occur on the stem part of the plant, on the sporangia, on the scales, on the lamina, sometimes on the stipe. Often it has dimorphic fronds, and the sori are always ex-indusiate, so always without indusia. But those hamate or hook-shaped hairs, which is the basis of the name Gripothrics, are common to every species that has been studied. And this group would be represented in India by what was Pronephrium triphyllum or Gripothrics triphylla. And Pronephrium sensu stricto, the type is from Java, and so far as we know, it does not occur in India. And this group has very strongly dimorphic fronds, so the fertile and sterile fronds are quite distinct, and usually has sori and indusia with these spherical yellow glands that are really typical of this genus and some very closely allied genera. So it's a character that seems to help define that clade. And then the genus Nanothelipterus, which is most diverse in the Philippines, was found to be highly nested within this group. So another discovery that would impact the nomenclature in India, or the classification there, would be that the widespread species that was treated as Sphaerostephanos unitus is now treated in the genus Strophocolon. And this is a very widespread species that occurs in Africa, all the way into the Pacific, into Papua New Guinea. And there are only 2 species that are in this genus, but they are ecologically and morphologically very distinct from all other members in Sphaerostephanos, and having very long, elongate, creeping rhizomes that are unlike anything else we've seen in Sphaerostephanos. But they also have these spherical yellow glands that's characteristic of a larger clade, and some characteristic venation that is also unusual and is unique in Sphaerostephanos. So one last change that would impact the classification in India would be the genus Parathelipterus. So the Parathelipterus bedomii is common all throughout the Paleotropics. It's widespread from China and into Papua New Guinea, and it gets into India. And that, in our analysis, comes out to be sister to a large group of mostly neotropical plants in the genus Amoropelta. So the genus Parathelipterus, as it was recognized by the PPG, resolves in 3 different clades. 2 of these are within what is recognized as Coryphopterus, but the type of Parathelipterus is in this larger clade of other members of Amoropelta. So we are treating this as an Amoropelta and in the subgenus Venus. So we used a subgeneric system. So it's really interesting to look at Amoropelta from northern Michigan, from Sri Lanka, and from Puerto Rico, and to see the amazing similarity in the morphology of these groups that had not been treated together in the same genus. So it's been a really amazing process of using both the molecular data to help guide some of the morphological study. And so just having all these different lines of evidence really made it much more powerful to be able to look at these things than using any on its own. So it looks like maybe my slides are up now. So I'm sort of nearing the end of the talk now. So I'm not sure where to go through with the slides, or if you would like me to review anything, but I think I was at slide number 43, if you wanted to catch up to where I had been. But at least everyone gets to see the slides now. Not in synchrony with the words as I had intended, but. So there we are. That's number 43. Okay, so this is an example. This is one I'm happy to be able to share with you. This is the one on the left is Amoropelta from Michigan. In the middle is an Amoropelta from Sri Lanka, and an Amoropelta from Puerto Rico. So you can really see the how similar the morphology is on these species from very different parts of the world and very different climates in some cases. Next slide. And so here we have the phylogeny, the global phylogeny, with a map of the clades onto the world. So you can kind of see some major patterns here. With the yellow is the Paleotropics, the green are the neotropics, and this blue is the color. You can see some radiations of circumboreal or temperate origin. And then what's really interesting to me is to look at the red. So looking at Africa. So there's very few members of the Leptiridaceae and a smaller fern flora comparatively, but they're very distributed throughout the diversity of Leptiridaceae, and this is a pattern we see in other ferns. So it could be the result of extinction of these really isolated lineages. So that makes working on the taxonomy in Africa really quite challenging. So next slide. So in this case, we sampled 4 different members of the genus Pneumatopterus from Africa, and each of them resolved in a completely different and distantly related genus. None of them had been, were even close to where they had been originally treated. So just suggests how little we actually know about some of these places when we're working within a framework that was developed in one part of the world and trying to apply the diversity of a different part. Sometimes there are a lot of surprises. So next slide. And so I would just like to say that I think there's just amazing opportunities for studying ferns in India right now, and it really is a milestone and a huge accomplishment to have this work by Fraser Jenkins et al., the checklist of Indian pteridophytes, which will just provide such a wonderful foundation. So when working on any sort of taxonomic work, we're always standing on the shoulders of giants, and we're always building on what has come before and refining hypotheses. And so we're just at a very exciting moment now where all of this literature is becoming more accessible and more available online. We have access to data in places like GenBank and all these different publications. And so the possibility for being able to do work now is just really exciting. And there's been some great recent work on Cristella in China and the Himalayas. So there's some better understanding of that group there, treated as Cyclosaurus subgenus Cyclosauropsis, which corresponds to our concept of Cristella. So great opportunity there, great opportunity for partnerships. And then the next slide. And another group that I think is really exciting and interesting and very diverse and poorly studied in India are these 2 sister genera, Trigonospora and Pseudocyclosaurus. So again, Pseudocyclosaurus is really diverse in the Himalayan region and coming into the northeast of India. It has been studied by Zhongyang Li in China. And then this group Trigonospora is very, very diverse in Sri Lanka. And so there's been some work there. And then I know Ranil has done a lot of great work in Sri Lanka. So there's just such a wonderful foundation to build on and to be able to use as a starting point to continue with this work. The next slide. And I just want to let you know that our system of classification is free and open access. So if you want to see any of the morphological features we used, a key and a description to genera, you can download our treatment for free from the Botanical Research Institute of Texas, which published it. So it's entirely free and open access. And then the next slide. And then the results of our phylogenetic study are available, were published through Systematic Botany in our research. Mentor and my closest collaborator, Alan Smith, who began work on the Leptiridaceae in the 1960s and has been just an incredible mentor to me and has been an enormous help in the new treatment. And I'd also like to recognize the University of California, the University of Vermont, University of Florida, Taiwan Institute of Forestry, and the University of Zurich Herbaria. So without having these amazing collections that provided the herbarium specimens and also the tissue, this work would not have been possible. And so the authors on this phylogenomic study were all very close collaborators with this. And then next slide. But they weren't the only ones who helped. And I just like to acknowledge Joel Nitta, who's the very first to offer up his tissue so I could have DNA for the Leptiridaceae. He had some wonderful collections from Costa Rica and from the Society Islands that he shared with me so generously. And so I just appreciate the generosity of the international community that really supported this work and helped out. in my lab, Nikisha Patel, who helped with a lot of the spore research and things early on. So with that, I can take questions, or we can revisit any of the slides that that anybody hoped they could get a look at that went very quickly. So with that, you can go to the next slide, and I will take questions.
Hello, madam.
Hello.
Yeah. Discussion. Yeah, if there are few questions you can ask now, please. Dr. Susan Fawcett is ready to answer your questions. Okay, thank you, madam. Thank you very much. Next.
Thank you.
Thank you. Thank you. Next one. Shall we start with the next speaker? Okay.
I was just, she was telling about the paleotropics, and she treated paleotropics in yellow color while showing the distribution. Only thing is that African part is part of the paleotropics, and why she treated Africa separately. Anyway, she is out of the screen.
ചോദിച്ചില്ലല്ലോ സൂസൻ പേഴ്സൺ ചിലപ്പോൾ റീജോയിൻ ചെയ്യുമായിരിക്കും. അപ്പോൾ നമ്മുടെ അവരുടെ ടോക്ക് നന്നായിരുന്നു പറഞ്ഞ് നമ്മൾ കൺക്ലൂഡ് ചെയ്തിട്ടില്ലല്ലോ. ജസ്റ്റ് താങ്ക് യു പറഞ്ഞ് അവസാനിപ്പിക്കുകയും ചെയ്യുന്നത്. അപ്പോൾ നമുക്കൊരു 2 മിനിറ്റ്സ് ചെറിയ ഇൻട്രാക്ഷൻ കൂടെ ആക്കിയിട്ട് ഇവർ ചെയ്തതിനെക്കുറിച്ച് നമ്മൾ സമ്മറിയും പറഞ്ഞു. ഇപ്പോൾ അവർ തിരിച്ചു വരില്ലല്ലോ. നമ്മൾ മറ്റൊന്നത്തിൽ നമ്മൾ ഓരോ ഇതോ സാറ് ക്വസ്റ്റ്യൻ ചോദിക്കുന്നുണ്ട്. സാറൊന്ന് പറഞ്ഞു.
Hello, shall we ask some questions, some clarifications?
Dr. Susan, thank you for excellent presentation, and it is indeed very pleasant to see you and listen to your presentation. I just wanted just one clarification. In your distribution map, you showed in red Africa, and you did not treat it as part of paleotropics. Only Asia and Southeast Asia have been treated as paleotropics in yellow.
Yeah, that's slide 44. You can go back. So I did misspeak. Africa, of course, is part of the paleotropics. So the blue color was or the
For neotropics?
The blue color is temperate,
Okay.
the temperate part of the world, and then yellow is Southeast Asia, Australasia, Malaysia, so the more tropical eastern part. So of course paleotropics was not the right term. So just separating Africa from Asia.
Okay, that suits your this thing, distribution.
Well, I'm S.P. Kuller. My point is, why this oversplitting? Don't you think this oversplitting is a sort of destabilizing nomenclature? You have a genus, then you have a subgenus, somebody else will come and then you say subgenus, subgenus. Why this oversplitting? This oversplitting, you know, harms taxonomy more than it solves more problems.
No, it's a great question, and this is the taxonomy that I propose. There are others. So Christopher Fraser Jenkins, for instance, he prefers to recognize a much broader concept of Thelipterus, but within Thelipterus he still recognizes these subgenera. So you could take my classification of all these different genera and treat them as subgenera. And so the idea of recognizing these taxa is to recognize these evolutionary groups. And so the idea for me, and so I will say also, there has been some splitting, but we also treated something in synonymy. So Parathelipterus, for instance, has been lumped into Amoropelta. Nanothelipterus has been lumped into Pronephrium. And so my goal with the classification is that, so each type for each genus should be represented by a monophyletic clade and a clade that is recognizable both by using morphology, so that it can be diagnosed morphologically, and then also should be evolutionarily, the members of the species of that genus should be much more closely related to each other than they are from a much more distantly related genus. So in the case of Pronephrium, in the case of Cristella, in the case of Pneumatopterus, these were extremely heterogeneous taxa. Cyclosaurus, broadly defined, also included a very challenging global mix of species that could not easily be diagnosed using morphology. So it does so happen that this classification is 37 instead of 32 genera, but my goal is to hope that these genera that we do recognize will be recognizable using morphological characters that can unite them and can define them so that they can be identified using keys, that they're cohesive geographically, and that they share an evolutionary history and a relationship to each other.
Thank you. Thank you very much. No, it gives a feeling that whenever a new person comes in, the person wants to be different from the things that have already been recognized. And this we put as advancement of knowledge. So in the garb of advancement of knowledge, we either split or we lump. And this thing has been going on ever since, lumpers versus splitters. So I don't really understand this whole idea of splitting and lumping. And this leads to, as I said, a sort of a nomenclatural disturbance. For example, you know, we had the genus Thelipterus and several genera that were recognized by Holtum, Pichis, and others. And then A.R. Smith himself somewhere in the 80s, I had his paper also. He suggested, well, a single genus Thalepterus is sufficient to cover all Thalepteridaceae. And Holtam himself had also suggested that we can have a single genus Thalepterus for the entire Thalepteridaceae, because the moment you mention Thalepterus, it gives you the picture that, okay, this is what it is. But as advancement goes on, splitting goes on, and as far as your classification is concerned, I think that would be in the category of the splitters. Thank you very much for your wonderful lecture.
Thank you.
As regards Thalepteridaceae, Holtam's classification is best. I still feel. Yes, sir. Nice.
Hello, Dr. Susan. I am Santosh Nambi. Thank you for your excellent presentation. And as you know, we have a very good diversity of therapeutic research in India. And also many of us are also very much interested to collaborate with you. Because in recent years, there is a lot of people are coming to work on the taxonomy of therapeutic Ferns. So in the future, we will be collaborating with you. Thank you for your nice presentation.
I look forward to that. And then the other thing I would like to say is if you ever have difficulty tracking down classic literature or old papers that are hard to come by, please feel free to email me and I can do my best to try to find these from library or get scans or to try to share some of this literature, because I know it can be it's very challenging to track down, and once you have this stuff scanned, it's very easy to share. So if you're ever looking for anything like that, I'm very happy to provide.
Excuse me, ma'am. I just wanted to bring to your notice that we in India have the Indian Fern Journal, which is the mouthpiece of the Indian Fern Society. I would request you to send your publication for inclusion in the Indian Fern Journal. We shall be highly grateful for that. And we'll treat your article as an invited article. So you can send it to me anytime. I'll ask the organizers to give you my email. You can just email to me as an attachment and we'll get it published.
Thank you so much.
I hope you'll agree to my request.
Thank you.
So thank you very much, Dr. Susan. Hope we will meet again in future.
I'm sorry I can't be there in person. I would very much like to be there.
Yeah. Okay. Okay, thank you once again. Your presence itself is a very inspiration for any seminar. Yeah.
Hello, very good morning.
I am Professor.
Can you hear me?
Yeah, okay, it's okay. I am Professor Madhusudanan chairing this session. Hearty welcome to Calicut University, the International Symposium on Research in Teratology, hosted by University of Calicut and sponsored by the Indian Fern Society, Chandigarh. Hearty welcome, sir. Can you hear me?
Yes, of course. I can hear you.
So, there will be a small introduction on your work by Dr. A. Benjamin. So, Dr. A. Benjamin will introduce you and welcome you officially. So, I request Dr. Benjamin to come forward and give an introduction. Please listen, sir.
Thank you, sir.
Okay, thank you.
Good morning to all of you. Good morning, Dr. Ranil.
Good morning.
So I'm very happy to be here to introduce Dr. Ranil. I had a very close relationship with him from 2007 onwards. We had a chance to meet at Philippines International Symposium on Pteridophytes in the year 2007. So I would like to just introduce Dr. Ranil. Ranil is basically he has done PhD on conservation biology in Pteridophytes. After that he has shifted to university as a visiting lecturer. Then now he is working as a senior lecturer in Department of Crop Science, Faculty of Agriculture, University of Peradeniya from 2012 till date. And I can say he is the only one man in Sri Lanka. He is doing the work on pteridophytes. For last, I think, several years we have seen him. He is a very good worker. And his specialization is he has done a lot of work on tree ferns in Sri Lanka. So on behalf of the organizing committee secretary, I welcome Dr. Ranil. I just request Dr. Ranil, he is going to discuss about conservation of fern flora, challenges and strategies. Now I request Dr. Ranil to start your session. Thank you.
Right.
Yes, please join us.
Okay. Thank you, Dr. Benjamin, for giving an introduction on me. So actually, once again, I want to know whether can you hear me properly?
Yes, we can hear you.
Oh, okay. Right. Let me share my presentation.
Yes, please start your presentation. Please start your presentation.
Oh, okay. Please. Hello, can you see the screen?
Yes, okay, please start.
Yes. I want to know whether it is in a slide mode or presentation mode.
Slide mode.
Not in a presentation mode.
Yes, you can start.
Okay, is it okay?
It's okay.
Right. Okay. Good morning, everyone. And first of all, I thank for the organizing committee for giving me this opportunity to share some of my thoughts and experience with you all. My presentation topic is conservation of fern flora, challenges and strategies. In here, I'm basically focused on conservation perspectives of fern flora in South and Southeast Asia. Right, so here you can see this distribution map. This is the very recently developed distribution map. It shows the global pattern of fern diversity and endemisms. I think no need to explain about how the tropical belt and Asia are rich in species diversity and endemism. It is very clearly depicts there. Right? And also this is very interesting article. Please read that article. We can have a very good idea on conservation aspects of the Asian ferns. Right? If I further elaborate, so according to the Ebihara in 2000 2018, approximately 4,700 species, including subspecies and varieties of pteridophyte taxa distributed across the Asia, right? Excluding the New Guinea. And also, it is very interesting that two-thirds of the genera, including 32 endemic genera, occur in Asia. So very, very interesting figures. So sometimes these facts, these figures may be slightly changed because we all know that the taxonomy is highly dynamic subjects. It's changed, that value, facts, figures change every time. So however, I think these figures are itself enough to show the importance of Asian pteridophytes within the field of pteridology, right? And also, Asian pteridophytes have very interesting biogeographic history.
Sorry
And
for the
also
intervention.
it shows some...
Can you make it in presentation mode?
Sorry?
Can you make it in presentation mode?
I think it is in a presentation mode in my computer, but
Navigate it in slide mode.
you are getting the slides mode. Okay. I will share it again.
Okay now. It's okay now.
Okay, wait. I will share it again.
Okay. I think you can continue. Please continue.
Okay, wait.
It's okay.
So how about now?
It's okay.
Okay, shall I continue?
Oh, yes, please. Please continue.
Oh, okay. So what I said earlier, the Asian pteridophytes have a very, very interesting biogeographic history. And also it shows close affinities of the fern flora, pteridophyte flora of other parts of the world as well. For example, Fraser Jenkins suggested that Sri Lankan and South Indian ferns show affinities with Himalayan fern in Northeast India and Southwest China, and also close relationship with Malaysian flora in Southeast Asia. And also he suggested that there is a little connection with African element as well. But however, someone has to do work on this area because it has not proven yet using a recent molecular taxonomic tool. So in here I'm trying to explain the importance of the Asian fern flora in a global level. So it is well known that despite the geographic limits or boundaries, pteridophytes are subjected to the threats due to various reasons. As far as my knowledge, I think less than 900 species have been assessed for only global red list. It's very less number. But most of the Asian countries, they have developed their own regional list for pteridophytes. So actually that's information itself enough to make some conservation plan and management plan for the ferns as well. So in here, I have compared the conservation status of ferns in 4 Asian countries: Sri Lanka, Japan, India, and China, right? So you can compare the total number of threatened species, right, which are listed in a blue color circle with the land extent of each country. So I was unable to find the recent statistics of the Indian pteridophytes red listing status. I used 2012 facts. If the present facts are changed, someone can comment at the discussion session. So in here, it is very clear that the island population is subjected to high rate of extinction than the mainland population. Sometimes it may be due to environmental reasons, sometimes it may be due to high rate of the inbreeding, right? However, this depicts that level of threats faced by
Dr.
pteridophytes.
Anil?
Yes.
Dr. Anil, can you hear me?
Yes, sure.
The slides
Yes.
are not moving.
Sorry?
The slides are not moving. So can I kindly guide you? Can you go back to on your screen, the screen presentation?
Wait.
Slideshow? Can you go to the slideshow?
Yes.
Okay, there on the top, you have the design, transition, animation slideshow. Can you click on the slideshow?
So, sorry.
Can you click on the slideshow option?
Then,
Top, next to the home, insert, design, transition, animation, slideshow. The top menu bar.
how about now?
No, sir. Can you please
No.
go back? Can you please go back to your slideshow? Don't make
Wait,
it.
wait,
Okay,
wait,
yeah.
wait.
There you
Okay.
are. Okay. Go to the slideshow. There is a second option. Choose the second option. Display in a window. And then share your screen. It will
A
come.
window. Yes, I shared it.
Is it shared in a window?
Yes.
Okay. Now share full screen. No, we still can't see.
Now it says the stop sharing.
Display in a window. Yeah.
Yes, it is in the window. Right.
Another option is keep it in slideshow itself, and as you move the slide, kindly click the next slide. At least then we can see the screen.
Okay. Share. Okay, shall I continue?
Yes, please.
Okay, right. Okay, right. So please carefully read the statement given in this slide. So by 2008, a global risk assessment had been completed for only 2% of the 11,000 species of fern and lycophytes. 89% of these ferns were considered to be at risk. So what it says, right? It depicts the level of threats faced by pteridophytes across the world. And also, though it has assessed the fewer number of taxa, it shows the gravity and depth of this problem. So I found very interesting article which was compiled by Ebihara and his group in 2012, right? But 10 years ago, but the results are still valid, right? These results are still valid, right? So I will briefly explain what the outcome of that study, because it's give a very clear picture on the conservation perspectives of the Asian fern, right? So in this study, 886 taxa were identified as narrowly distributed taxa in Asia, right? 880 taxa, right? And also it is suggested that approximately 1/5 of the Asian pteridophyte taxa are narrowly distributed and need special attentions of their conservation, right? And also it includes 101 taxa from South Asia as well. So this is the grim reality behind the situations of the fern flora of South Asia. And so in these two, in this agreement with these two statements, we all know that our species are largely concentrated in montane forest as well as the lowland rainforest. Right? For example, in Mount Kinabalu in Borneo, so it provides habitats over 600 pteridophyte taxa. It's a big number. And also in Western Ghats of India, over 320 taxa. And Singharaja World Heritage Site in Sri Lanka, in a very small forest block. It occurred nearly 150 taxa, right? The interesting thing is it is more than 1/3 of the island fern population. So, but unfortunately, we all know that montane region and also lowland rainforest have a number of vulnerable ecosystems, right? And also they are highly subjected to rapid rate of the deforestation. So in this graph, you can have an idea on rate of deforestation in global scale, and then second map, you can see that the rate of deforestation in Southeast Asia from 1970 to 1990 in a regional scale. And last map, it shows the change of forest cover from 1956 to 1999 in Sri Lanka, that is local scale. So it implies that severity of habitat degradations of ferns and also changing the microclimatic of ferns, right? So number of organizations, number of institutes have proposed number of strategic plan for the species conservation. So they have developed the different protocol, right? They have developed the conservation strategies. Yes, of course, those are fine, but every country should have their own strategic plan and locally adaptive solution mechanism for conservation of their species. So whatever the plan developed, whatever the mechanism established, we should adequately address and necessarily address to the three main components of the conservation cycles. That is use, study, and save. Okay. Now, I will briefly explain each element of conservation cycle in relation to the Asian ferns. Okay, the first element of conservation cycle, study. Right. So it is well known fact that current teridological studies are more towards the genomic analysis. And also most of the Asian countries, so including our region, we are working on diversity analysis and some classical taxonomic works. But whatever the study we did, whatever the study we conducted, the ultimate objective, the final outcome should be coupled with the conservation of these species, because the ultimate objective should be the conservation of these unique gene pool in the world. Right. So the question is, do we have enough information for the conservation of these plant groups against these growing threats? No, I don't think. I don't think we don't have a basic or mostly the basic requirement, basic information to require for the set the conservation and management guideline. For example, we don't know about the exact distributional patterns of the many species, that is the distributional ecology, and their unique habitat characteristics, the population dynamics, reproductive biology, how they reproduce, the pattern of the reproduction, response to climate change, changing climate, and for spore disposal mechanism, etc. Right? Because those are the basic need, basic requirement to use in preparations of conservation plan for each and every species. I know that it is a big challenge. It is a big challenge. These areas are not in a research agenda of the many countries. They are not highly demanded research, and who can fund for this type of the research? But if you want to prepare a comprehensive and meaningful conservation plan, of course we need to provide these basic information. So I will draw your attention on one of the realistic, couple of the realistic examples. In here, you can see the Scrv Schemes, commonly known as Scythia crinita. So why it is endemic to Sri Lanka and South India? Why it is common in high altitude area of Sri Lanka? Why it is rare in India? So do we know the exact ecological requirement of them? And do we know their reproductive biology? So then if it is not, how we can prepare the management plan for that, conservation plan for that. The next example, Erythrophylum sinuata, Scythia sinuata. This is the only known simple leaf, simple frond tree ferns in the world. Right? Why it is confined to Sri Lanka? Why it is restricted to the southern lowland rainforest? Why it is confined to the only 2 or 3 lowland rainforests? Why it is confined to, why it is restricted to the few populations of that species? So we don't know their exact requirement. We don't know how they reproduce. So we need such basic information for the conservations of such species. And another two examples here, Erythrophylum Accumulator and Lincia pectinata. In Sri Lanka, these two species are only known from single population. Why they are sustaining in a single population? So we don't have an idea, right? We don't have an idea. So what I'm trying to say here, in the future research focuses, we need to think about this area as well, because this area should directly influence on the preparations of the conservation guideline. Not only that, as I earlier mentioned that Asian ferns show a very interesting phytogeographical abilities and has a very interesting biogeographic history. Right? Such informations are also useful for the conservation aspects. For example, as Holden suggested that South Asian, particularly Sri Lankan tree fern shows are very closely resembled to the African tree ferns. But later, Thomas Jensen and his group identified that such groups do not show any relationship with the African counterpart. It is a monophyletic group and a highly isolated gene pool in Indian Ocean. So these are the very, very interesting information. So in here, what I am trying to say, in a conservation cycle, under the study component, we need to explore almost all information related to the particular species, because such information are highly useful for the conservations, right? Then the second element of the conservation cycle is use. So someone can claim that conservation and uses are contradictory to each other. But no, the concept is conservation through utilization, right? The conservation through utilization, right? Actually, we all know that for example, we cannot conserve these biological components just like a conservation of archaeological site, because these are highly dynamic, highly changing with the time, with the climate and other external factors. Right? Therefore, sustainable utilization is one of the options. So we all know that unlike seed plants, ferns do not have a high economic significance, do not have high economic importance, right? But ethnoteridological study provide wealth of information on potential use of ferns. Maybe food value, medicinal properties, and ornamental. I think except India and China, other Asian countries have not been adequately documented the ethnobotanic, ethnoteridological information associated with the ferns. But I don't know in India also whether such informations were validated or not. However, documentations of such information is very much useful for its users. And there is a very, very interesting article. It shows, it was prepared by Victor Amoroso and his group in Philippines. It shows how the tree fern species used for livelihood development of the traditional community in southern Philippines. So it is very interesting article. And also in Sri Lanka, the ornamental industry, floriculture industry is dominated by exotic fern species. So we prepared a very good list using local or indigenous fern species which has potential value to develop as ornamental plants, but no one look at these things, right? This is the utilization. Then the third element, the last element is sale, right? The sale. Yes, it is very, very important, right? Rather than thinking or sticking on usual ex situ and in situ conservation method, we need a holistic approach to address this issue, right? And also, rather than thinking about the conservations of individual species, we need to think about the conservations of entire genetic diversity with their habitat. So otherwise, ex situ conservation will not be a meaningful one. And also, as I earlier mentioned that every country should have their own protocol, their own strategic plan to conserve their species. It should be a locally adaptive solution and also balanced approach. Because we know that we can hire the technology from other countries, but we cannot import the solution for our problem from other countries. So that is why we need our own develop the mechanism or strategic plan for the conservation, right? So this is the conservation utilization and study model of Sri Lankan ferns that we developed, that we proposed this model. So actually this model is highly country specific, right? Highly site specific. Because the Sri Lankan situation is completely different with the other neighboring countries. In here, we don't have a pteridophyte research culture. There are no any active or full-time pteridologists working here. So therefore, we are giving much priority for international collaboration and data sharing in this model. So, however, similarly, every country, every region, they should have their own mechanism, their own strategic plan and model to address this issue. Right. So then, the final slide, that is the role of conservation biology and conservation biologists. Yes. So whatever the plan we develop, whatever the mechanism we establish, conservation biology and conservation biologists have a big role, right? Because they provide required information for policymakers, decision makers, administrative bodies, and also conservationists to set conservation guideline and to prepare a management plan. So in the future, we need to focus on our focus on this conservation biology aspects of the pteridophytes. So because it provides all the useful information for future conservation aspects. And also we need more conservation biology, same as molecular taxonomies. Okay, that's all. Thank you for your attention.
Thank you very much. Thank you very much, Dr. Rani Rajabaksha.
Yes.
So, please interact with our participants.
Okay, sure.
Okay.
Yes.
So yes.
Yes, good morning, Dr. Anil.
Hello, good morning.
Yes, I am R.R. Rao from India.
Hello, hello.
Yes, I have my doubt with regard to the biogeography of ferns.
Yeah, yes,
See,
sir.
in my opinion, the biogeography of ferns follows completely a different path when compared to biogeography of flowering plants. This is my
Okay.
experience with regard to the Himalayan flora I am talking. Now, roughly 50% of the Himalayan ferns are in South India, Western Ghats, and maybe 1/4 of that in your Sri Lanka. Now, all these are the result of Pleistocene relics, or is there a, they are all vicarious species? Still I do not have an answer for this. If 50% of the Himalayan ferns are in South India, we don't know whether it is Pleistocene relics.
Yes.
So this biogeography, I think, needs to be, especially for ferns, needs to be thoroughly investigated. Similarly, when you see the Himalayan flora, the flora of Nagaland, flora of Meghalaya, flora of Sikkim, more or less follows the same pattern with regard to ferns. Whereas in the flowering plants, the flora of Nagaland is more closer to Sikkim rather than Meghalaya, which is only 160 kilometers. So this shows the biogeography of these two groups are completely different. I don't know how you comment on this. See, because when I visited the Animity also, your Adams Peak, large number of Western Ghats ferns I could see there also in your, all the Western Ghats ferns were there. How is it? Is it because of the maybe Gondwana connection, or is it because the Pleistocene relics? I do not know of these things.
Yes, I agree with your views and idea. Actually, I just present here that is some of the hypotheses given by the different people, different persons. So that is why I suggest that we need a comprehensive study on the to identify these biogeographic relationships. Because that in one of my slides, I very clearly present that. So based on the Holton's, his hypothesis was Sri Lankan people shows a very close affinities with the African counterpart, African elements, right? In morphologically, he was able to identify some of the relationship between these two. And also then later, it was deeply studied by the one of the group from the Natural History Museum, the Paris Herbarium, Thomas Jensen and his group, they identified that there is no such relationship, right? So similarly, there are a number of hypotheses, and we have to do whether these studies in details using the present molecular or whatever the other technologies. So other than that, we cannot say there is a strict relationship with this and that, and this like that. So for that is why my basic idea is we need to focus on this area in the future.
Thank you.
Yes. I would like to add a couple of examples here. So one of the fern species, Aspinium gardneri, right? It is only found in Sri Lanka and Sumatra, right? Sri Lanka and Sumatra. So how we can explain these type of the things, right? Rather than just based on the morphological and ecological characteristics, we have to go for genomic analysis and other things for the validations of the such hypotheses. So it's, this is the things I want to highlight it here. So future studies.
No. Maybe a case of polyendemic you are talking. No, only Sumatra and Sri Lanka only you said. It may be
Yes.
it may be a polyendemic. Originated in two different places simultaneously because of this similar selective pressure.
Mm-hmm.
Yes.
Hello, Dr. Ranil.
Hello. Yes.
I am Dr. Irdhiraj.
Okay, right. Nice to hear from you.
Sure. I am the
Yes.
president of Indian Fan Society.
Okay, yes, I can
Okay,
remember
no doubt.
you very well because I had a nice time in your institute before 10 or 12 years ago.
Yeah.
Yes,
Yes.
I can remember you very well.
Very good. My major doubt, among the species which are confined to South India and Sri Lanka,
Yes,
there are several species which are not threatened in South India, but become threatened in Sri Lanka. When I wrote the thesis for my last PhD student, I came across such a concept. That is
Yes.
several, those species which are confined to South India and Sri Lanka,
yes,
several species are not threatened in India, South India, but threatened in Sri Lanka. What
Yes.
may be the reason?
yes.
Any
Of course,
anthropogenic reason or climatic change or forest depletion?
Yes. We also can suggest a number of reasons for that. But to validate these things, we need a detailed study. That is why one of my questions, when I show in the photograph of Saithya crinita, I said that it is very common in high altitude area in Sri Lanka. It is very common, but I think it is quite rare in South India. Right? Still, we don't have enough information to give the exact reason for that. Because when we are describing ecology of the particular species, we are always talking about the temperature, rainfall,
Yes,
moisture,
yes.
soil,
Moisture,
and
yes.
elevation.
Come to the
So,
point.
other than that main factor, there may be a number of unknown factors
Yes,
which
yes,
decide
yes.
the ecology and habitat requirement of the species. So that is why I said that we don't have enough information for
Yes,
the,
yes.
yes.
For the time being, I
Yes.
have hypothesized that mainly, the reason may mainly the requirement of maritime moisture and land moisture for particular species. That is, when you consider the South India, southern part, there are 3 sides sea coast. But when compared to your Sri Lanka, although it is large island, when compared to the tip of South India.
Yes.
That is a major factor for, as far as my knowledge is concerned, my experience is concerned. So we have to confirm with more studies.
Yes, I also agree with that because it is well known fact that the island populations
Yes,
have more
getting
risk
depleted.
to extinction than the mainland population, right?
Yes,
So
yes.
it
But-
only may be an environmental reason.
But in the meantime, Lincia species are more in number in Andaman Nicobar Island
Okay.
and South India.
Oh.
So they require more maritime moisture. That is in such a way we have to compare and study. Okay, thank you very much.
Yes. Thank you, Dr. Irdhiraj.
Hello, Dr. Ranil. I am Dr. Raju Anthony from Tropical
Yes,
Botanic Garden and Research Institute, Tirunelvelim, India.
Right. Okay.
And I think that there is a more chance for, in island flora, there is a more chance for endemism. First, I want to know that how many endemic species you have. And regarding Saithya crinita, the main reason for becoming rare in South India is mainly due to overexploitation. And in South India, nowadays, Saithya crinita are mainly seen in Mannamanchola area. That's in Idukki district. It's a high altitude area. But what happened that there are chance of habitat destructions and over exploitations. And tribal people in Mannaman, particular tribal people in that area, extracting the leaves for thatching their huts, etc. That is also a reason for becoming endangered. But you are saying that it is very common in your area.
Yes.
And we are saying that so many species are endemic to South India and Sri Lanka. My opinion is that we have to need a detailed study because there is a good example for us that Bobitis subcrinata, we studied that it is endemic to South India and Sri Lanka. But the recent studies it is found that it is not a subcarinata because subcarinata having a broader sporophyll, and in our plant it is a narrow sporophyll, that is a terminans. And Fraser Jenkinson published as a new species of Volpetis subcarinatoids. And later he found that it is not a subcarinatoid, it is a already published one that is Volpetis terminans. And earlier it is known as a different name, but it is elevated to now Volpetis terminans. And is there any chance to collaborate your work for, we have to study the South Indian and the Sri Lankan endemic ferns. If you study well, there is a chance to get to new species.
Yes,
Okay.
of course. Yes, thank you very much for your idea. So actually, in the first questions, you raised that how many endemic species in Sri Lanka. Actually, it was earlier 47 species, which was described in Flora of Ceylon. But very recently, we compiled a Sri Lankan fern list with the support of the number of international terminologies. So we reduce it up to 42, right? 42. And also, I know that the Fraser Jenkins, he has developed a list for the Sri Lankan and South Indian endemics. Yes, as you mentioned, that's there are a lot of similarities, and we can come up with a new knowledge, new findings with the collaborative work. So of course, I like to do some collaborations. So as I earlier mentioned that in here we don't have a territorial research culture. So I am also a by profession lecturer. I am teaching on tropical agriculture, but not of the ferns. Ferns is not in our research mandate. But still I prefer to collaborate with some other people and do some collaborative work with the betterment of the field of heritology.
Dr. Renil, 42 is a good number. And in India, earlier we believed that there are more than 200 species of endemic species in India. But as per the recent studies, it is found that only 50 or 56 species are endemic to India. And Sri Lanka, you have a good number. It is 42 when compared to Indian ferns. It is a good number.
Yes, when compared to the land size as well, right. And also the total number of species is 389. So 389, we confirm that number, but there are a number of doubtful species as well.
Okay. Our next speaker is waiting, so thank you very much, Dr. Rajbaksha, for your enchanting lecture. Thank you very much.
Okay.
You have a beautiful garden in Paradinia. Yeah. So you are
Thank you very much for giving the opportunity to share my knowledge with you all. Right?
Please keep in touch. Thank you.
Okay, thank you.
Thank you. So our next speaker is waiting, Professor Harold Schreiner from China. Please conduct.
Sorry, now my talk with you online. Now I have to stop talking. Okay. Okay.
Hello. Hello. Professor Schneider, sir. Hello, Professor Schneider, sir.
Hello.
Hello, sir.
Professor Schneider, can you hear me?
Yes, I can.
Very good morning. Welcome to Calicut University,
Morning.
Kerala. Welcome to Calicut University, Kerala for the International Symposium on Research in Technology, sponsored by the Indian Ferns Society. Can you hear me?
Yes, I can hear you. Yes, very nice, clear.
Yes, welcome to Calicut University. Now, I invite Dr. Santosh Nambi to give an introduction to you. Please.
Yeah. Okay. Now.
Hello. Hello.
Yeah.
Hi. Good morning.
Hey.
Good morning.
Good morning.
Nice to meet you again.
Yes, long time ago.
Long time, almost 25 years back. Almost 25 years back we met at Leiden. Hello.
Yeah.
Yes. Can you hear me?
Yes, I can hear you.
Okay. So very good morning. I remember the days we spent together at Leiden. And in fact, we want you to be here at physical mode. Okay. Now, because we are running short of time, we move on to directly introduce you. And now we have Professor Harold Schneider. He is presently the professor at Tropical Botanical Garden, UNAM. He took his PhD from University of Zurich in 1996. Then after, he had 2 years of work at Leiden Herbarium, Netherlands. Then in different levels of teaching as well as research, he worked in Field Museum, Chicago, University of California in 2000, then Duke Herbarium in USA in 2001, in Germany in August University in 2002 to 2006, then Natural History Museum London in 2006 to 2015, then in China from 2015 to 2017, and now in Yunnan from 2017 as professor there. And he has more than 2,200 publications and over 17,000 citations with an H-index of 60. And we know he is also one of the major contributor of the PPG system of classification. And he has a lot of publications in the major journals like Nature, Nature Plants, Proceedings of the National Academy of Science, Current Biology, Molecular Biology, and Evolution. So in lot of leading journals, he has published many publications. Now, because of shortage of time, I invite Dr. Scheiner to move on to his presentation. Thank you.
Okay, thank you very much, Professor Nambi. Now, see if this one actually works. Do you see my screen? Okay, let me see. Let's try it again. Oh, I see that you haven't enabled to share my screen. Okay, now most likely it should be working. Yep. And now see if this one works.
It's working.
Okay.
It's working.
It's working now, yep. Okay. Good afternoon, or good morning, or whatever works. It's a great pleasure to me to be here and present my talk. And thank you very much for this kind invitation. And I know it's a little bit challenging sometimes with the internet. I'm Professor Harald Schneider. I'm leading the macroevolution group at the Shishibana Tropical Botanical Garden an Institute of the Chinese Academy of Sciences. I'm very, very grateful for this invitation, and preparing this presentation I was going back to the beautiful memories of the time where Professor Nambi and I were both in Leiden and enjoyed this beautiful town and the beautiful and fantastic colleagues in Leiden. Now I'm professor in Shishibana Botanic Garden, which is located in southwest China in the most extreme corner from Yunnan, close to the border of Myanmar, Laos. It's a very small area. We are one of the most astonishing botanical gardens in China and the world. Really a beautiful garden to visit and to live in. We are an international research institution which belongs to the Chinese Academy of Sciences, and maybe you know that where we are several international faculties in Shishibana. We are located in the northern end of a biodiversity hotspot, so it's Indo-Burmese biodiversity hotspot. So we are sitting in a very rich tropical flora, and we offer advanced research facilities in, you see here the grain, which we for clinical work, therefore we have the fantastic research facilities. My research group is studying plant diversity evolution over past and present, with perspective in the future, with focus on how plant biodiversity will look and support the ecological civilization. We have high access quality research facilities and actually really beautiful, fantastic research setup. The team, which is rather international, which includes Associate Professor Liu Hongmei, which is from China, Assistant Professor Pio Kaikine, which is from Myanmar, and we had postdocs from Japan, Tao Fujiwara. Current postdocs include people from Zimbabwe, Kenya, and India. Similar to the PhD student, master students are rather international. And if you are interested in this location, think about and want to join, please contact me. We have still an assistant professor position open, and we have always training opportunities for postdocs, PhD students, master students open. Anyone interested in plant diversity, especially ferns, are very welcome to apply. Today my talk will be on more or less summarizing some of the insights for a study on the evolution of fern genomes, which is now online available on Annals of Botany. This was a postdoc project of Tao Fujiwara. Tao is now back to Japan in his home countries. This has a large number of co-authors internationally, including people from Argentina and UK, and I want to express my thanks especially to two long-time collaborators, Professor Liu Hongmei, who is actually one of the leaders in the plant genomics research efforts now, and of course, Lincia, one of the very good old friends in Kew Gardens. Whole fern genomes are maybe arguably the mighty mountains of fern biology. Therefore, we expect to find very, very important insights when we study whole genomes of ferns. But currently the access is a problem, which is maybe not so surprising given the situation that whole genomes are only becoming available frequently since 2000 when Arabidopsis thaliana was published. Now we have more than 800 plant genomes available covering all lineages of plants. It's amazing progress. But if you look into the phylogenetics to it, here's a phylogeny of plants we've shown on the family level. You have the hornbirds, liverbirds, mosses, lygobirds, ferns, and then the seed plants. And when you have a red line, it means that we have not a single terminal genome for this family. Only when we have black lines, we get families, we have at least one genome. Therefore, you see still a lot of gappiness. And if you really want to read in a kind of comparative phylogenomics, the sacred history of this life, which is written into this genome, we have to expand the sampling. But actually, the things looking rather good. We got 3 identifiers already, 2 Genera and 1 Isoedes recently. Then we got 2 ferns, heterosporous ferns, Azolla and Sardinia, and draft genomes Erythrophylum. And several genomes are in preparation, will become available soon. Using this data, we can actually look what is going on, and we actually see a lot of interesting patterns. But the 2 repeated patterns appear that whole genome duplications are critical, and we find very interesting patterns in the non-coding DNA, which are distinct ferns from angiosperms. However, the sampling density may be not sufficient to do macroevolutionary patterns yet using whole genome sequences. Therefore, we applied a different approach. We looked first to genome size, so-called DNA C values, which you can obtain by cytofluometry, a relatively cheap method, which is a little bit time-consuming and requires a lot of expertise by the person who's doing it. Then we use chromosome numbers that has a long history, and a lot of chromosome counting has been done. Still, a lot of counting has to be done, but that's actually rather good. And when you combine the 2 values, you get another evidence. You can see chromosome length, which is here estimated as the average amount of DNA per chromosome. Rather nice, simple values. And you see when we started to do that one, the initiation were other previously published studies, and I want to point out only Clark et al. 2016, which was published in New Phytologist, which was a study led by me when I was still in the Natural History Museum in London. James Clark was at that time a master's student. So despite we had very limited number of genome size data, we were able to show that the different lineages of ferns show very distinct trends in the genome evolution, measured by genome size variation and chromosome number variation. Tao Fujiwara took it on to expand the data set by doing a lot of new genome size measurements, and by doing so, he doubled more or less how much species we have. Only the gymnosperms actually are better sampled now than the ferns for genome size data, but we still have to be aware that we have some limitations. There was a variation, also orders varies a little bit. Filmy ferns is still an order which needs much more data. We have only roughly 3% of the order sampled, whereas the horsetails are very good sampled. We found a huge amount of differentiation. When you look to the smallest versus the biggest genome, you see how much genome size differentiation actually exists. We were able to confirm what we have known for a long time, that or for some time, better I say, that the chromosome number and the genome size is correlated in ferns. Is you do this one with partitioning in different lineages, you always find pattern. You realize that when you look to the dots, that the actual measurements show some variation which is not explained, but only ferns and lycophytes actually shows this pattern. Seed plants doesn't show that pattern. In summary, what we find is a huge variation of genome size disparity amongst the lineages of ferns. And I want to point out a couple of things using this order structure. You see when you look at this figure, you see the orders and you see the size variation, which from a small to a big size. You see some groups have very small size, some get very big as Pseudocyclosaurus, whereas some groups have very little variation. One of the groups which we're always pointing out is the heterosporous ferns, which have long been at the time recognized to be divergent from other ferns in the genome evolution, and they show a reduction in genome size and chromosome number. And actually the Lee et al. Paper took advantage of that one, that their genomes are a little bit cheaper to sequence because they're smaller. But interestingly, despite they have the trend of a small genome size reduction and reduction of chromosome number, they show still several whole gene duplications. And in this way, they are very similar to angiosperms, where you have repeated whole genome duplications, but downsizing of the DNA afterwards. Very different example here is Osmondelis of oil ferns. It's a paper we published in 2015, which was motivated by the discovery that some people found fossil nucleus, this is this picture inserted, which is about 200 million years ago. And when you look to this nucleus and compare it with the modern taxons, they have the same genome size. And actually what you see in the oil ferns, that you have genomes frozen in time. So you have nearly no genome size variation, and you have absolutely the same chromosome number fixated. And the chromosome of Osmondelis are a little bit unusual for ferns because they're a little bit bigger than other fern chromosomes, a little bit larger. But interesting what we find in when you look to this group of ferns, they have genome stasis, or the genotype is in stasis, phenotype is in stasis, and in total we find low diversification rate. That's meaning the rate how new species coming in, how is the rate how many species going extinct. And that's actually interesting. We have here a long time survival model. And you may be familiar with the Red Queen, which Van Walen introduced, where Van Walen argues that you have to change all the time to survive. But that's what these taxa are not doing. They seem to be very conservative. And what you actually could see is a pattern which is much more so what Mike Benton, a good friend of mine who's a paleontologist working on large-scale patterns, he argued for cultures that you can be conservative as long as you have the same habitats all the time. Ferns have also other strange things. Here, another study which published a couple of years ago, which we call Genomic Gigantism and the Fern family. The taxa which we studied was Bicipterus obliqua, which is an epiphyte in Australia. And what you actually find is that you got very, very large genomes. And actually, this is the second largest genome, which is nearly as big as Paris japonica, which is a terrestrial monocot in temperate regions. In general, Pseudotales show a trend to have large genomes. Also, Pseudodium has a relatively large genome. Interestingly, Pseudotales are sister to Ophioglossalus, which are the host of the record holder of the largest number of chromosomes ever counted for any plant. This Ophioglossalus reticulatum, which have about 700 chromosomes per haploid genome. And therefore what you actually find is an interesting trend that the two sister taxa show a trend of gigantism, one more in genome size, as Pseudotales, and one more in chromosome number, which is Ophioglossalus. We reflected on the pattern that ferns may be actually really interesting when these large fern genomes, therefore, when you look, there's very large genomes, therefore this bar shows that the genome size from very small to large. Very large genomes are very rare, so you can only find in a couple of angiosperms, a couple of ferns, and a couple of strange vertebrates. But actually, maybe only ferns provide us really the sample density to look to answer some of the key questions, especially about the mechanisms to keep very large genomes stable, which is actually really one of the challenges. In summary, what we see when we look to all the orders of ferns, and I restrict myself here to the ancient fern lineages, for example, is that we find distinct trends. Horsetails, Equisetalis, large-sized genomes, medium to large size, large chromosome number. The chromosome number is totally fixed, but genome size not. Ophioglossalus, extremely accumulated of medium and large size genomes with very large chromosome numbers, a lot of variations there. Pseudotales tended to accumulate very large chromosome and genome size, and Osmodales, royal ferns, I talked about stasis already. So Martiales are interesting again because they show some variation, but mostly maintain small, relatively small, medium-sized genomes and chromosome numbers. I could continue on all the ferns, but I want to restrict myself here and ask questions. What is the factor why we have this differentiation? One factor could be ecology, something outside, or it's actually differences in the genomic processes, mechanisms shaping this accumulation. Glimpse to that one can be found in the literature. Here in one study which done many years ago by one of my PhD students. Therefore, we showed that upper mixes, meaning the change from sexual to asexual reproduction, seem to be linked to genome size expansion. Clear trend. Do we know why? No. Another beautiful, fantastic study, and actually one of the most important papers published on this topic in the last year by my associate professor Liu Hongmei, published in Journal Systematic Evolution, where we actually looked not only to genome size variation in chromosome number, but also to the chromosome length. And what we actually found that chromosome number doesn't explain everything. We look to Asplenium as a core example, but also to Polyproteus in general. But actually what we also find, and this is the thing which need to be highlighting, is here geographic patterns. So for taxa which occur in climate season, in areas seasonal climates tend to have smaller genomes. More data and data evidence need to be done to see if this is consistent. But we see a trend. But why is this the case? Need to be explained. Now, finally, I come to topic where a lot of people would be interested in polyploidy. Polyploidy is English again recognized an evolution and ecological force. I copy here a title from one of Van der Peer's papers. And we asked people ask again, how important are whole genome duplications in the accumulation of diversity of the tree of life? Several studies use transcriptome data to look to that one, and I here use Huang et al. 2019 is arguably one of the most comprehensive ones for ferns. They reconstructed the phylogeny of ferns using transcriptome data representing most of the fern families. Very good then sampling. The phylogeny had very few surprises, but what's the most important they were able to look is evidence where we find evidence for whole genome duplications. And actually interesting, we didn't find too many in the derived ferns, which is actually surprising given some trends there. One of those took one step further because they said polyploidy is a force, therefore if this is true, we should see shifts either down or up, no, shifts correlated with whole genome duplication events. And actually, they found some link, but it was not very strong. And therefore, I would say it's non-ambiguous if between whole genome duplication shifts and investigation rate shifts. Before the Fujiwara study was now taking this one in the center. We argue a little bit broader, we say it's actually what the whole genome duplication is only one aspect. We looked at the whole dynamics of the genome changes. We accumulate the rates of changes for genome size expansion, which is mostly the consequence of whole genome duplication, contraction, which is a consequence of diploidy and related processes. Or we looked also to rates of change in structure of the genomes using chromosome length expansion and contraction as evidence. And that we correlate with diversification rates. As a reminder, diversification rate means balance between new species appearing by birth, speciation rate, or species going extinct, death rate or extinction rate. And therefore, if your diversification rate goes up, this means either by increased speciation rate or decreased extinction rate. For what we actually found, and this was really groundbreaking result, that we find when we look to the rate of genome size evolution, that is correlated with species diversity evolution. So for what you see in these figures is the upper one is a log, on the y-axis the log number of species, on the upper lower one is a diversification rate, or as a log, and then we compare this, look to correlation to three factors: change of holoploid genome size, means the whole genome; monoploid genome size, that was a way to see that we reduce the effect of neopolyploids; and then look to the chromosome length variation. And what we find in all, we find a positive correlation, which was often highly significant. In summary, what we actually see is that genome size, or genome rate of genome evolution is arguably closely linked with the rate of speciation or diversification rates. Therefore, for the first time, arguably, since I am working on Ferns, we got a glimpse for a reasonable answer to one of the questions which have never been properly answered. If we look across the phylogeny of ferns, we find variation in species richness. I'm looking here to the number, Equisitates have, I use PPG1 as a reference to be consistent. Horseates only 15 species, and whereas polypodiales have more than 8,000 species. None of this variation can be explained by lineage age, and as Huang and al. Showed, it cannot be explained by whole genome duplications alone. But what the Fujiwara study now says that the dynamics, the rates of changes in of the genomes may actually allow us to explain that pattern. What does actually genome change mean? It means that you reorganize the information. You change your genotype to be able to change your phenotype. And now we're coming back to adaptation. Therefore, able to change your genomes fast may allow you to adapt to respond to change. Now, final conclusion on all that would be Ferns supported Charles Darwin. I come here with one of my favorite citations of Charles Darwin. I have a lot of Charles Darwin citations which I like, but this is really one of my favorites. He wrote on this topic related, it is not the strongest of the species that survives, nor the most intelligent, but the one most responsive to change. Really, he focused that the response to change is critical in speciation and survival. And Charles Darwin had at this time no information about genomes and genome evolution. Now, our new insights in genome evolution start to resolve one of these questions which evolutionary biologists were hampered for centuries. With that thing, I want to thank you for your attention and for listening to me. I hope that my talk gives you some ideas about what this mystic genomes of ferns will allow us to answer when we understand them better. I hope I removed a little bit of the clouds here. And with that one, I thank you for your attention and wish you all a very successful meeting and very productive discussions and good health. Thank you very much.
Thank you very much for our inspiring lecture. Please interact with our participants.
Here it is, good morning. So it's a beautiful slides actually, and thank you so much for conveying those evolutionary ideas for the young brains. My question is there any correlation between the genome size and the distribution of the ferns? Is it affecting?
It's a good question, and I think it needs a bit more attention. Yes, it exists some trend. The trend is not so strong that I could say that's the pattern. But what you see, and I can illustrate this on one slide, when you go back to the data on the paper, in the paper of Liu Hongmei and published in XVI, you see that actually some lineages show a trend to have smaller genomes and some lineages have tend to be bigger genomes. When I look to Asplenium, that is on the left side, what I immediately see is that temperate taxa tend to have the smaller genomes, and the tropical ones seems to have the bigger genomes. If I look to Triopter I have the same feelings. When I look to Triopter because it's mostly sampled here, that's the figure on the right side. If I look to Triopter, the Polystichum tend to have smaller ones. If I look to Erythrophylum, now we go back to tropical one, getting bigger. For simply temperate versus tropical. In the Pteridaceae, that's not obvious. In Pteridaceae, they tend to be always small except the epiphytes. But Pteridaceae, as you know, often occur in seasonal stressful environments, that could be the reason. We see also some links to epiphytes or non-epiphytes. That was a little more complicated, but I think something is there which needs more data, I would say.
Thank you so much. A humble question, one more from a novice in this field. What is the method, for example, are you people are going for a hybrid sequencing or you're depending on single platform for sequencing?
There's so many ways now to do sequencing, and it's very difficult to say which one of the sequencing platforms is the best, and which analytical platform is the best. Basically, this really depends very much on the question you have and what you want to answer. You know, for some of the questions I have, I need to read a complete whole genome. That's expensive and time consuming. But if you really want only to know, if we are interested, for example, do we have gene flow between two related species, then you actually make your life much easier. Therefore, you only need skimming some of the autologs. So you actually can use either a transcriptome data set, which requires you to work with RNA, or you do a skimming data set and fishing for the autologous genes. And that is actually not so hard. Needs a lot of, it's actually sequencing cost gets not too big, but it's really a lot of bioinformatic tool lines to clean up the data, but it's extremely powerful. You know, in the past, what the shift is, in the past when I was teaching students, they spend a lot of their time in the lab to generate data. Now it's actually, most of them, we send up the material to genome sequencing companies, they do the data, and we sitting on the, my students sitting on the computer have to learn how to manage data. That's actually the biggest difference.
Thank you. Thank you so much. Thank you.
Well, Professor Schneider, I am SP Kuller speaking. I'd like to have an observation on a point that we have two primitive groups of ferns, the Osmondaceae and the Ophioclausaceae. They are both very primitive. In the Osmondaceae, the chromosome number is, say, n is equal to 22. That's the gametic number. And the same gametic number has been found in the fossils also of Osmonda cretoniana, right? Now, in the very, what you call related ancient group of ferns, the Ophioclausums, the chromosome number has multiplied so many times. What could have been the reason for this, although both of them have existed almost simultaneously, you could say? One group, pardon me, one group remaining static at a low chromosome number, the other multiplying unlimited.
It's one of the really interesting questions, and I think one of the interesting points here I want to make, coming back to the slide here. Really interesting one is, because it's a very interesting point, that the different ancient lineages of ferns show very different trends in species richness, but also how the genomes are changed. And Osmondaceae, when we look from the modern point of view, they are very conservative. Their morphology is rather conservative. You know, the classical example for morphological stasis, when you look to fossils out of Triassic, or which have more or less the same leaf shapes as modern ones. But for what actually this lineage seems to do is, that's why I look to this Mike Benton was writing about long-term survival and stability. This group seems to find a strategy where you're not changing it or be slow, a kind of evolutionary slowness strategy. But they always find this habitat which they survive in. And you see, when you look to the ecology of Osmondaceae, it's rather interesting. Most of the Osmondaceae which we know are growing in forest habitats, wet forest habitats, in temperate regions, but also sometimes in the tropics, but not too much of the tropics. It's mostly Planacium. Then you have Leptopteris, which is the most emerging one growing in New Zealand. But most of these ones are morphological rather similar. Therefore, not too much change. Would argue Ophioclosaceae are the total opposite. Ophioclosaceae, especially when you work as a temperate person, this is Ophioclosaceae coming from Botrachum, Sceptridium, you see that these ones going in alpine meadows, in grasslands. These are young habitats, they're now typically angiosperm dominated young habitats. This taxa need to change quickly, therefore they're adapting to something. And if you look to Ophioclosum itself, therefore Ophioclosum is also adapting very quickly to small changes in the habitats. And I think what we see is that this in the Ophioclosaceae is rapid change, trying to speed up, keeping going on with the change. Therefore, they are much more what I call a red wing. Therefore, they are more trying to move with the change of their habitats. Therefore, what you actually see is I think these two groups are really in the core of the arguments. Whereas the Osmundaceae keeps more diversity, but don't change too much. And they've always tried to stick more or less to the same ecological, the same adaptation. So Ophioclosaceae goes opposite. They try to do strange things. They try to adapt. They change. And you saw Ophioclosaceae have very strange morphology, very unusual for ferns, very strong differentiation. And you say Ophioclosaceae in old lineage. One of the challenges of Ophioclosaceae is, yes, from the molecular dating, we say it's an old lineage. But look to the fossil record. We assume that the Ophioclosaceae and Pseudocyclosaurus originated somewhere in the late Paleozoic or early Mesozoic. But we have no fossil record for that. And I think the reason for that is that these things change so much on their phylogeny that we have a hard time to very challenging time to recognize the fossils of that groups. And that's actually where I think where this group is very interesting. Something real weird going on, very changing on this group.
Well, may I add to what my query was? Both these groups of ferns, the Osmundaceae as well as the Ophioclosaceae, they have a limited number of species. Leaving aside the clutter of names that we have presently in the Ophioclosums, if we leave that aside, very few species in Ophioclosums, very few species in Osmundas, and you say that long-term survival without change in the Osmundas, but in just similar circumstances, we have the chromosome numbers multiplying, but not much change in morphology. One leaf, variable in shape and size, and bearing the sporangia. There's not much change either. It's only the chromosome number which is changing. Yeah, whatever.
Okay. If you push me down to Ophioclosum, I talked already of Ophioclosaceae because, but Ophioclosum is really a challenge. I agree with that. And I think one of the questions which come back to that one is when we have Osmundaceae, hybridization and polymorphization, we are driven by hybridization, seems to be very rare in Osmundaceae. We have now only one tetraploid example. We have a couple of things which could be hybridization between close relative species, but it depends on the species concept. I refer here to the problem of Osmunda chambonica versus Lincia. If they are really two different species, I don't believe that so clearly. But in the Osmunda, what you see is that hybridization between species seems to drive polymorphization in chromosome number changes. And this doesn't happen in Osmundaceae, and this is actually one of the limitation factors in the Osmundaceae. But in ophioglossaceae, that is a driver. And the question will come up, you're right, how many species we should recognize in ophioglossaceae, in ophioglossum. If you didn't trust morphology alone, we will not recognize many. If you think about using chromosome numbers as a key factors, we will get a couple of more. But I think that's maybe also still one of the interesting questions for integrated taxonomy to figure out how many more species there are. And one of the critical questions for me is, how old is this genus really? That's something I don't know actually.
Thank you very much. Thank you. Well, Professor Schneider, I'd like to introduce you to the Indian Fern Journal. The Indian Fern Journal is being published for the last 40 years almost, and I would like that you send your article for publication in the Indian Fern Journal. We would be very happy to publish it in that. You can send it to me. I'll send my email address to you. You can just email it to me, and we'll be very happy to have it in our journal. Thank you very much.
Okay. Thank you very much for the invitation.
Thank you, Dr. Schneider, for your inspiring and beautiful presentation. So one more question.
Okay. Thank you very much.
Dear sir, my question is related to Selaginella. And the Selaginella having basic chromosome number is smaller compared to other genera, but it is having more species. What is the reason for that?
Oh, wow. I was hoping that I can avoid Selaginella questions. Selaginella is a really interesting challenge in this way because something drives Selaginella genome evolution in a different direction. And Selaginella has a very, very small genome size, which is arguably linked to heterospory and a particular question of heterospory. And as long we are on, and I think Selaginella needs very, very careful work to understand what's actually driving here. And the problem when we look to rates is because rates is a relative thing, therefore it's more or less a proportion of changes. And in Selaginella what you see is a very limited change, but actually the species number is rather high. And I think what is actually driving is something similar, something the reproductive system drives speciation high up in this group. But this needs to be a very careful study, and I currently have no, I can speculate, but this is the only way I can do it. Therefore, more work to do for people that do the Selaginella genomes to understand what's driving Selaginella genomes.
So thank you, Harold Schneider, for your inspiring and beautiful presentation. Please visit Calicut University.
Yeah, I will come.
Yeah. Please come.
Thank you very much.
Hearty welcome.
Thank you very much.
Thank you.
Bye. Bye.
So now we conclude the technical session 3 of this symposium. In technical session 3, we have 3 online presentations. First one by Dr. Susan Forsyth from California, USA. Second by RCG Ranil from Sri Lanka, Paradena, Sri Lanka. And third one by Harold Schneider from Yunnan, China. Dr. Susan spoke on the taxonomic revision of the limited race. It's a phylogenomic data set. Dr. Sanjappa and Dr. Kudlar asked some clarifications, and she answered all questions conspicuously. Dr. Ranil Rajpaksha presented the paper Challenges and Strategies in the Conservation of Fern Flora. And he requested for implementation of regional and global strategic plan that is needed for the conservation. Dr. Raju Antony, Dr. R.R. Rao and Dr. Hridayaraj interacted with the speaker. And the third speaker was Dr. Schneider from China. He spoke on the evolution of fern genome, that is biodiversity in the past, present and future on the basis of the phylogenomics. And he said that the genome size or length, the expansion or contraction of the genome is correlated with the diversification rates. Change of genome enables adaptation. That is his conclusion. Dr. Kudlar and Dr. Leslie Augustine, from Tevara, and Dr. Raju Antony interacted. So this was a very, very live session with the three important papers presented by international trilogists. So I thank you very much, my rapporteur, Dr. Binoj Matthew, his humble support, and also the audience for your kind patience. Thank you all. I think now we conclude the third technical session. Thank you once again. I think now it is tea time.
and now he is handling the section of Department of Science and Technology which fund for FIST, and I know, I think most of you are aware of the FIST programs which is being, that is the Fund for Improvement of Infrastructure in Science and Technology for higher institutions, and he is heading that, and we as plant biologists are really proud that plant biologist is one of the top officials in Department of Science and Technology and handling one of the important sections. So with great pride, I invite Dr. Arindam to continue the session. Thank you.
Thank you, Professor Jose. It's my immense pleasure and privilege to be a part of this August gathering, and thanks to Professor Santosh Nampiji, the organizing secretary, and all the dignitaries on the dais, young researchers and fellows, and young students. So, this conference being organized by the Indian Fern Society. So, as we all know, importance of higher plants and even gymnosperms are much higher than as compared to the ferns and the pteridophytes. And also the number of researchers, we have very limited number of researchers in this field. So, just to keep in view that the interest of the researchers who are working particularly in this field, as well as researchers who are interested to, you can see from my humble beginning as a MSc in botany, then PhD in plant-insect interactions, I also got an opportunity to move into, other field, especially in the area of malarial parasite biochemistry and molecular biology. So it's all up to an individual's interest. If you want to move outside your domain area from the PhD days to further explore other areas. So you can also carry forward those areas as well as keep continuing to explore newer avenues and newer niche areas within your domain research areas of the PhD work or the postdoc work. So now, as I am representing a funding agency, the Department of Science and Technology, so I would like to apprise all the researchers and all the senior faculty as well as the mid-level faculties about the opportunities and schemes of funding that are existing in DST, and also the autonomous body of DST, the SCRB. All of you definitely will be aware of the SCRB. So SCRB actually, they provide funding for a lot of, mostly for individual-centric schemes and grants. And they have a lot of schemes that caters to different levels of facultyship or even, young researchers who are joining to initiate their career. So a lot of schemes are there for SCRB, catering to each level of faculty level. And thereafter, I will be also touching upon definitely some of the DST schemes, which will be, somewhat mission-oriented, and some of the schemes are like the one division that I am associated with is the R&D infrastructure division that provides support to develop infrastructure in departments or create S&T facilities that will help the entire department, depending upon the theme areas that each of the department or the college, like we under the DST FIST program, we also have support for the postgraduate colleges. So those are infrastructure-like schemes which DST is looking into. And under the mission mode programs, of course, there are also individual-centric projects that are given. So we have several funding bodies in the science and technology system in India. And as you can see in this slide,
DST is one of the major or the most primary science and technology funding agency, surrounding which a lot of other funding agencies have come up, like the Department of Biotechnology, in fact, even the Ministry of Environment and Forest, Ministry of Earth Sciences, then DSIR, they were all carved out from DST. And apart from DST, we also have DAE, then DRDO, then CSIR also funds projects and other schemes. Then the ICMR, ICAR, State S&T Council, and a lot of other bodies. But the primary agency that that actually hold the center stage is the Department of Science and Technology. In the year 2020, we completed the Golden Jubilee, DST being established on 3rd May 1971. So what are the core mandates of DST? Basically, we have a role to generate policies relating to science and technology. Then, as I mentioned, under the research and development capacity, we have human and infrastructural as well as institutional capacity building, extramural R&D funding. Then we have also schemes or programs under the garb of technology development, innovations, and startups. We also have international S&T cooperation-based R&D projects. S&T interventions for the needs of the society. We have one division fully catering to the funding that needs to be done at the societal level. Then we have statutory bodies like the SCRB, TDB, and as many as 19 autonomous institutes, DST autonomous institutes. Professional bodies, which includes the 4 academies, INSA, then Indian Academy of Science, National Academy of Science, and as well as Indian National Engineering Academy, and also the Indian National Science Congress. Then we have 3 international bilateral S&T centers, the Indo-German Center, Indo-French Center, and Indo-US Center. And through all these mandates, we make the nation future ready by providing schemes for S&T innovation, and we, through our other ministries like Ministry of Earth Science, Environment, Forest, MNRE, DBT, etc. So we have multi-stakeholder base, and to run this core mandate, we have socio-economic ministries, the youth, we have to support the researchers, we take care of women scientists, then scientists of the weaker segments, innovators, entrepreneurs, industry, and many other stakeholders we have. You can just go through the slides, go through the content of the slides. And so in the last so many years, that funding that we have been providing, so how that has helped the Indian S&T system. So this slide actually lists out some of the important progress that we have made in the recent years. So India's gross expenditure on R&D has increased by more than 3 times during the last 10 years. And our per capita R&D expenditure have got doubled in the last 10 years. Today we stand at 3rd position in terms of number of publications in SCI index journals, 9th position in terms of the quality, 3rd position in terms of number of PhDs that are being produced in the country, 3rd largest higher education system of the world, 3rd position in terms of number of startups, 3rd position in terms of number of unicorns that are being generated, 9th rank in terms of resident patent filing. We also have 48th rank in terms of Global Innovation Index that we improved from 81st position that we stood in 2015. We have got women participation in extramural R&D that got doubled in the last 6 years, and as many as 3.42 lakh R&D personnel, that is an increase of 40% in last 6 years. And India have moved up around 79 places in last 6 years in its World Bank global ranking in ease of doing business. So today under DST we have 3 major umbrella schemes. That is the S&T institutional and human capacity building, the R&D umbrella scheme, and the innovation technology development startups, the umbrella scheme. Apart from the 3 umbrella schemes, as I already mentioned, we also support around 19 aided institutions and also support the Survey of India and ATMO, these organizations. So little detail about the 3 umbrella schemes that we have, the S&T institutional and human capacity building. Under that, we support the schemes under R&D support like infrastructure programs like FIST, PERS, SAF. I will expand these when I come separately to these slides. Then we have the SATE S&T program. Then we have a policy research cell that basically generates different S&T policies as per the requirements of the country, keeping in view both the national as well as contemporary changes in the S&T, different S&T areas. Then we have the Women Science Program under the division of Kiran. We have Women Scientists A, B, C schemes. Then we have Curie program for women universities. For the human capacity building, one of the major programs, flagship programs of DST is the INSPIRE scheme. I think all of you must have heard about the INSPIRE scheme, under which we give fellowships, scholarship fellowships, then faculty fellowships, to encourage young researchers and to set up a career in, to encourage them to set up a career in S&T. Then also we have the Swarn Jayanti fellowships. Under the second umbrella scheme, that is the research and development, we have the international cooperation. Then we have nano mission and technology. We have mega facility for basic research, climate change program, national supercomputing mission, and the frontier and futuristic division, that is the inter cyber physical systems program. The third umbrella scheme is the innovation, technology, development and deployment area, under which we provide a lot of schemes under the technology development program, the TDT. Then we have technology missions division, which again have energy and water, as well as the other areas. Then we have S&T program for socio-economic development, like the National Entrepreneurship Board, National Science Communication, the Science for Equity and Empowerment Development, that is a SEED, and SCP and TSP. These are specially under these funding is given for the weaker sections of the society. We also have schemes under the drugs and pharmaceutical research, as well as technical research centers. So now I would little bit highlight about the SCRB schemes, that being individual-centric schemes. So SCRB, the most important program for an individual researcher is the core research grant. So the scheme provides core research support to active researchers to undertake R&D activities in frontier areas of science. The call for proposals is twice in a year, and that is notified through the website at subonline.in. And selection is based on the recommendations of Program Advisory Committee that is constituted by the board. So there is an incentive for the CRG grant, those who are getting the CRG grants, and that is incentive has been termed as SCRB Science and Technology Award for Research, that is a SERB-STAR. So SERB-STAR is actually a prestigious award that was instituted by the SERB to recognize and reward outstanding performance of PIs of the SERB projects. So therein we have fellowship of 15,000 per month, research grant of 10 lakh per annum, and 1 lakh per annum as overhead charges for a period of 3 years. Call for nominations would be made once in a year, and nominations are generally made by head of the institution of the nominee. The nominee should have completed individual-centric SCRB project in previous 3 financial years with excellent grading. These projects are also graded after completion of the project. So if the excellent grading is there in the project, so those PIs definitely fulfill the eligibility conditions, and the head of the department can nominate them. And the age of the nominee should be within 40 to 50 years as on December 31st of the year in which the call is made. Then for the young researchers, we have the Startup Research Grant. that aims to assist researchers to initiate the research career in a new institution. It's a 2-year grant, and it's basically to help the researcher to set up his or her lab and work in frontier areas. Startup research grant is also a one-time career grant, and the upper age limit is 42, plus 3 years for SC, ST, OBC, and physically challenged and women candidates. For the young scientists also, there is an incentive award if the performance in implementing those projects are very good, and that is called the Distinguished Investigator Award, or DIA. It is devised to specifically cater to younger scientists who have not received any other prestigious award or fellowship. And it is also a one-time career award, and maximum of 35 awards per year are given. Here, fellowship of 15,000 per month for 3 years is given with research grant for 3 years, which is again optional. And the eligibility is, as I mentioned, they need to be PIs of SCRB, EMR, ECR, or DST, individual-centric completed projects which were rated excellent. Then again, for young scientists, we have Early Career Research Award, the ECRA, and the National Postdoctoral Fellowships. So I have listed in the 2 panels, ECRA, what are the requirements, and for the NPDF. So ECRA is for those researchers who are having regular academic research position, and upper age limit is 37, whereas for NPDF, 35 years is the upper age limit, and postdoctoral fellowship at the rate 55,000 per month is given. And for ECRA, we have 50 lakhs budget limit to overheads, whereas for NPDF, 2 lakh research grant per annum and 1 lakh overhead per annum for a tenure of 2 years is provided. Then SCRB also has Ramanujam Fellowships. The objective of Ramanujam Fellowship is, like we have a lot of good scientists who are working abroad, and they're looking for opportunities to come back to India. So Ramanujam Fellowship is a vehicle that they can opt for to come back to India and continue the research. So the eligibility is like PhD, he or she should have a PhD degree in any basic science, along with 1 or 2 years of postdoc experience outside India. The salary is 85,000 per month with a research grant of 7 lakh per year. And the duration is for 5 years. SCRB also has Prime Minister's Fellowship. These are given to complete the PhD work. And this was launched in a PPP mode and being implemented jointly by SCRB and CII. The objective is to place PhD students in 2 scientific professional environments, both academic and business. Double scholarship, that is 50% provided by SCRB and the balance 50% by a sponsoring company or industry. Currently, we have 100 ongoing fellowships. And under these fellowships, the research that has come out of that 6 patents have been filed in the years 2015 to 17. So these are basically the new initiatives that are being taken up to have more industry-academia linkages and to encourage, like young researchers also to develop a path to have a joint ventures or to do research in areas which have both basic as well as, translational values. So this kind of fellowship have been started. And under that, young researchers are identified and provided support to carry out both basic work that can be translated later on. Then SCRB also has overseas visiting doctoral fellowships. This is a program for Indian PhD scholars registered with academic institutions to carry a part of their PhD related work at any reputed institutions, research institution abroad. It helps to create opportunities to build long-term R&D linkages. The duration is up to 12 months. A monthly fellowship amount that is equivalent to US $2,000 is provided. One-time contingency, preparatory allowances are also provided. The airfare, the shortest route airfare is provided, and 1 visit, each by the Indian supervisor to the overseas institution where the student is working, as well as the overseas faculty to host Indian institution of the student is also provided during the tenure of the fellowship. SCRB also has a scheme called Teachers Associate for Research Excellence, that is the SCRB TARE. See, many of the teachers who are especially in colleges or in institutions which are at less endowed places, due to variety of reasons, they are unable to carry out their research. They may have research interests and at one point of their career may have carried out a lot of good research, but due to many reasons like academic, administrative charges or teaching load, they are unable to carry forth their research interests or their research vigor. So to encourage such researchers who would like to definitely have or to continue the research to some certain extent that they would, in case got the opportunity, would have carried out. So this scheme was initiated. So as it's mentioned here in the slide, it facilitates mobility of the faculty members in less endowed academic institutions. And the research that they will carry out, so they will have to tie up with endowed and good established institutions like IITs, IASCs, ICERs, or any other national institutes. Eligibility conditions include PhD degree in science or PG in professional courses. They should hold a regular academic or research position. We have an upper age limit of 45 years. And there should be a mentor who should be at least associate professor or scientist E or higher level. Until now, 500 awards per year are being provided with a fellowship of 60,000 per annum and research grant of 5 lakhs per annum. SCRB also has a very unique scheme, the J.C. Bose National Fellowship, which caters to senior level faculties who are working and who has established themselves as the leading researchers in the country. And to recognize the talent for their outstanding performance and contributions, this J.C. Bose National Fellowship was initiated. All areas of science is under its purview. The candidate must have a regular position in any institutions or organization. Fellowship is open to Indian nationals residing in India with upper age limit of 68 years. Duration of fellowship is 5 years, and the value of fellowship will be 25,000 per month in addition to the regular income. Each fellow also receives a research grant of 15 lakhs per annum, and the institution in which the candidate is working would provide the necessary R&D, administrative, as well as basic infrastructural support for conducting research. Under SCRB, also there are certain infrastructural grants, and in fact many of the infrastructural grants as well as some special calls for proposals are made based on two ways of support they adopt. Like one is top-down approach, where SCRB actually recognizes the theme areas which are relevant to the country as well as in context of global R&D. And after having recognized that theme area, they will advertise and invite proposals. So IRFA is coming under that kind of scheme. IRFA standing for Intensification of Research in High Priority Areas. And some of the recently recognized areas that SCRB has given call include geospatial technologies, then research around cryo-TEM, then BSL-3 facility, etc. And the average cost of the proposal, it ranges from 5 crores to 15 crores. And another way of supporting is to have a bottom-up approach, like where committees consisting of eminent experts in domain areas, they are invited and brainstorming sessions are conducted to identify some of the research domains which are very relevant, and special calls for proposal are given. So based on the interest of the researchers, the theme areas that the researchers would like to conduct in the country, special calls are given. And currently, in fact, it is also currently advertised. So 3 or 4 areas have currently that are notified under the SRB website includes wearable electronics for biomedical applications, reagentless organic synthesis, and one of the most important themes that would cater to the plant scientists is the biology of plants under extreme environments. So these calls are again in terms of like co-research grant mode of proposal. So under that like co-research grant, the format and the eligibility conditions are there. So even under these special calls for proposal, so these proposals can be framed in terms of like the co-research grant kind of proposal and submitted to SARB for further consideration. Now I come back to DST programs, and myself being associated with the R&D infrastructure division, I would like to present before you a little more on the R&D infrastructure division schemes that are existing. So currently we have 4 S&T schemes under the R&D infrastructure division. We have the FIST scheme, that is the Fund for Improvement of S&T Infrastructure. Then we have an university level scheme, that is the Promotion of University Research and Scientific Excellence, called the DST PER scheme. We have a regional based S&T infrastructure scheme called the Sophisticated Analytical Instrumentation Facilities, or the SAFE schemes. And then we have a national level S&T infrastructure facility program that's called the Sophisticated Analytical and Technical Help Institutes, or the DST SATHI scheme. So under FIST scheme, this was initiated in the year 2000. We have completed 21 rounds of support. Until date, we have supported around 3,000 beneficiaries. And in a particular year, around 90 beneficiaries are identified. That includes also the postgraduate colleges, with a average budget of around 80 crores per year. 80 crores is for the entire program for a particular year. Similarly for PERS, the scheme for the university, we have totally identified around 53 universities till date, with a budget of average 50 crores per year. Under the SAFE scheme, currently there are 15 ongoing centers with a budget of average 25 crores per year. The SATHI scheme is relatively very new, and it was only initiated in the year 2019. And currently we have 3 SATHI centers, one each at IIT Delhi, IIT Kharagpur, and BHU Varanasi. And the budget for average is around 160 crores per year. We also initiated one synergistic training program utilizing the scientific and technical infrastructure that has been created under all these 4 schemes, called the DST Stuti scheme. This is not a full-fledged S&T scheme, but it's a short-term training program, 1-year training program that we have initiated just this year itself, earlier this year. And under that, we plan to identify training hubs and this training program through different hubs, and who will further identify some other institutes who will carry out these training programs. The eligibility conditions include that any Stuti recipient should at least have been supported under the existing 4 schemes, S&T infrastructure schemes of DST. So the target is to train around at least 7,500 researchers and 7,500 students awareness team with a budget of 25 crores. So as I mentioned, FIST has completed almost 21 years, and this map shows you basically a landscape of the FIST support across the country. So although you can see good distribution across the country, but still certain states, some of the states and some of the regions are not that well supported. And so our effort is more to encourage those regions, especially the Northeast, the J&K region, then we have the Chhattisgarh, Jharkhand, even parts of Madhya Pradesh, Rajasthan, Gujarat. These are the places from where we hardly receive any proposals. We have very few proposals. So it is always to encourage organizations, different research organizations, the universities, the colleges, to apply for DST FIST. This FIST program is advertised once in a year, and normally it is advertised in the month of April. So, and through the DST website, you can get the information. So, this slide shows you the supported universities across the country. So here also, I think most of the states having performing universities have been provided the PERS support. PERS actually is supported based on the H index and as well as the I-10 index of the individual faculties who are working in the different universities, and it's an incentive grant from DST in recognition of the performance, excellent performance in R&D areas. So under the SAF scheme, the major outcomes I have listed in this slide. So about 20,000 scientists or users are using these facilities every year. About 1,25,000 samples are being analyzed. That has helped in producing around 2,000 research papers. Under the SAF scheme, short-term training programs are being organized on the use and application of various instruments and analytical techniques to create awareness. And these facilities, individual researchers can avail the benefits of these facilities on payment of nominal charges. So the overall earning that these SAFs had in the year 2019-20, it was around in range of around 10.4 crores. And the average expenditure in last 5 years is about 17 crores. The DST investment stood at around 17 crores. This slide again shows you the landscape of the SAF centers. We have 15 SAF centers currently across India. And the Sathi scheme that I just mentioned, DST has set up these Sathi schemes as professionally managed S&T infrastructure facility that will readily be accessible to academia, startups, manufacturing units, industries, and R&D labs. The basic purpose of Sathi scheme is to encourage industry research. The Sathis are actually, they would support about 80% industry research and 20% the academic research. And this is again to foster academia-industry linkages. As I already mentioned, 3 Sathi centers have been set up. Each Sathi center is supported with 125 crores that is spread over a 3 years duration. And the next call for around 4 to 5 new Sathi centers would be in the new financial year. So what are the new major directions and programs? Some highlights I would like to show. So DST's main vision and components for the next few coming years would be to enhance the quality and relevance of the R&D. So apart from the basic research, importance is also given, and emphasis is also given on the translation research, that is technology and innovation to the market. Then, as I'm already conveying, connecting academia, R&D labs, industry, startups, MSMEs, and the other ministries. We need to also have societal connect and contact, science for people and people for science. That is the mandate that the vision of DST will be in the coming years. Diversity and inclusion, that includes both youth, women, researchers in the rural areas, as well as our less endowed sections of the society. And the research that we will be providing through different schemes that needs to align with S&T national priorities, that includes the water, energy, environment, transport, health, manufacturing, waste processing sectors, etc. So vision components also include, we need to have better science communication, technologies for rural empowerment, need to have schemes that helps reversing drain to brain gain. And of course, through that, we will be able to bring the best of global science and scientists to India and secure our technology future. So addressing the national priorities, a few key initiatives. So under technology, we have 5 technology research centers for translation, waste management technologies. Then we have schemes under Digital India, catering to cyber physical and reconstructing heritage. Then we have also introduced science and technology of yoga and meditation, that is the Satyam scheme. Schemes under advanced manufacturing technologies, we have the National Supercomputing Mission. Then under imprint, we have 10 technology sector priorities, and this program is run with collaboration with the Ministry of Human Resource Development, MHRD. We have mission innovation with DBT that includes smart grids, clean energy, off-grid cooling, etc. We also have initiatives under the clean coal technology in collaboration with Department of Power, BHEL, IGCSR, NTPC, etc. So the future of DST's mandate would include all the schemes, not just DST, even this is a message for all the funding agencies that whatever research funding is being provided to different researchers through different schemes, that needs to have connect with the society. So we have a policy on the scientific social responsibility. So this policy basically, it encourages or gives a responsibility to different researchers to effectively utilize the R&D infrastructure and expertise of funded scientists and institutions that benefit other SRT stakeholders and society at large by providing service to students, schools, colleges, MSMEs, startups, industry, artisans, etc. And some of the simple actions include infrastructure and knowledge sharing, mentoring or training students as well as teachers, fostering research cultures, provide internships, having good public outreach and knowledge diffusion. I would also like to highlight about a relatively new initiative called the Indian Science and Technology Engineering Map, the iSTEM initiative. This initiative was taken up by the Principal Scientific Advisory, PSA, to the Government of India. See, what happens is we have several schemes under which we established and also are still establishing high-end equipments, major facilities, and also under the mission mode programs, some individual researchers have also been benefited with very high-end sophisticated equipments and SNT facilities. But somehow, while we have established these equipment and SNT facilities, the benefit of that has not been optimally utilized. In terms of many of the researchers who are bereft of such facilities in their institutes or nearby areas, because of lack of institutes, because of lack of infrastructure, they are unable to carry out their research work. So to address this gap, this iSTEM kind of initiative was launched, and it was considered as one of the transformative idea. And this is an IP protected, security certified dynamic web portal. It provides option to researchers to check the availability and operational status of geographically dispersed facilities and reserve the most suitable ones online. Geographically dispersed facilities means these facilities are existing across pan-India. I mean, a researcher who is working, say, in a remote area of the country may not have access to these facilities which are far off from his or her place. But through this portal, an individual researcher can just log in and check which facility is existing in which research organization in India. As of now, there is no database or the outcome of funded projects in public domain, so duplication of high-end R&D facilities also occurs. So through iSTEM portal, we also would manage to avoid this kind of duplication. And it will help in the turn, it will also help in generation of funds for maintenance and manpower support. And the capital expenditure will be reduced by avoiding duplication and enable government to support many researchers by providing funds through more number of EMRs and more research grants. So securing India's future, 5 major technology missions that has been launched by DST that includes the cyber physical systems, the electric mobility mission program, quantum science and technology, clean fuels that includes the methanol mission, and MAP India. MAP India is an effort to generate high-resolution topographic database and GIS maps, and this is in collaboration with Survey of India. So what is the way forward? DST has been stepping up its engagements in policy formulation as well as promotion of R&D activities in the country. Currently, there is a positive growth trend in publications and patents, but we still need to commercialize intellectual properties and connect knowledge to wealth creation sectors. So DST is preparing itself for such function developing synergies and partnership with other stakeholders. And DST hopes to work towards the aim of serving India through science. Thank you for patient hearing.
Thank you, Professor.
Any questions?
Bhattacharya. Now I open the session for discussion.
Sure, sir.
Thank you, Dr. Saab. I was also in DS for almost 14 years in the PSC. And now there is a sea change. Now you want to improve science in the country. Your science, or anybody talks of science today, refers only to high science. The traditional science or the basic science in the country is totally ignored. In no university you find an anatomist, you find a cytologist, you find a taxonomist. Of course, this university is an exception, I don't say. But in most of the cases, people do not know even the basic things. But we are building a huge mansion without foundation. I think DST should think on this line also to include at least 1% of the budget towards the basic sciences. I think that's a must from my side.
Thank you, sir. So basic sciences are being definitely, things have changed. It has been more basically mostly due to the availability of budget. So that's how things have become competitive. And under competition, only such projects are being selected. I mean, out of so many, then finally the number of researchers that have gone down, the number of projects have gone down. Having said so, sir, the basic sciences research that is being supported under the DST through the Scrv schemes. And also we have our other ministries like Ministry of Environment, Forest, Climate Change. Then we have the DBT who are also supporting basic sciences research. But of course, basic sciences, as you mentioned, those days have gone where, even simple basic plant sciences or microbial sciences or even within the plant sciences, we have several groups, as I also mentioned, the pteridophytes or lower group of plants. The projects under such areas are much less in number, and that also those which have applied aspects. So this definitely needs to be addressed, and we are addressing to a certain extent, but again, to keep a good balance. So, sometimes what happens in the new initiative from the government that is being coming up is that different organizations are giving similar kind of projects having almost similar objectives. So how to avoid that? So what is being planned now is like there will be again brainstorming across different departments, scientific departments, and create one portal and give one advertisement that will cater to, like DST will cater mostly to areas which are not addressed by other ministries. Like we have DBT, ICAR, ICMR addressing more of the life science based proposals. So the proposals which are in the areas of physics, chemistry, material science, so these areas DST will be more flagship bearer. Through SCRB, of course, we will cater to such areas of plant sciences, as I just mentioned, that special call. Now, this is an area which, in recognition that this area is holding so much relevance, the stress biology and the plants under extreme environment. So this is a good call. I mean, this kind of programs are being evolved, but the number or the number of calls or number of projects that are being supported that have gone down substantially.
Sir, I have a couple of queries. Sure. Can we go back to your very first slide, the very first one? Yeah, this one. Right on the top, in that orange square, you have written private bodies. What's really the meaning of this word, private bodies?
So private bodies means we also have some stakeholders. We are running projects which are in PPP mode.
Okay.
So
now a bit of digression on this. We have the Indian Fern Society registered. Would you give some funds to that society? It's a private body, and you've rated private body.
We have, through SCRB, we have the scope. SCRB, through SCRB, we give funding
It's a registered society under the Government Act and all that, right? And we have been there since 1983. We are publishing the Indian Fern Journal for the last 38 years. 38th volume was released only yesterday. 39th is in progress, without any funding from any government source.
So please explore the SCRB.
What we do is...
SCRB, they have the schemes.
Sir, Dr. Bhattacharya, just listen to me. We have been begging, but not stealing, okay? And borrowing, but not stealing. So on these criteria, we have been able to run this journal, and it is recognized by the UGC also now, and all other agencies. And for your kind information, I will tell you, even foreigners publish their articles in our journal. There are not many Indian journals where foreigners would publish their work.
Sure,
but ours is the journal where you will always find, in any volume, take up 1 or 2 articles by foreigners. No funding from any government source. So when you said private bodies, I thought perhaps maybe you could give us a few lakhs to run our society. Is it possible or no? Please don't give me an answer that the money will come through the institution.
No, it is possible. It is possible.
Because then I have to run after those.
It is possible, sir.
Then, grease their hands to get my bills passed. and then they will say, sir, हमें भी कुछ दे दो. हम कहाँ से देंगे भाई? This is what is, my dear Dr. Bhattacharya, this is the practical part of it, what I'm telling you. This is really going on. The scholars are not getting their scholarship because the clerks sit on it and they say, भाई हमारे कुछ दे दो तब तुम्हारा bill pass करेंगे. तुम तो आते नहीं, तुम्हारी attendance नहीं है, and things of the sort. There is silly questions. Then another thing that I would like to ask you is there any sort of a program in your case where you give funds for book writing? Suppose I am there. I retired as a university professor. I wrote a book, The Illustrated Fern Flora of Western Himalaya. It was in 2 volumes. It's now out of print. If I want to revise it, right, I have to get photographs, things and all that. Will your body help? Help means financial help in getting that book published. I can give it to your, the manuscript to your institution. They can publish it as their publication. I don't care for that. But will they fund it? That's my query.
Yes, there's a program, users program.
Dr.
Earlier it was
Bhattacharya,
under SAR, DSC.
Just listen to me.
Sure.
You say, yes, you will fund it. Then what you will do is send us the script. I will send the script. The script will be sent to some experts who are ignorant. They do not know what it contains. If the person is friendly, he will say yes. If he is not friendly, he will say no. And he will use that data as his own publication. It has happened. It has happened. I've seen it happen.
So I must say that this is one of the incidents
Dr.
because
Bhattacharya,
we have been supporting under this scheme.
What I'm saying is out of experience. It's not imaginary. It's
Sir,
experience.
it may have happened. I'm not
Professor
denying
Punita is
that.
here,
It may
and
have
he will
happened.
also like to express his own views on this subject. We make these beautiful charts and all that, and then somewhere along you said, up to the age of 45, up to the age of 45, a person just is enough that he has accumulated some knowledge. But beyond that, you need more experience.
Sir, every
You
scheme
see,
has its mandate, sir.
Let me complete.
Yeah, sure, sir.
You see, Isaac Newton was once asked, how is it that you can see more than the others? He said, I stand on the shoulders of a giant. That means he stood on the past work that had been done, and then he could only see. In our country, what we do is, once a person is retired, he's thrown away. The other person comes up, he reaches the same stage, he goes away, with the result that our science is in a plateau stage. We never seem to proceed upwards. So
somewhere we have to
if it
strike a
is
balance, sir.
possible-
I mean, with the youth as well as experience, we have to have a balance. That's
you,
what I
sir.
can say, sir. We have to promote youth also. So both the things are there, sir.
Madam Swami, just one addition. Well, Dr. Bhattacharya, actually, you can say their frustration or
One
something
more
like that, you can
question.
say. No problem. Now he and what to say about Dr. R. Rao, also a long time. Basically, we are talking about the Indian Fern Society. I do not know whether you are aware of the history of the Indian Fern Society. It was established way back in 1983 with a mere few people there. And the annual budget for 1983-84 was say 7,000 or 8,000 rupees merely. And now perhaps he has got some handsome amount at his disposal, for major part of which is now gone for the publication of the Indian Fern Journal. It is about 2 lakhs per volume it is being given. Well, perhaps you came here yesterday, you must have seen the gathering over here, and we have the 16th annual session of the Indian Fern Society.
Right, sir.
So the interest of the people, interest of the younger people, interest of those students who are working on terrifiers, it has now been multifolded, you know. Perhaps few only 10 or 12 students were present in the Patiala first symposium at Patiala. And we have now sometimes 100 or more than 100 students, they are coming in, they are attending every annual conference of the Indian Fern Society. I'm talking mostly about the Indian Fern Society. Well, we have some discussion, a very brief discussion without disturbing you, talking about private bodies definitely, since our society is a private one, you know. For number one, for publication, for other purposes, if you give a good amount, I cannot say how many private bodies you have given the funds to for carrying this type of research. If you get some good amount of money, all the experts who are sitting in the Indian Fern Society, they can select students in different parts of the country that what amount can be given as a fellowship or at one-time amount per year to those students who are unable to find out any source for their research. You know, it is the ferns or it are the pteridophytes which are really a neglected one in India only. You said the Snyder you have seen, who told a little bit earlier, or Apteraceae, that lady from the New York, they are practically of no use as a matter of fact. They are practically of no use for the general public of either here in India or the States or in Britain or China elsewhere. But for the academic interest or for the raise the academy or the education of the general people, it is important to work on those basic fields of the science of the or plant kingdom as well, including pteridophyta. So we don't have such funds here in India.
Please summarize.
Okay, yeah. If there is any possibility to give some amount for the Indian Fern Society, then we can give some more scholarships or fellowships to those students who are unable to reach to DST or to UGC or CSIR or any funding agency, including the state science and technology councils. So that is what he really means for that. So that I personally request you, your presentation was definitely, it was very attractive, it was eye-opener, but definitely the answers were not up to that mark. Thank you.
We will discuss, sir. I mean, we can discuss that. How to
One more query, not more than one. No, only one query.
As a president of IFS, I want to say one thing.
Please.
Yes. Sir. Yes.
Sure, please.
You can discuss after
We
the
can
session
discuss.
is
Okay.
completed.
Okay.
You can
I may
discuss
discuss
after the
personally.
session.
Okay. I may discuss personally, sir.
We can discuss later.
Okay. Thank you.
Time is over. Please.
But see, I just mentioned you, there are schemes for
Thank you everybody. Thank you to everybody. Now I
for
invite.
college teachers, there are
Now I
for
invite
college teachers,
Dr.
there are schemes.
Puretha for Thank you. He will convey your idea to the department.
I will take that
Ministry of
comment. I will
Department.
not answer. I will take that as a comment.
Now I invite T. Pathu to honor Dr. Bhattacharya by a small memento. Thank you, sir. One announcement.
Please excuse me, Chair. There is an important announcement to be made. The participants of Future Scientist Award competition, please note, there is a slight change in the program schedule. The competition for Future Scientist Award, which was to be held during technical session 4, will be conducted after lunch as a part of technical session 5. The next lead lecture will be followed by the panel discussion. There is no change in the poster presentation competition. All the participants for poster presentation competition are required to submit their posters in registration hall by 1:30. Thank you.
Now I invite Dr. Catherine Sara to deliver her lecture, and I also invite Dr. Shunoj Kumar to introduce her in the dais. Dr. Catherine Sara and Dr. Shunoj Kumar. Madam, 30 minutes for your...
Good afternoon, everyone. Now we have another, this session we have a topic on the tissue cultures, biotechnological aspects of the ferns. So to deliver this talk, we have Dr. Catherine Sara, and my duty is to introduce her. And we know that we are running short of time, so I'll just reserve my words of introduction. Dr. Catherine Sarah, she works as an assistant professor at the PG and Research Department of Botany, Holy Cross College, Autonomous, at Tiruchirappalli, Tamil Nadu. And she had her PhD from the Center for Biodiversity and Biotechnology of St. Xavier's College, Palangotta. We know that that institution is one of the hub of Fern activities, research activities in our country, especially in South India. And her work is now concentrating more on the Fern tissue culture and phytochemistry, and her team is working on raising rare ferns through spore and tissue culture, inducing and culturing apogamous and aposporous fern plantlets, then callus culture, regeneration, germplasm storage, isolation and quantification of DNA, and phytochemical and antioxidant activities of ferns. And her team has published a number of research papers and won a number of laurels. And with these few words of introduction, I request Madam to deliver her speech. Thank you.
Good afternoon, one and all gathered here. So I'm from Holy Cross College. So during 1994, I was a PG student. I met Father B.S. Manikam, my guide, and Father is my mentor, guide, Abba the Father, and above all, the inspiration to carry out the works on pteridophytes. Magnanimity and generosity allowed me to work on his project on conservation of ferns of Western Ghats as JRF. In the lab, Center for Biodiversity and Biotechnology, I learned so many skills on tissue culture of ferns. First of all, I thank the organizers, Dr. Santosh Nambi, Dr. Madhusudanan, Dr. Iridhiraj, President of Indian Fern Journal, for recognizing me as one of the persons to give a lead lecture. I deem it as a great pleasure and feel it as a homecoming to my father's house and see all my people of pteridophytes throughout the country. I am overwhelmed with joy and hope the joy will fill its fragrance to the younger people who are listening to this also. I very much feel the commitment, conviction, and love for pteridophytes by the team of professors of Indian Fern Society, Dr. Iridhiraj, Dr. Kulla, Dr. Smita Hegde, Dr. Punitha, Dr. Goswami, Dr. Uniyal, Dr. Chima Haminder, and so on. All the time, when I read Indian Fern Journal, I used to imagine about the faces of the people and feel the depth of the work, their sincerity and concern for the plants very much. But after 25 years, I have got this golden opportunity to see all of them in one place. And this is a mega feast for me and an encouraging booster to carry out the work on the same field on the coming days. We are in omics age, but our pteridophytes need more attention to fill the gap according to the present classification based on molecular systematics on what the senior professors explained yesterday. Whatever changes may come, we have to work hard to harmonize and promote the works of pteridophytes under one umbrella and also to justify and fulfill the lacuna created so far, and still more avenues are there to work with pteridophytes. We are having so many areas related to pteridophytes: cytology, taxonomy, tissue culture, phytochemistry, biochemistry, physiology, anatomy, ecology. There are so many areas. So one cannot complete all the areas in their lifetime. Now I am going to deal something related with my work, that is life cycle of pteridophytes. First of all, the youngsters should know the homosporous life cycle as well as heterosporous life cycle. Shortly, I'll brief it. So this is the life cycle of fern, life cycle of homosporous ferns. As such, here we are having only one type of spores, which will produce the sporangia. Spores will produce the gametophyte and then undergo fertilization, and once again will produce the plant. In the life cycle, this is an example for Selaginella. If you take normally in the textbooks, we'll be having this. Here we are having two types of spores. As such, we are getting the complicated or otherwise complicated than the homosporous one life cycle. Now coming to in vitro culturing of ferns, ferns are non-flowering lower plants possessing high phytochemicals and helps to maintain the ecosystem in the shola forest. They are mostly epiphytes, terrestrial plants, and very few are tree ferns. Due to the clearing of forest and extension of cultivated lands and clearing, they are becoming extinct. To conserve them, we are doing tissue culture and micropropagating it and restoring it in original sites. What we did for our PhD work. So concerning this tissue culture, we should understand spore culture as well as tissue culture of ferns. So we are having so many explants in pteridophytes, not only a single explant just like the higher plants. We are having spore, gametophyte, sporophyte. Life cycle studies we have to go for apogamy, apospory, all those things we are having in pteridophytes. Now altogether life cycle under in vitro conditions is successful in nutsens and knobs medium with different cultural conditions devoid of hormones and sucrose as reported in the above mentioned species studied so far. We have brought so many plants with this medium, that is nutsens and knobs medium, devoid of sucrose and hormones. They don't need hormones. So at the early stage, they don't need hormones. And also we have studied micromorphological studies, intraselfing, interselfing studies, gender analysis, reproductive biology, relocation also we have done for the in vitro raised plants. So at that time, we have raised Cyathea crenata. For that, we used to go every month, we used to collect the spores. We don't know when the spores will mature. So we used to go and collect it from the Shenbaganur. There is one tree called Cyathea crenata. We have to collect the spores and then we will cultivate the spores in the medium. Because on those days we don't the medium, exact medium, which plants will grow in which medium, we don't know. So we will be standardizing continuously at that time. So collection of sporophytes and spores also is a very difficult task for at that time. So these are some of the plants which I have worked. Adiantum capillus veneris, Belechnum species, Asplenium. These are all collected from the Colli Hills, that is Eastern Ghats. For my students' work, Ceratopteris thalictoroids, these are the waterlogged ferns. Yesterday also we have seen the campus, Ceratopteris thalictoroids. These are all from the Kanyakumari district. And this is another fern called Acrostichium aureum, marshy fern. And then Adiantum laticifolium, necklace fern. And all these ferns we are having a pet name also, along with the name, a common name we say. I consider it as a pet name. And single frond of Accumulator radiata with sporangia at its under surface and also dehiscing spores. Now I can see the Pterogramma calomelanas, variety calomelanas. It's a weed-like plant, but we have worked so many papers on this. Now you can see the imprints of spores during collection. So it's a very beautiful imprints we got. Whenever we collect plants, we do like this only. We will get an imprint also. Now spore culture we did in liquid medium, and most probably we preferred liquid medium than the solid medium. In solid medium there is no much growth. Nutsens medium, NOS medium, Miller and Miller medium, MS medium as well as half MS medium under the pH 5.8. Now this is a laminar airflow chamber where we are having the preparatory materials for the inoculation of the spores. Now this is the sterilization procedures which we do with the mercury chloride or sodium lauryl sulfate. We will give some time for the sterilization procedure, and we will be having in the filter paper, and then we will pour it on the petri plates and then we will inoculate it. Now this is a fern called Adiantum chordatum. So you have collected the spores. This is one example I'm showing. The spores of Adiantum chordatum were cultured under varied conditions. So were trilete spores, rugose spores, and 20 to 50 micrometer in size, and germinated after 12 days after spore sowing. So during 25 days time, this one-dimensional filament undergo two-dimensional change, the anticlinal and periclinal divisions by the meristematic cell at the apical notch formed on the filament. Then prothallium plate also develops and into a chordate-shaped gametophyte with dermal hairs and evidence adiantum type of development. After 45 days of growth, the pre-sexual prothalli were well developed with broad wings. See, for the development of, for the germination type as well as for the development type, there is a Bible. It was written by Nair and Cover in 1964. So I regard it as a Bible till today for the works of this. So these gametophytes are hermaphrodite and hence possess antheridia and archegonia on the same prothalli. We are having U-shaped apical notch with pluricellular meristem. Few rhizoids also present at the ventral surface. In sex organs initiated after 60 to 80 days, number of antheridia is very few, 2 to 3, and when compared to the archegonia in nutsens medium. We will be working on nutsens as well as knobs. Some plants will respond to knobs medium, some plants will respond to nutsens medium. The first leaf is formed after fertilization in the hermaphrodite gametophytes, and root was also produced consequently. Sporophyte formation was seen after 120 days, and the percentage of sporophyte formation 69 in nutsens C medium. Now this is the agar medium where we can pour the spores, sterilized spores directly, and we can get the gametophytes. And we can culture like this also. You can use brick pieces as well as plastic containers with the tree fern fiber or some other base materials with the medium. You can cultivate this. And this is a liquid medium. Here also we can sprinkle the spores on the surface and we can cultivate it. You can see the gametophytes, fine gametophytes grown, as well as the sporophyte also we can see here. And these are the pictures which we have taken for. And this is the plant Adiantum leticifolia. Now this is a plant called Phymatosaurus colopendria. Here you can see the spore stage, 30th day stage, 40th and 60th day stage with formation of first leaf, and then sporophyte emergence also. Now this is the protocol for the so-called, I'll skip this. Here we want to know about the different three kinds of gametophytes which is formed in culture medium, and we have to observe whether it is intergametophytic or intragametophytic, whether it is syngamy, embryo formation, all these we use to observe regularly. Now this is a Salvinia plant with the megaspores and microspores, sporocarp. Now tissue culture as such, so far we have seen about the spores which we have cultivated. Now this is about the tissue, the sporophytic tissue, that means the leaf tip also we can have it as a leaf tip, or root tip also we can have it as a explant. The medium is like that, nutsens medium, knobs medium, Miller and Miller, MS and half MS. pH is 5.8, agar 0.8 to 1 gram per ml per liter, sucrose 2%. You can see some of the pictures of Atherium nigripus which we worked on 2000 and all. They are taken from the Atherium nigripus is collected from the on the way to Silent Valley with my guide. And the sporophytes under in vitro condition, if you see, shoot tip will be raised by using kinetin plus 2,4-D. First you get pale yellow friable callus for 12 weeks, and then initiation of multiple shoot and root from pale yellow friable callus. Another thing is gametophytes. Now the explant here is gametophytes. We can also use the gametophytes as explants. So we get spongy rhizogenic callus in 2,4,5-T. There is another hormone. It is supplemented with supplemented medium. We have to culture it for 10 weeks. We have to wait for the callus will come. Pale yellow callus show initiation of green globular bodies in medium treated with kinetin. We can see 2,4-D, 8 weeks. And also from callus we can regenerate the plants by using kinetin and T4D, 2,4-D from. Here we are using NAA, kinetin with the NAA. We can, we have regenerated apogamous multiple shoots with the scales. And there is another plant, Erythrophylum multiflora, which I have raised by using kinetin. We got miniature gametophytes as well as apogamous growth, and it was published on 1998 in the journal Current Science. So polyembryony, obligate apogamy, regeneration of miniature gametophytes, they are resulted in Erythrophylum, Atherium, Cyathea crinita. Micromorphological studies also we have done in all these plants. And regarding the Cyathea crinita, I have raised the plant and have relocated the plants to the Shenbaganur, and it is growing very nicely in the Shenbaganur garden. Now this is the plant which I have collected. The first one is the plant Cyathea crinita, and have raised during when I was doing my PhD work. These are the sporelings which I got on those days. And this is the second plant, that is Atherium nigripus. Third one is Theliptris confluens. So these are all hardening procedures which we have done during the work. And regarding this Atherium nigripus, we are having some more just like this callus work. And this is callus from Atherium nigripus. And this is a very special feature where we get miniature gametophytes from the old gametophytes. From the old gametophytes, we get miniature gametophytes. We can use this also for the next generation. Regarding the phytochemical profiling, the steroidophytic plants being capable of synthesizing a vast array of secondary metabolites with antimicrobial potential are a source of new therapeutic agents. So we have done so many works on this phytochemistry. So the distribution of pterophytic plants with high altitudes, with the different ecological features, microclimatic variations, increased UV irradiation, low air temperature, unfavorable water regime, generally increase the accumulation of secondary metabolites. So these plants are experiencing all these conditions in the higher altitudes. So they accumulate more secondary metabolites equivalent to higher plants and its bioactives. So we believe that they are having more accumulates of these bioactives. So based on this, we have done so many works, more than 6 plants we have worked. We got phenol, cardioglycosides, terpenoids, steroids, and coumarins. In Cyathea, Acneopteris radiata, Adiantum caudatum, Hemionitis aerifolia, Adiantum latifolium, and Phymatosaurus polopendria. So these are the methods which we do, that is preliminary phytochemical analysis. Before that, we will produce the solvents with the dried material, and then we'll go for the preliminary phytochemical analysis. We do all the analysis, and then we'll go for the quantitative analysis, and then separation of the compounds, pure compounds going for column chromatographic technique as well as the thin layer chromatographic technique. Then UV-vis analysis to find out the specific compound which is present in the plant. Now this is an example for which the UV-vis analysis of Angiopteris vector which we have done. Then FTR analysis, here also we can see the functional, we can find out the functional groups which is present in the bioactive compounds which we are working by GCMS analysis. If you want to identify the phytochemicals which is present in the compound, we have to go for, after performing this FTIR, we have to go for the structural determination by comparison of mass spectral patterns using the database of National Institute Standard and Technology NIST library, so that we can find out the unknown compound which is present in the our sample. Next one is nuclear magnetic resonance analysis. This also we have done for 2 plants, for Phymatosaurus polopendria and Adiantum laticifolium. So here we have done carbon NMR and hydrogen NMR. So we have referred, this is the output of the carbon NMR. And this is the output of the carbon NMR for the methanolic extract of Phaimatosaurus scolopendria. Here we can refer the compounds from other compounds, whether it is pteridophytes or higher plants, whatever plants. Based on the GCMS analysis, we have done this analysis of carbon NMR and the other hydrogen NMR. Here we can see some of the references which is given here. Most of the references will match with the compounds which we have studied. Or otherwise here, chemical shifts, it is very much related with the compounds. So likewise, we can predict the compounds, what are the compounds, whether it is flavonoids, terpenoids, or coumarins or something like that. Secondary metabolites we can predict by doing NMR analysis. And before going for, suppose we have found out that the particular compound was there means, for example in Phaimatosaurus we have found out coumarins. It's coming under, what you can say, this steroid compound. So before going in for, we have to find out the medicinal property of a plant by references. We will first of all we will see, and ethnobotanical medicinal property is also there. People are using these plants as a crude drug in the area where the plants are there. That means in the high altitudes or otherwise the tribal people they may use. We can correlate with them and then we can go for the work. But we didn't went like that, but we have referred the references and we have went for the further clarification of the or other screening or otherwise how we can probe or explore the compounds, how it will be useful for the society, something like that. We went for, and as soon as we have found out whether it is a terpenoid or a coumarin or something like that, we'll go for the disease or disease which is related with the references. And then we have used the bioinformatics tools before going for animal study as well as the cell line studies. We went for the bioinformatic tools. We are having our center, bioinformatics center in our college. So we have given the samples to them and then they have worked out for us. And we have predicted through the in silico works on bioactive compounds which we have worked on. So in silico docking is nothing but the molecular interaction patterns between the receptor and the ligand molecules were represented using in silico molecular docking analysis, Discovery Studio 2.1 version software. So first of all, we have to prepare the ligands, small chemical molecules from the GCMS which we got from the GCMS analysis. Then we have to trace the drug likeliness properties by Lipinski rules of 5. So we have to find out whether these compounds are relating with the Lipinski rule. It is obeying the rule, that rule. Then we have to go for the preparation of receptors. We have to find out from the database the target proteins which is very much related with the ligands. Then active site prediction and docking by lipdock method. So prediction studies based on docking confirm the significant interaction between the ligands of the plants and cancer pathway proteins, and thus in silico evaluation was successful, which leads to anti-proliferative study through cell line testing. So what we have done is for Phaimatosaurus scolopendria, it was very much related with the lung cancer cells. So we have predicted this through this lipdock method. Then all these plants, Angiopteris evecta, Phaimatosaurus scolopendria, reveal the efficiency as a powerful anti-cancerous agent for colon cancer cell lines and lung cancer cell lines. Now these are the 4 compounds which we have selected from Phaimatosaurus scolopendria, and ligand molecule was developed as 2D structure and generated into 3D molecule using ChemSketch software. Now these are the X-ray crystallographic structures of receptors were collected from PDB data archives and opened in the software window. These are all procedures which is given by them. And then now this is the 3D structure of receptor proteins which is we got it from the database. And then active site prediction we have done and docking by lipdock. The result will be like this. We have to consider the poses for the number of poses when we do the docking. So first, second, third and fourth compound got 91, 100 poses, whereas the first compound, we didn't get poses, so not get results. So we went for, and now this is the interaction. Interaction is depicted by the green colored linkages which we can see in the picture. Now these are the three molecules which they have interacted with the target proteins which we have selected for cancer cells. So after this, when we predict, after prediction, we have to go for the anti-cancer studies, whether with animals or with the cell lines. So we have done cell line works for this. Apart from this, we have done, for anti-diabetic works, we have done animal work. Now here, this is the procedure for that MTT assay. This is an MTT assay. Here, anti-proliferative effect of an angiopathy vector on non-malignant cell line. So this is non-malignant, but malignant cell line is also there. This is the viability will decrease when the concentration of the drug will increase. The next one is, see this, after the MTT treated assay, we have to stain the cells with the acridine orange and ethidium bromide, double staining. So when you stain, you will get these cells. The cells depict apoptotic cells. We can see more apoptotic cells, that means killing of apoptotic cells by the drug could be observed here. So first one is control cell, the second concentration, third concentration, it kills the cancer cells. Flow cytometry analysis also has done, they have done the, so the DNA content, you can see the debris, more debris in the flow cytometric analysis, HT29 cells treated with angiopathy vector. So the treated cells, there is more debris of DNA. You can see the picture in the picture. The second picture, you can see the more debris of DNA. And one more analysis is there. This is what we say it as comet assay. Here also we can see the DNA damage on, this is the lung cancer cell lines. Here also we can see the tail. There are more DNA damage in the third diagram. We are having the more tail and less DNA. And also we do the toxicity studies, which is very much related to the content of the material. So if you want to go for drug discovery, you have to go for all these procedures. And this is the basic procedures, and still we have to go far away from this. So you have to do all these things, and then you have to go for the discovery and development of lead compounds. You have to go for, next we have to go for lead compounds, preclinical research, clinical research, FDA drug review, FDA post-market drug safety monitoring, all those things, then only you can release the material, or otherwise the your drug. So, so far we have seen about the importance of these ferns, what we have done in our lab, and experimental biology. If you see these ferns as experimental biology, it serves as an explants for gametophytic studies, sporophytic studies, high plasticity for experimental studies. Gene expression could easily induced and modified using PGRs and other chemicals. Gene switching and phase reversal could be studied. Study of heterospory and seed habit, genes between flowering and non-flowering nature, natural apoptosis during degeneration of megaspore tetrads. We are having so many features in pteridophytes yet to explore. Secondary metabolites and sporophytes and gametophytes, single cell culture, molecular farming. There are so many avenues in pteridophytes to do more work. And so ferns are not inferior to higher plants because they contain more secondary metabolites than angiosperm plants qualitatively and quantitatively. They contain secondary metabolites not found in angiosperms also, maybe. They are very easy to handle and study under in vitro conditions. So now these are the students which worked all these, did all these works. First one is Manunmani, second one is Revati, third one is Ramya, fourth girl is Sujata. So they did all these works and helped me, and this is my department. And the first one standing there, that's the last, She did the experiment for, in angiopressy vector, the work on anti-cancer work. It's a very good work. And all these works we have done by the fund given by UGC. So I acknowledge UGC this, in this forum. I got three projects. First one is about watershed management, that is very much related to the extension activity in the college. Second one, I got the experiment for isolation of DNA from gametophytes. That paper also I have published in Indian Fern Journal. And then the major project is screening of phytochemicals from pteridophytes. So I extend my thanks to UGC. And if you give more funds, as Sir Bhattacharya Sir has come here, so please help us. If you help us, we flourish in like anything. Yes, ma'am. I thank, once again, I thank the organizers, our principal, Dr. Sister A. Christina Bridget, Holy Cross College, my beloved guide, late Reverend Father Dr. V.S. Manikam, pteridologist, and very good inspiration and moral support to enter into this field, and funding agencies, UGC, who have helped so far to carry out these works.
Thank you,
Thank
madam.
you so much.
Now the session is open for discussion. 2 or 3 questions. Yes, sir.
Yes, sir.
Go back to your slide where you depicted the picture of Adiantum capillus veneris. It was amongst your first few slides. Yeah, yes, this one, right? Here you have Adiantum capillus veneris, and there on the top there is some other
Blacknums
one.
there, sir.
Now,
Blacknum species.
what I wanted to ask is, when you collect the spores, you collect them from nature?
Yes.
So, in this case, you would have collected from Adiantum capillus veneris?
I didn't work on that, but I said that this capillus.
Possible, right? Now, how would you prevent the spores from the fern on the top falling on the fronds of the fern below? My question is, how would you ensure the purity of your spores, that they belong to only this species and it's not a mixture of others? How would you ensure that? Even in the air, there are spores of other ferns also. And it's quite likely that a few of them may have settled on the front of the other fern.
Yes, of course.
How would you ensure the purity of the spore for your experiment?
When before inoculation, we have to find out from the.
No. Inoculation comes later. First of all, you collected the spores, and you've collected them, as you said, in nature, right?
Yes, sir.
How do you ensure the purity of the spore, that they belong only to this species, and it's not a mixture of one or two other ferns coming in?
We have to get some more special
Did you examine the spores
procedure
under
for that.
Did you examine the spores under the microscope before inoculating them? You did not.
Yes, sir. Yes, sir.
The other thing is,
We usually.
listen. How many times did you repeat your results? Have you repeated your results, or these are the observations of only one time?
Repeated 3 times. We have repeated
Only
3 times. Yes.
And the source of the spores was the same?
Yeah.
Or did you repeat the spores from other collections also?
The same collection we used to do 3 times.
No. That is what I am saying, that it could have been a mixture. 1 or 2 spores from other ferns could have been in a mixture.
Maybe.
In your culture. Maybe means the exactness is gone. Then you cannot say for certain that this, whatever your results are of this species.
But
if you were to repeat it, collecting the spores from different places, maybe your results would have been better. That's one thing
Yes,
that I
yes.
wanted to know. The second is, you studied the development of the gametophyte. Did you study the ontogeny of the Anthridium?
Pardon, sir?
Did you study the development of the Anthridium, how the wall formation? within the Enthridium? No.
That continuously we will use to observe that.
Dr. Sara,
Yes, sir.
the question is, the development of the gametophyte is very well known, but there are gaps. There are gaps like how the Enthridium develops, how the wall formation within the Enthridium takes place, what's the shape of the first wall, shape of the second wall which is hemispheric, what's the shape of the third wall which cuts out the cover cell. These are the things which require attention, not that, okay, fine, gametophyte develops it, fertilizer, that's already known. Try to find gaps and try to fill up those gaps. And before you do that, ensure the purity of your samples. Thank you.
Question? Yeah. Where?
Okay, ma'am, congratulations, ma'am. Very good presentation. We everybody enjoyed and benefited. The quantum of the work is very nice. It's equal to 2-3 PhDs, isn't it? So I'm wondering how you are managing this, even though you are heading the department. The introducer missed. She is the presently head of the Department of Botany at Holy Cross College. And I appreciate you for, and thank you. I know you are the product of the B.S. Monica, my teacher. I missed that opportunity. My students are doing the same work from your advice. For plus or doubt, I want to explain. That's why I stood up.
Yes, sir.
After the collection, we are collecting only the sorus with the front.
Yes.
We brought it to the laboratory, and before the inoculation, we sterilize it, wash it. 100% wash it, 100% without any amalgamation or yes, debris or from there. So 100% pure. After we culture, the spores germinate with 100% of the specific species.
Whenever I collect from the field itself, I used to collect in a neat white paper. And then I will make it
so there is no possibility of mixing or adulteration or other spores from other species.
So from the field itself, we will collect it very properly. So that there is no possibility, but even then, as sir told, we can have that possibility.
So from the classical, you've done all the advanced studies. A small doubt, because my students are so intelligent in phytochemical analysis. Many parameters you are doing, but my doubt is FTIR for the analyzing the functional group, GCMS analysis for the LCMS is there. From that we can easily elucidate or enumerate all the secondary metabolites, alkali, tryptophan. What's the need of doing UV visible spectrophotometer? UV spectrum, what's need?
The compound, whether the compound is present or not. The second one is functional group. If from the compound we can understand, we can differentiate. For that purpose we are doing the UVs.
Proton NMR and CT NMR, is there any
So there is a need that, that means wavelength we have to find out. For that purpose we are doing that.
But my humble request, advice is when we are indulging many parameters, many work, GCMS, LCMS is sufficient to enumerate. Instead of doing all the chemical parameters, this is enough, I think. Congratulations, madam. Thank you. Thank you.
Yes, sir. Thank you.
Thank you, madam. Now I invite Dr. R.A. Rao to honor madam by giving a small memento. in this session to
Okay, sir, hardly we get only 1 hour for this discussion and we are running out of time. So I may start with that. So we are here, some Kashmir se Kanyakumari tak sab log yahan aaya hai. Happy Holi. And we are here to discuss on the future of, I believe, I wish to convey my regards or my concerns over that. Rather than the terminology, I feel that the teratologists are more endangered. Teratophytes may look after their part because they have come over this stage, seeing many hardships, and after hearing the lecture by Harald Schroeder, we have seen that it has its own mechanism to overcome the struggles. So, but we don't have any such mechanism, so we have to find certain ways to ensure our own stability. So maybe and here on the desk we have Dr. R.R. Rao sir. Hope you will be, all of us will be knowing this. Again, I am introducing him, and he's having one of the rarest kind of career background. That means going forth and back to academia and to research. So he was joined as in academia, that means in a teacher, then gone to BSI, again come back. Again, he's continuing his research. So he shows his passionate research. And other great personality, as we all know that, none other than Sanjapa sir. I'm quite happy to stand in front of him. He also played a key role in formulating the strategies for botanical research in India as the director of the Botanical Survey of India. And he's still continuing his involvement in research, especially in formulating the strategies and advising the government. And during his period only, we got that ICOPTAX project, I think so. And I am a beneficiary of that project. And in the morning session, we have discussed the difficulties in getting the proper funding or support for the teratologists, especially the field like us. It's a totally neglected field. And no doubt about that, during the last so many years, we are not getting that much support. And it was during his period, a revival for the program, that means the botanical research, as the ICOPTAX project of the Ministry of Environment and Forest. And I can surely or definitely say that Dr. Beniam is a gem class scientist produced as a product of that ICOPTAX project. I hope he'll be agree with me. And it was led under none other than Father V.S. Manikam. He was the coordinator for the ICOPTAX project for Teratophytes and Gymnosperm. And here, Professor P.V. Madhusudan was the a collaborator with that project. And I was also working in that project. And that is how I earned or I have acquired the experience in teratology. But in the recently, we have made another try to revive or reintroduce or the second phase of ICOPTAX project. But due to the COVID pandemic situation, it was about to sanction that and nearly approval. It has reached the approval stage, but due to the COVID pandemic situation, it was kept in abeyance. Otherwise, we would have here with a proud, that means we are going through the second phase of ICOPTAX project. So my concern is that, and I hope all of you may join with me in saying that we are without much support. And in most part of the world, the reassessment programs are going on. That means in all parts, not only the teratophytes, but in all other groups, they have already completed the flora part. Then they have again gone for the reassessment part. For example, let me quote one example from Japan. I know that it is not fair to compare a condition from Japan with India. Japan is a small country, but the way they have implemented the reassessment program, we have to notice that. So they have produced the reassessment by collecting or incorporating the details from more than 2 lakhs of herbarium specimens. And half of them, the interesting part is that half of them was collected or contributed by the amateurs. And we don't have any such situation in India. And another part is that they have completed nearly 75% of the cytological data of that. And more than 95% of the RBCL sequence. So can we imagine at what time that kind of situation will reach in India? So that's one point I'm presenting in front of the panelists. I hope they may guide us. Then another thing is that we are going through an age of plant blindness. Even though everything is available on the web or it is easily available, most of the students or most of us are least concerned about the role of plants, and they don't have the capacity to identify the things. And whether we have to do anything for the improvement of that situation. See, in most of the universities, now botany is phasing out. It has already been declared as an outdated one. But we have to fight back, otherwise it will be very difficult. See, let me quote one example. Sometimes we may get ID request from students or amateurs. So if it is from an amateur, usually we will not ask much about from where it has been taken, the photo has been taken. But when it is getting from a student, I may sometimes query something. That means, is it for your project or it is for something like that? But what happens is the pathetic situation is that usually the untrained, I'm not blaming any students, but they may not be properly guided. They may be sending the part of the frauds. Even we know that the identification of the pteridophytes, we need rhizome, details of rhizome and details of the sporophytes. It may not be there. It is not because it's not the problems of the student, but the problems of the guide or the mentor. Or they may not be even able to, means they don't have the idea to Google it or something like that. So that's another situation we have to discuss. And then third one is no doubt of that, the funding situation. So do we have any chance? What are the mechanisms or what are the methods to either beg or to plead or threaten? So I hope they may be giving some guidance on that. And recently we have seen an interesting picture. I wish to share one experience also, that Dr. Sanjay Prasad was on board of a tracker, and it was circulated in most of the taxonomy groups or plant groups. So it shows his passion for botany and field biology. See, nowadays everything has been integrated with modern techniques, but still he's a stalwart and he's ambassador for the field biology. Sir, I appreciate you, sir. And I invite Sanjay Prasad to initiate the discussion part. I'm not putting my words on other parts. I'm not putting my comments on their mouth. I humbly request you to comment on this aspect or any other aspects that will be guide us.
Thank you very much for giving me an opportunity to share whatever I think about the present situation. In fact, I am an odd man out here because I have never studied ferns in my career any time, except that as a young boy, grazing buffaloes and cows in my village, I was moving around boulders of my hillocks. Under boulders, I have seen these what is called as Acrinopteris, isn't it? Actinopteris, and also Hemionitis. So these are the ferns which attracted me right from my childhood. But we were more interested in looking for some edibles, particularly fruits in the field. But all the more, these plants, particularly under the boulders, in the shade of boulders, I was attracted. And when I reached my MSc class, I took them and got them identified by my teachers in the college. So that's how I was introduced to ferns, in fact. But otherwise, I don't know. My panel discussion as a person of angiosperms, and in angiosperms also, it is something like doctors specializing in left nostril or right nostril. So I am specialized in legumes, mostly legumes. But other groups, some general knowledge I have because I made a general survey in Great Nicobar Island for about 6 months continuously. Coming to the panel discussion on these ferns, my most important request to all the teratologists and the members of the Indian Fern Society is that now that we have so many practicing taxonomists in the ferns and lycophytes, still we have not been able to complete the flora. There are some areas, no doubt, not only for ferns, for all the groups of plants, there are some areas which we have not surveyed as yet, particularly in the northeast region, for various reasons. But during my tenure as director, I was able to revive these teratological studies by recruiting more and more people, because till such time, there were hardly any teratologists in Botanical Survey of India, except that Dr. Dikshit and Dr. Panigrahi. So they were into a different aspect, and Dr. Dikshit was the one to bring the census of Indian pteridophytes, and that formed the basis for many of the people to work on. Subsequently, when I brought, my idea was within about 5 years' time, because all of you are aware that there are not more than 1,200 to 1,300 fern species, pteridophytes in our country. And if strictly go by what Fraser Jenkins says, it's around 1,000 species are there in our country. But that is debatable, because ferns, the generic limits, the species limits are all still in a very liquid state. They are not completely stabilized. And for the variations that they have, the hybrids that they produce. So that has been always confusing. Maybe in the modern days, if the people can help us by using molecular methods, if they can develop the boundaries of the species and genera, that may be very, very useful. But in my opinion, it's basically the traditional taxonomists who have to build the boundaries of genera, species, and families. The number of families have come down in Christian Ho's work, Schneider's work. So recently that linear classification, there are only 48 families. So if you look at what Dikshit has mentioned, 116 families. So see the drastic change of number of families, number of genera, and number of species. So my most humble request to the Fern Society and those who are practicing fern taxonomy are to complete the flora of India. Not only you. See, your complementation is very, very important for the Botanical Survey of India to do. There are only 4 or 5 teratologists in Botanical Survey of India. So the topmost priority should be produce a fern flora of India. But there are some regional publications like Professor Kullard's Western Himalayan, and also Dr. Pandey, H.C. Pandey, late H.C. Pandey also produced something like that. And when Sarnam Singh studied Northeast India, but that is not sufficient yet. But we want to have a comprehensive flora for the country. That's very, very important in my opinion. After that, you debate, discuss, and work out, using anatomical study or DNA sequences or whatever that you want to use. You use all that methods to crystallize the boundaries of each of the genera, families, and species. So that is, as a former director of Botanical Survey of India, that is my top priority, if you ask me, to complete the flora, and it should be targeted. It should be within, say, whatever target you decide. It should not take more than 5 years. So when they say, therefore, we need not have to ask more money from Arindam or anybody, because If you complete at least that aspect, then experimental aspect you take more. And I am sure, because if you go to academies, if you go to any funding agencies, the main problem is, if you go with that, I want to revise the flora, Thelipteridaceae, they will not give you money. But if you say that, no, I want to do the molecular studies of Thelipteridaceae and delimitations of the taxa within that, and evolutionary, phylogenetic aspect of that, then they will give you money. This is unfortunate, but this is happening. And not only happening, and it has happened in my own presence, because I was there in DST committees and EBT committees, and you name a committee, I was there. So if I single-handedly, if I fight for funds, it doesn't work. It has to be, you have to convince Punita Saab, R.R. Rao Saab, and someone else also in the committee. Then only it is possible to get. Otherwise, if you go to academies, the same problem. If I nominate Punita Saab for Indian National Science Academy, nobody will vote for him, except Sanjaba. The problem is, they're all microbiologists, molecular biologists. If you go to even the sections have been changed recently, remodeled. Molecular biology has been sent out of the section. But in spite of that, this is happening. So what I want to say is, at an early date, we have to complete, we should set a target, whether the Fern Society sets a target, have a discussion with the Director Botanical Survey of India, and see that in less than 5 years' time, we bring out a complete flora of the fern and lycophyte flora of our country. That's very, very important. Yes, Dr. Fraser Jenkins, he came to Botanical Survey of India in all the herbaria, particularly in Sikkim, Arunachal Pradesh, and also in Central National Herbarium. He spent a long time, and he has modified, the arrangement, the identification, he has done quite a bit. But our own people like Dr. Kolia, Benjamin, they have been working very hard. But my set target is only the flora of India. So that should be the top priority. Then you take up any other thing. Because how long? Because we have so many pteridologists.
Sir, can I ask just to add that? The problem is that whether do we have that kind of thing? That is a question raised by the most of the funding agencies. Everything has been done. That means either checklist or floras of regional levels are available. So what is the need of any kind of things? That is the question that usually they ask.
No, a comprehensive for the whole country. See, every country has a national flora. So national pteridophyte flora you should have first. Then you work on anything.
It need a lot of investment. That means from the part of students and everything. Usually,
then you see, then taxonomic revisions will continue.
Yes.
It is not that it will be the end of once you write the flora. Taxonomic revisions, refining the boundaries or the circumscription of the genera, species, families, all those things will continue. But at least as a reference book, you should have one. Whatever variation, you have to depict all the variations that you see in a particular species. Indicate that these are the chances. This may be hybrid, this may be variant, this may be ecological variant. All such things have to be depicted so that you get a clue for the students. The next generation of pteridologists will get a clue that what they have to do, undertake.
Okay. Thank you, sir.
Thank you.
There is just an announcement for the participants for the poster presentation.
Please, just 1 minute.
BSI people, they have merely copied what Fraser Jenkins has said. And Fraser Jenkins himself says nobody is infallible. Try to find out the mistakes in his class work.
Yes, I totally agree with you.
That's what I pointed out yesterday also. Don't follow him blindly. Try to find out his faults, point them out. It's no use reinventing the wheel. By following Fraser Jenkins, you are merely reinventing the wheel. That man corrects his mistakes if he has made.
Yes.
You see his works from 2008,
Yeah,
2014, 2020, so on. There are nomenclatural changes. But if the BSI people are going to follow him blindly, the result would be disastrous. Look, in China, they took the help of the people from Holland, Leiden people. They got him, the persons from there to frame that flora of China, pteridophyte flora of China. Similarly, in Pakistan, they took the help of the Q people. Why can't these people have the help of our own people? Why? Why shy away from them? Either they are afraid that those people know more than them and they know less, and they want to hide their faults. What is it?
That is not the, no, let me, sir, please, let me, I understood your point. So let me tell you, it is not a question of hiding from their...
It's not the prestige of the BSI, it's the question of the prestige of the pteridologists of India.
Exactly.
Sir, may I interrupt? There is an announcement. The participants for poster presentation are requested to be present in the registration hall. The competition starts by 3 p.m. So now...
No, the idea is classification level, I don't know, I mean, Dr. Kulia or Benjamin who are there, they have... Pichi Shermuli, we have Cheng had his classification. No, see, the problem is they have to interact with all the pteridologists. They have to interact and then come to a common consensus on a particular classification or a modified system of classification. No, they are not shy. I don't think they are shy. No, unfortunate. That should not have happened.
Which circle is interested with which, has been entrusted with which genera or which families? Can you tell me?
We have not entrusted.
But they know it and they will not pass
No, the point is, for example, let me tell you, for example, if Dr. Benjamin was placed in Arunachal Pradesh, because the area was not surveyed for fern flora. So, R.R. Rao is there, I know, but even then the collections were limited. So entire Northeast India we wanted to survey.
Sir.
That's fine. I mean, I don't say whatever family, whatever classification you want to follow, my point is you complete the flora. Then you think about classification. Why not? Why if the globally accepted genera are there, globally accepted families are there, then you can try afterwards.
Sir, I think there is a matter of great debate.
There should be some consensus. You cannot be differentiating yourself.
Sir, it took a detailed discussion. I think it will take a lot of time for discussing that.
Yeah, I am sure it will take. Sure. Because classification means, I agree with Pichu Sharmali, someone else with Cheng's classification, someone else with the linear system of classification proposed recently.
Okay, sir. We can prioritize that part. That means the first part is discussion.
All said and done, there should be a flora, that's all.
Okay. So, may I request Punita sir to comment on this aspect?
Well, friends, really, I agree with both Professor Fuller as well as Dr. Sanjay what they have told. But what we have to decide right now, because such type of discussion cannot take place time and again. So what I understand that we must request Botanical Survey of India to organize a discussion platform where few of us can sit, because the Fern taxonomists who know taxonomy and classification or the phylogeny, they are very few. So they can be adjusted at one place and make a plus minus consensus. If it is not possible that the total consensus, it is possible that since everybody is a learned man and he must be having his own ideas, his own concepts, his own inferences about whatever he has learned through his life. So there may be some differences of opinion rather. So we can sit over there and we can find out some route to reach to some consensus. Well, as far as we have recently the checklist of Pteridophyta of India, Pteridophytes of India is concerned, published by Scrv Schemes. That is the recent one, and that happens to be the talk of the discussion. Well, he has done a great work really, it is true. He has compiled or he has written something which was not before published. But there are still many lacunae, and personally I don't agree with his classification as well as his distinction of the genera as well as places with species as well. So, I mean, and more so the main lacuna in his checklist is that he has reported a plant from, by the way, Uttarakhand or Himachal Pradesh or UP or some other place, but he has not given any precise locality. And what to say, any distinctive feature is not described by Scrv Schemes, which is a main lacuna. And if we discuss this matter, we must give some distinctive feature of that particular plant, no matter whether it is a genus or no matter whether it is a species or rather a subspecies. So that will definitely benefit the coming generation who can easily understand that what plant I am dealing with. So this is the thing about the flora. Well, as far as the classification is concerned, classification even little before when we heard about that lecture from that lady from Berkeley, California, and also from Snyder and what to say, this Sri Lankan person. Even these two, the Snyder and that lady, the lady was rather more, she was involved in the D. Almeida's phylogenetic, molecular phylogeny of Thalapteridaceae. And Snyder was also involved in the phylogenetic of PTG1. So this Snyder, he accepts that he was there, but there are some lacunae in that classification as well. That cannot be even perfect. So keeping in view both the traditional taxonomy, traditional phylogeny, and that those if we can take, we can consider looking onto the pluses and minuses of both the classifications, no matter is the phylogenetic or the molecular one, we can accept the medium path, or we can accept some plus points from one, the other plus points from the other, and within that time limit, what say by the speculated time, what I debate is say 5 years, we can complete it. So with that note, I can again I will propose that the Botanical Survey of India should be requested if it can do some such type of work.
Sir, the thing is that we have reached only to the checklist level only. See, it is just like after this COVID pandemic situation, so most of our habits have been changed, especially of the students. See, most of them, they are stuck to the mobile screens. So what we are doing is that we will just, this is our habit, is to zoom into the details. But after a period, after a stage, we cannot, it will be pixelated. So it is just like that. We have reached, at present we have only a low resolution picture of the Indian terminology. So how we can improve that? So whether we need more fieldworks, whether we need more herbarium collections, or do we think that all these are waste? So that's the point. I think that is the point. Classifications and other things, the preparation of all that will come. Most of the other countries, others have gone far ahead of us. So where do we stand? I think that is the point we have to discuss. Sir, R.R. sir has a lot to make on that.
Thank you. Thank you. I am very happy to be here in the gathering. Thanks to Dr. Amphi for giving me this opportunity. I think the discussion went on very well between, I think, Dr. Sanjappa and Dr. Kullar. I fully agree that we should have our own pteridophyte flora, complete flora at the national level. I also agree that classification, why should I follow Ching or Holtam? What Professor Kullar said is also I agree. Why should our own Indian pteridologists propose a classification? I don't think there is any problem in that. Any expert can propose a classification. Nobody stops, nobody prevents. So therefore, I think seniors like Kullar and others, they should come out with a classification. I think we will accept that one. That's a different thing. Now regarding writing the flora within a time-targeted period of 3 years or 5 years, this responsibility should be taken. Fern Society itself should take the responsibility with adequate funds from either DST or even Ministry of Environment and Forests. This should go as a recommendation to see that in 5 years, Fern flora will be complete. So that recommendation should come so that BSI, Environment Department or the DST should fund so that once for all that work is done. So that is my one point. I have coupled a number of points. The second point is, yes, enough literature has accumulated, enough collections have been made. The total number of species in India is expected maximum of 1,200 species, which constitutes the 1/10 of the world flora. I think that's the estimate. And we have already collected much of this. Data is available, literature is available, only somebody has to sit and compile. If you see the Flora of Taiwan by Dewall, it's a wonderful compilation. I think very good work that Dewall has done on the ferns of Taiwan. I think that pattern at least we should adopt and then try to put our own classification as Professor Kullar has said. That is number one. And collections, some 40 years ago, we did 2 state floras. One is Nagaland, one is Meghalaya when I was in Neho. I think fern collection and identification is not very difficult. Only problem I used to face is the closely related species, the variations are so continuous that it is difficult to segregate for us. So therefore, some amount of revision or biosystematic studies are needed with regard to the ferns particularly. Closely related species very difficult to identify. That is one thing. And another thing is marning, as I said, biogeography of ferns is completely ignored. Biogeography of ferns we do not know exactly. In fact, flowering plant biogeography, we always talk, the Himalayan flora has entered South India through Pleistocene glaciation or Satpura hypothesis, that kind of discussions are there. Ferns are much older in age compared to flowering plants. Now, even before the Western Ghats, even before the Himalaya erupted, flowering plants were there. Now, large number of Himalayan ferns are there in the Western Ghats, whether they are discontinuous distribution or disjunct taxa, or it may be a part of that just glaciation. We just do not know. The biogeography is completely unknown. As I told, there are lots of differences between the flowering plant biogeography. When I was working in Nagaland, the flora of Nagaland is more similar to Sikkim with regard to ferns, with regard to flowering plants, rather than Meghalaya, which is only 160 kilometers. There is more affinity. But ferns, there is a continuous distribution. 65, 68, 69, that is the percentage of distribution we find. So that shows there is drastic difference in the biogeography of ferns as well as the flowering plants. I think that should be looked into. The other important point I would like to emphasize again here is that you see, it is not just necessary that we should only make a catalog, identify, description and write a flora. Now the time has come to convert our biological wealth into economic wealth. I think ferns are a wonderful group. Why should we not go for bioprospection and product development? I think there are wonderful medicinal plants in ferns, and marking some novel molecules also she has shown. Now we should go for bioprospection and go for product development on one side. The other side, all the fern species are ornamental. Why not we develop agrotechnology, popularize the farm so that everywhere, develop entrepreneurship here in different places, so that ultimately we should convert our bio wealth into economic wealth from out of the foreign group. Now, if you want to do that, any amount of money I think DST will give if you go for bioprospection of foreign species. I think that's one point I would like to emphasize. And another important thing I want to, the phylogeny, I think this is very important. Now, we have been very blindly following, say for example, APG, the flowering plants, angiosperm phylogeny group, where only the molecular biologists have given us a classification which 300 years old, our classification we have thrown and accepted a classification which has come just 50 years ago or 20 years ago. This is not justified. I don't understand why it is. No, the giants like Taktajan, Cranquist, Hutchinson, all these fellows are fools or what? See, they have given wonderful phylogenetic classification which we have bundled it and thrown it against that APG system of classification which has come only some 20 years ago, and within the 20 years more revisions have come. So that means it is not still stood the test of time. So therefore there is a need that we should study that classification before we change our. Now similar mistake we should not do with regard to the ferns. Now I think there is already a group started to work on fern phylogeny group. Now where only the molecular biologists sit and do the work. Why not we strongly should organize that the classical taxonomy should also be there in that group so that we can take care of the real phylogeny of that group. I think that point we should very clearly say this. Otherwise what is happening is entire classification is based on molecular and DNA based classification which has come. There is nothing, no place for variation, and if there is no variation, any synthesis does not explain variation pattern in plant, and that taxonomy is incomplete. So therefore, this is high time, this is beginning, before they propose any classification which we very readily accept. That should not happen. We should all sat together. I think the teratology group here under the Fern Society should put their voice saying that if we are developing a FPG group, yes, classical teratology should be there, Ferns especially should be there, so that real classification should come. I think that's a very challenging area. I think it should be done. And as Professor Kullar said, I think I am also thinking that why not from India, one classification we should produce from our experts. Why should we blindly follow other classification? So this is one point I think we should do. Thank you so much. I think this
Thank you,
is
sir. Thank you, Rao, sir, for pointing out the complexity of pteridophytes groups. So we can just compare the pteridophytes with the angiosperms. So it is having an array of complexities, because we can't just expect natural hybridization in that much prevalent in the angiosperms, but it is possible in the pteridophytes. It's possible. So our approach should be in that way.
That creates more problem
in identification, and so the way we look into that matter is we have to think about that. So earlier, usually the taxonomists are doing just like the angiosperm taxonomy, but it requires a more efficient method, I think so. Now, I think...
one point I want to...
Yes, sir.
See, this APG system or PPG system that we just... This APG system or PPG system, whatever system that we follow, these are continuously evolving. They are not final in themselves. And again, they are based on few selected gene loci. They are not based on, because now full genome sequence has come. And you have seen in the lecture of Snyder, he was mentioning, yes, they have worked out the full genome for 10 species or 20 species which he was mentioning. And it is mind-boggling, because they are unable to come to any conclusion based on that, and what was concluded earlier based on few gene loci. So therefore, when the sequencing comes cheaper, that's why Earth Biogenome project is there, and many countries have participated, including India. And when full genome sequence is done, the classification will go totally different. I am 100% sure. Not only APG system, PPG system also, and even bacterial system also.
Yes.
This is what I want to say.
I think Sudha Hegde madam would be able to make more on that, because she's power packed. We have seen yesterday and today also. So in this aspect, so what would be the approach we have to be adopted in? So she's a classical turned to molecular biologist.
Before I deviate the whole discussion towards molecular biology, I would also like to have the opinion of another leading taxonomy.
Yeah, thank you, ma'am. And I also thanks the organizers to call me here. As Dr. Shinjappa has told already, we are just out of boundary. We are not pterophytes, but we are the classical floristics, and we work on the different floristic studies. But the much apprehension of the flora of pterophytes, flora of India, or the flora of India that I've seen actually, I have observed. And we also assure for that we will, whenever we go for that, we will definitely go for a more stable classification of pterophytes of India, and we'll do that, we'll follow that. Because on the background, if I say that, even Dr. Sanjappa and Dr. Rao also knows that, because last time in RMC meeting, that is Research Monitoring Evaluation Committee meeting of BSI, it was highly criticized to BSI authority that they say that the country cannot wait for 70, 80 years to have a flora of its own. Because that's the main cause we have initiated immediately in 2 years back with the present director's guidance to bring out the flora of India and also the flora of the pterophyte flora of India. If you'll see that, we have all the manuscripts of ours, and the manuscripts has been submitted by the very renowned taxonomist of the country, though maybe these manuscripts are a little bit old, and these manuscripts need to be revised in the sense of the nomenclature issues, and also we take the permissions of all the older or senior colleagues regarding this, and we are doing that, and hopefully by the year of maybe 1 or 2 years, gradually 1 or 2 in series of flora of India will come, and we hope that we maybe by 2 years of time we will complete that. We have given this assurance to all our seniors and also to the ministry. And regarding the questions what Dr. Kulhar has given to us, he definitely will look into that, and the classifications what we talking about, we will definitely look into that, and we will definitely take the opinions of all the persons of all the big pteridologists of that, and we will definitely adopt a most stable classification, because the main aim to have the flora, the pteridophytic flora of India, rather than to go for any controversy or rather than to go for any sort of other way for making a classification. And I have also heard regarding all the deliberations what the last 2 days has given regarding the pteridophytic research in India, and what I see that many of the things has been based on the many of the applied subjects like cytology, or you can take the example of the isolates. When Dr. Gosai was talking, he was reiterating about the isolates of the last problem, isolates of the Peninsular India. And regarding Dr. Unyal also given a brief, complete, elaborated description for what are the challenges for us. The most important thing that, and all the authors and all the speakers, and even if my panel members has agreed to that, the most important thing is the collaboration. I take this is the most important challenge before us, how to go about to collaborate with other scientists, because even in the angiosperms or in the pterophytes. So this is the most important thing, and I also invite all the persons or those who are able to submit any of the manuscript with them, so we will definitely accommodate those genus or those families in the coming pteridophytic flora of India. And for this, the nodal officer, 2 of the persons has been engaged from Botanical Survey of India, Dr. B.S. Korea and Dr. A. Benjamin is looking after this, and they may contact them and they can share their own manuscript. Because whenever we're talking about the collaboration of projects, and we are just handing off the financial structures, because this is a national cause, and we must come forward, and everybody should come forward, and what are the manuscripts they have with them, they should give it to us, because this is a national cause, and the country, we understand that the country cannot wait for the last around 7 years to have a flora. And about how to go about this flora, we have more and more stable manuscripts with us, like flora of Meghalaya, flora of Nagaland, and flora of Tirap district by Panigrahi and Sarnam Singh, and Western Himalayas flora of Professor Kulhar, and flora of Arunachal Pradesh, because if I'll go back to something, a lot of collections has been made from the Northeast India and also from the Arunachal Pradesh in recent time. And Dr. Benjamin, as Dr. Sanjay was told that, Dr. Benjamin was engaged to collect all the specimens from Polypodiaceae and Aspediaceae from Northeast India. So he has collected, extensively collected from all the specimens, and now those things has been identified and by the series of experts, and we have a complete list. And but what the Dr. Kular is saying about we should not follow the same to same what Dr. Fraser Jenkins has given. We definitely will look into that, and what are the stable classification is there. We will adopt that, and we will follow and bring out the flora. And rather, and the rest of the things, sir, what I mean to say, the last decades, when we were talking about the taxonomic impediments, this is mainly because of the lack of the expertise or lack of the collections from the different areas, or many of the experts who has been already retired. That's why this has been taken out. But however, with the changing of our time, and all these manuscripts what have been submitted to us, and what are the persons to those who worked hard in the last 2 decades, this is fading up. That's why if we will right now we will not bring all these floras as in when, this will not pave a way for our young researchers how to go about it on the next generation things. That's why that is also another cause of things. And another thing is that this is all the things of the integrative taxonomy what I'm talking about, because in the last 2 days of deliberation, what I observed that the taxonomy of pteridophytes is not only depends upon the descriptive taxonomy, but also it depends upon the classification, depends upon the your karyological studies or phylogenetic studies. That's why we expect, and also we need your support for bring out these floras, and as and when I request to all the senior colleagues here and all the senior professors here, if they will come forward to help it out, and definitely we do that. And what Dr. Punyata is told about to bring out to call for meetings for this, how to go about it, definitely we will do that. And I'll send this message to my director, and very soon we'll going to arrange that one. Thank you, sir.
Thank you, sir. Now, madam.
Thank you so much. As usual, they keep the best for the last.
Yes.
Yeah. About you warning the audience about me speaking, I'm reminded of a small incident. Professor Kular had invited me as a speaker in Jammu for the Indian Science Congress, and we were at the airport, and there is a big sign in Jammu airport which says, "No sharp items allowed beyond this point." And my husband told me to shut my mouth because they might take my tongue. So I'm reminded of that. I'm sorry if I distracted you. However, that was for people who were feeling drowsy. Coming to a different perspective altogether, whenever we talk about research, research is not only about getting good funds, but also getting good manpower. Quality manpower is also an absolute requirement. And another area in which we need to pay attention to is the dwindling interest in young people about pteridophytes, in pteridology. So how can we kindle more interest in pteridologists? And as all the eminent people on the dais have given us directions, maybe the future would be to collate classical taxonomy with molecular biology. So initially, the diversity studies, etc., were done with the isozyme analysis. So isozymes were the ones that were giving us a difference. So after that came the molecular markers. And in molecular markers, they were largely used to augment whatever data is there by taxonomists to, reiterate it, or to find out what is the reason for difference. Molecular biology was being used. However, in the next generation, as I foresee the taxonomy area, as and when molecular biology is getting more and more refined, like in medicine, it is going to be individual-based medicine. So each person, based on their genome, will be given a different medicine. Same way, in future, molecular biology will come to that level where you will not only be able to identify species, but different individuals in the same species. So this is going to only increase. It's not going to decrease. Whether we like it or not, it is going to get more and more magnified, and because we are having a barrage of information. With all this sequencing coming into the surface, it is here to stay. Whether we like it or not, molecular biology intervention is here to stay. Another aspect that I wanted to talk about is molecular biology can also be used for another very important purpose of conservation. So in Kudremukh National Park, they were saying that there are these group of ferns, and there is one more area, but we want to develop one area into some tents or some tourists, so we are going to destroy this area or this area. Which area is more diverse? So doing DNA fingerprinting, we will be able to tell that there are 100 individuals here, but all of them are of the same genotype, or they may be having a common germplasm. But in this area, there are about 50 of them, but each one has got diverse genetic makeup. So it is worthwhile focusing in conserving this particular area over the other, because development is going to happen. So maybe molecular biology will help us to focus more on where we need conservation based on the data. We can assess the genetic diversity of the given population. And another story I'm going to tell you, there was a person, of course it's a fictional story, there was a person who wanted to find music in violin. So he took the violin, broke it to pieces, and then declared there's no music in the violin. So whole genome analysis is something like that. You take the whole genome and break it into pieces, and then you sequence it, and say, okay, we got so much information. Where's the music? Where's the music? So the future is going to be not on whole genome analysis, but in gene expression. How is it going to create music? The next level would be transcriptomics, and in that, the RNA world perhaps will give us little more information and make us wiser than just running behind whole genome analysis. The molecular biology can also help us to understand developmental biology. Professor Kula was asking Dr. Sara, where is this first division of the cell when antheridia, or how do you know where this cell differentiates? So, yes, what's the shape? Yes, I get. So to get that total picture, if I were to tag a molecule which will make the wall, so using molecular biology, if we tag, we are able to monitor the entire process, like we are doing how to find a block in the heart. You're going to inject a dye. Wherever the dye goes, it will tell us where the blocks are. Same way, you take a developmental gene and then tag the RNA expression of it, and you will be able to screen where this gene is expressing, what it is causing, what differences it can make. So it's a very powerful technique. So I always say this, please don't consider it as a casteist statement. I say science is never wrong, but the scientist interpretation may always be wrong. So in Sanskrit we say, Shastra tappirudilla, Shastri tapp madbo. So I don't intend to use any caste statement in that. I'm just trying to say molecular biology is just a tool. How we are going to make it or use it, it's like fire. You can cook your rice or burn the neighbor's house. So it's a very powerful tool, and we should learn how to modify it and use it for our advantage. So instead of looking at whole genome analysis, maybe developmental studies and expression would give us much more information. Then coming to phytochemical analysis and novel products, a small note of warning is everything green need not be safe, because the most potent toxins in the world are plant-based. So cyanide-like compounds ferns are known to make. So I respect these plants a lot because they are very, very potent, and we need to be very cautious before dealing and jumping and saying, this is medicinal, okay, use it, and then it kills a cell, fine. But what is it doing to a normal cell? That also needs to be studied. and yeah. Yes. So they have what to say, a warehouse of unknown composition and unknown potential. So before we jump and say that it is medicinal, we have to have a lot of respect and tread with caution. Another upcoming aspect in pteridophytes is phytoremediation, where we use plant tissues for removing heavy metals. And with this context, I would also like to say, those who are working on climate change, pteridophytes will offer you a good opportunity, because they were there on surface of the earth much before we came, where the conditions of the world was more hostile, and I'm positive they will survive even after we go. So they are here, they were there long before, and they have survived this hostile condition. Maybe over a period of time, to adapt to variations, their genome has modified and accumulated and become such a redundant volume. So it is very interesting to know where are the genes that are involved in tolerating radiation, using heavy metal pollutions, etc. Then coming to another compound, plant, I told you they are very potent toxins. So we need toxins for benefit of mankind. In which area? Agriculture. So biopesticides, where they are being used, which is not harmful to us but harmful to an insect. I saw a couple of posters in that line. It's an upcoming field, but do not be happy just going by the effect of the extract. Try and find what compound it is, and many of them are getting patented. The genes have been isolated, and it is being portrayed as just next to Bt cotton. So you have this Bt gene, Bacillus thuringiensis gene. So the next one, TMA gene from the misnomer of Tecteria macrodontia, TMA13 gene has been isolated, which is a very potent chitinase and is known to kill the whitefly. Transgenics, Indian NBRI has done it, Pradyumna Singh, and it's a patented technology. A lot of scope is there in other such genes may be there, which may help mankind to store our rice without getting weevil, as one of the posters said. So kindly look into the climate change, and these are some of the aspects. And to conclude, I would like to say that when we talk about funding and science, when they see a rocket being launched in Mars, and then we have so many satellites, 128 satellites being launched one day, very happy, and that institution will get so many 1,000 crore grant. And a lion's share of India's R&D grant will go to that. But let me tell you, one P.V. Madhusudanan launched so many rockets, so many missiles. And because of one such person, 40 years down, we are having this department that is thriving. So we teachers in the universities are launching so many human manpower missiles, but we need to give them funds as well in terms of fellowship, in terms of sustenance. So that is my appeal. Not only do we need funds, we also need quality manpower. So all the youngsters there, hang on. These are some of the areas in which you can work with. Thank you.
Thank you, madam. Thank you very much. Actually, I have also already crossed the time allotted to me. So let me conclude, and let me thank all of the respected experts. So to conclude, I think you may be agreeing with me in concluding that our need of the time is to complete the Fern Flora of India, either by BSA or with the help of all others. Then we need more persons also. With different perspectives, we will continue the studies on the teratologic research. I think with these words, I am stopping here. Thank you, sir. Thank you very much.
Thank you.
A memento as a moment of talk as a love token to Dr. S.S. Das. Sir, please. Now I request Dr. Smita Hegde to present a memento as a token of love to Dr. Sangappa M.
I get more time with her to argue many points.
Now I request Dr. N. Punita to present a memento as a token of love to Dr. Raghavendra Rao. Sir, please. I request Dr. S.S. Das to present a memento as a token of love to Dr. K.P. Rajesh. Sir, please.
Yes, it's all. Okay, sir, we shall continue the discussion over the food or the tea. So they have to move.
Thank you so much. Thank you all. Now, we are moving to the Future Scientist Award competition, and I invite the judges on the dais. Dr. A. Benyamin, Dr. N. Punita, Dr. Raju Anthony, to be please seated on the dais.
to the interaction among the biotic and their abiotic factors in the microenvironment. So, I'm going to talk about the plant population ecology and demography because it can provide insights among the interaction of species with their particular microenvironment, and regarding that, we can come down to the analysis of physiology and anatomy of plants and present it according to the soil profile. So the aims and objectives are to examine the effect of alternating edaphic conditions on plant growth pattern, to quantify the trace elements present in different compartment sections of plants, plant tissues such as leaves, stems, roots, and to examine whether this affects their stomatal behavior. So the materials and methods are, we chose 5 study areas among the BDU campus, Bharatdasan University campus, and then we applied quadrant methods to analyze the population and to analyze the species richness, abundance, and their frequency. And then samples were collected, soil, roots, stems, and leaves for the analysis of trace elements and the stomata. So for analysis, we used ICP, inductively coupled plasma, and the results were obtained. And for stomatal analysis, we used the procedure given in the paper by Paul et al. 2017. So the results and results obtained were, the vegetation diversity and individual abundance were recorded, and in the 5 study sites, among the 5 study sites, in the study site 2, you can see that species richness, that is number of species present, were 11, and the total number of individuals were 166, and it topped the chart among the 5 study sites. The most abundant species was Cypress deformis. Some of the other species present across the 5 study sites were these species. Totally we found 52 species, different species, and 28 of them listed here were very common. And you can see there are many different types of habits, such as annual herb, perennial grass, short-lived cool season perennial grass, and subshrubs, etc. So when we are talking about the trace elements, the bioconcentration factor plays a major role because it indicates the concentration of the elements in the soil. And the chart was topped by, again, study site 4. As you can see, aluminium, chromium, and ferrous were the highest among these study sites, followed by study site 2 followed with nickel, cadmium, and copper. And then lead were always, it was always between 6 to 7.5 in all the study sites. So the overall decreasing order was study site followed by study site 4, followed by study site 2, then study site 5, then S1, and then S3. So no matter how much concentration of this trace elements is present, some part of the plants can compartmentalize it and distribute it according to the requirements. So this slide presents the translocation factors of trace elements in the studied individuals. And you can see that in the table, the Fe had the value of 0.04 in study site 5, H. contortus, and study site 3, C. odoratus, and the following table should be read like that. And the PC analysis for the translocation factors for leaf stem, stem root, and leaf roots were also done, and it is presented as a PCA. Next, this is the hierarchical clustering dendrogram obtained.
We can give another 1 minute. Huh?
Okay, sir. Thank you, sir. So this is the analysis for the translocation factors in leaves root and stem by root and leaf by stem. Overall, you can see that S4 is always like it has a most trace elemental concentration. Moving on to stomatal analysis, when we see that, you can see that we chose 3 main plant species, that is Acacia penninervis, Eriocolon decangulare, and Cyprus deformis, because these three were common in all the study sites. So as you can see, APS4 had the highest somatal density when calculated, that is 16.470. The same went for CDS4. It has the highest somatal density of 11.464. So this coincides with the trace elemental concentration.
Can you show what are the plants you have selected for the study? What are the plants you have selected?
Pictures?
We didn't see any ferns in your
So yes, sir, this study is not actually related to ferns, sir.
Okay, okay.
Yes, sir. Thank you.
Okay, thank you, Mrs. Darshini. Now that time for questions. If anyone asks any questions, you can ask. Take 1 minute. 1 or 2 questions only for audience.
Okay, sir. Yes, sir.
When you have enough grasses,
yes, sir.
You can always find a bird.
Okay, sir. Yes, ma'am.
Frequency you have given there, but could you see in the IVI value, because it is very important when we are quantify the ecology.
Okay.
So it is the important value index.
Okay.
So, and another is you were showing the stomatal density, not
Yes,
the
ma'am.
frequency.
Yes, ma'am.
So generally we are doing the stomatal frequency, so, and there is no statistical standard error or deviation you are showing in your data that I have observed.
Ma'am, because we have to
So
further
EPCA
carry out
you have
this
done, but you are not enough clarify or given the discussion about that, because 2 or 3 lines I have given, they have the negative line in
Oh.
your PCA graph.
Yes,
so
ma'am.
you have not described enough that. That's all.
Oh,
Thank you.
oh, thank you.
Okay, thank you. Thank you very much. Next, I would like to invite Govind. He is going to discuss about ethnomedicinal importance of pteridophytes traditionally used by tribal people of Nilambur Forest, Western Ghats of Kerala. I request Mr. Nipun Govind. Okay. I think, yeah, FSA02. Otherwise, we will go to the next participant. FSA3, Sujit. FSA03. She is going to give the presentation on isolation, purification and characterization of an antioxidant proteins of Nephrolepis cardifolia. She is also not there. I think she is also not arrived. Next, FSA04, Ajay Babu. Yes. Yeah, Mr. Ajay Babu, he is going to discuss about mapping of pteridophytes flora, Dharmadam region of Talaseri, Kerala.
Good evening, everyone. I am Ajay Babu. I am pursuing my post-graduation in botany from Government Brandon College, Talaseri. Today, the topic is mapping of pteridophyte flora of Dharmadam region of Talaseri, Kerala. We know that pteridophyte is included in all botany syllabus across our country. Beyond the simple identification of this plant group, nothing much is appreciated in our course. And this this plant group is facing biodiversity loss. So in this background, there is a need to assess pteridophyte flora in our locality. So that's why we move forward with this work. When coming to objectives, to map the indigenous pteridophyte using online and offline mapping tools, taxonomic assessment of local pteridophyte flora, to develop the resource materials for the systematic study of pteridophyte flora. Conservation strategies, both ex situ and in situ, are the main objectives. When coming to the materials and methods, the study area is the Dharmadam. Dharmadam is situated northwest to Talsheri. It is a small island, and the other details are given. And coming to methodology, first of all, we mark the area of study and done extensive field visit, and locating the target plant with the help of GPS tools like NodeCam, GPS camera light, and the photographs were taken. And the identification of each plant is based on flora and expert opinion. And the main procedure of this process is mapping of this identified taxon. It is done based with the help of softwares like ArcGIS, Google Earth Pro, MyMap, etc. And finally, herbarium preparation and ex situ conservation is also done. Coming to result, 19 families were identified. These are the list of 19 families. And I am discussing each family in single slide. In this slide, we can see the Lycopodiaceae family, which include the species Lycopodia lacernoa, and it is a geographical image of our study area. Study area boundary is marked, longitude, latitude, direction, and all other details is given in this map. And the Lycopodia lacernoa is present only in one locality in our study area. The next family is Selaginella. Species like Selaginella wildenoi, involvens, crassipus, and other Selaginella species is present in our area, having a moderate level of distribution. Next family is Marataceae. Angioteris evecta is the species. It is present only in a single place in our locality. Schiaceae family, having Lygodium fluxosum species. It have moderate level of distribution in our area. Next family is Pteridaceae. Pteridaceae is the largest family having highest distribution in our area, which include the species like Acousticum aurum, Pteris vitata, Pteris confusa. And next family is Parkeraceae. Parkeraceae having Ceratopteris thalicroides species, which only present in a single area. Then Parkeraceae. Then next family is Hemionetidaceae, which include the species Pterogramma calomelanos. It is having a moderate level of distribution. The images of circinate vernation and sporophyll also shown. And the next family is Adiantaceae. Adiantum philippines and Adiantum latifolium are the species. It is the second most largest family having higher distribution in this area. Then next family is Linciaceae. Lincia ensifolia is the species which is present in 2 or 3 nearby areas. And the next family is Oleandraceae. Nephrolepis exaltata is the species present in only 2 areas. Cyatheraceae family, which include the species Cyathea gigantea, present only in single area. Then Glycinaceae. Glycinaceae having Trigonospora linear species, which is present in 2 or 3 nearby areas. Telipteridaceae having species Cristella dentata, it having moderate level of distribution. Then Aderaceae, Aderium hohenacarianum is the species only present in a single area. Blacknaceae, Blacknum oriental is the species, and it having a distribution in 3 or 4 nearby areas. Then Polypoidaceae, these are the epiphytic pteridophyte group, which include the species like Drinaria curcifolia, Pyrosa lanceolata. Then Marsilaceae having Marsilia minuta. Next Salvinaceae. Salvinaceae have Salvinia mollusta and Azola rubra. Salvinia mollusta, we know that it's a weed, but it has a controlled growth in our herbarium garden. And next pie chart showing about the distribution of the family and the distribution of species and the habitat diversity. Most of them are terrestrial and some of them are halophytes, epiphyte, aquatic, etc. And some of the Nephrolepis species which is used as ornamental plants. And the image that we conserve ex situ conservation of the narrowly distributed plants. Then coming to discussion, the species like Adiantum philippines, Pteris linearis, Pteris vitata, Selaginella species having common distribution in our Dharmadam area, and the species like Lycopodium cernua, Angiopteris evecta, Cyathea gigantea, Aderium hohenacarianum having very narrow range of distribution, and some of our ornamental plants are also present in our locality. And coming to the future study, the reassessment of pteridophyte flora of this region, and we want to do the fine mapping of the pteridophyte members in this area, preparation of pteridophyte flora, bioprospecting, conservation, and DNA gene bank are the future study ideas, and these are the references. Thank you all.
Thank you, Mr. Ajay Babu. It's a very nice presentation. So, from audience, if you want to ask any questions, most welcome. Only 2 questions.
Because you are the beginners, so in your slides when you are showing the figures, so you please put the author citation properly there.
Okay, ma'am.
So keep it up.
Okay, thank you, ma'am. The Irudayaraj sir published a book in 1992 and followed this West Ghats, and
Ajay, it's a very nice presentation. I'm very happy about your presentation. I would like to ask one more questions. So as you have collected 20 species from that your area, out of his maximum number of species has been reported from particular area, right?
Yes, yes.
What is the reason that maximum species are present in that particular area? Can you explain any reason for that? What is the reason?
Most of the species is identified in a particular region because this land area having laterite type of soil, Glycinaceae family and Angiopteris, Cyathraceae, Lincia and Lincia family are also present in this common area. It's a landslide area, so having laterite soil. Blacknum is also, Blacknum oriental is also present in that area.
Okay, thank you.
10.68 square kilometer.
Very small.
Square kilometer. – 28 species. Thank you.
Next, I request Priti Giri, FSA 05. She is going to discuss about phytochemical analysis of Nephrolepis cordifolia and Nephrolepis biserrata.
Good afternoon. I am Preeti from Delhi University. Today I will present about phytochemical analysis of Nephrolepis cordifolia and Nephrolepis biserrata. And next I will discuss about the general introduction. Pteridophytes includes ferns and ferns alliae, and they are the first non-flowering vascular plants on land that reproduce and disperse through spores. Pteridophytes are divided into two classes, Lycopodiopsida and Polypodiopsida. According to Grumbridge and Jankin, there are about 13,025 species present in the world, and India harbors more than the 12,000 species in 191 genera, distributed mostly in the Himalaya region, central India and western Ghat. In the recent year, people are shifting their trust from synthetic drug to plant-based product medicine according to herbal medicine. According to the WHO report, nearly 70 to 80% of the world population relies on the herbal medicine. And according to Herbal Medicine Market Research 2020, Europe accounts for the largest share of the world market for phytomedicine, followed by the Asia Pacific and America. According to FICCI reports, India has low export of medicinal plant, about 63 billion USD, which is just about the 2% of the global trade against 30% of the China. Why I am selecting Nephrolepis cordifolia? Because these plants have the various ethnomedicinal importance. Nephrolepis cordifolia belongs to family Nephrolepidaceae, commonly known as fishbone fern, and it is also used in the diuretic, contraceptive, and also used in the treatment of skin disorder, scabies, sinus problem, toothache, cough, chest pain, jaundice, diabetes, liver disorder, and paste of the fronds is also applied to stop the bleeding from a wound, and freshly water tuber of the Nephrolepis cordifolia are consumed to quench thirst and also used in the treatment of stomach ulcer and acidity. Tubers are partially boiled and used as a vegetable salad ingredients. And the ethnomedicinal usage of Nephrolepis biceretia, it is used in the treatment of dysmenorrhea, wounds, boils, abscess, and blister. And young frond of the Nephrolepis biceretia also used in the preparation of vegetable salad. And objective of my study, objective to determine the biochemical composition of frond, it includes moisture content, ash content, crude lipid, crude protein, total carbohydrate, total energy, and objective to determine the amino acid profile of the fronds by LCMS according to Dick-Java Baker et al. 2015. And objective 3, determine the secondary metabolites of selected fern. That include total phenol content, total flavonoid content, total polyphenol content, total tannin content, and objective for identification of the functional group by using FTIR. And material method, plant samples were collected from Department of Botany, University of Delhi. After that, plant samples were thoroughly washed under running tap water and dried at room temperature. And after that, samples were powdered and stored in glass bottle at 4 degrees Celsius for further biochemical analysis. And these are the result of the objective and proximate composition of the presently studied one. And Nephrolepis cordifolia has the higher amount of the moisture content and ash content as compared to the Nephrolepis bicereata, while protein content, crude lipid content, carbohydrate, and energy content was found significantly higher in the Nephrolepis bicereata. And the result of the objective 2, amino acid profile of the studied one, in which Nephrolepis bicereata have the higher amount of alanine, arginine, glycine, leucine, valine, methionine, phenylalanine, tyrosine, and proline as compared to Nephrolepis cordifolia. And the result of the objective 3, quantitative estimation of the secondary metabolite, in which phenol content, flavonoid content, and tannin content was found significantly higher in Nephrolepis cordifolia as compared to Nephrolepis bicereata. And objective 4, Fourier transfer spectroscopy is an analytic technique to identify the functional group, and it is used as an absorption and emission to detect the functional group and obtain data in the form of sharp and instant characteristics at different wave numbers. And these are the result of the FTIR of Nephrolepis cordifolia. Out of 20 peaks, there are the 7 different groups are found. Number 1, aryl disulfide, alkali halide, protein, aromatic compound, hydroxy group, and alcohols. And in Nephrolepis bicereata, various out of 25 peaks, 11 different groups are present, that is protein, alanine, carbonyl compound, ketone compound, transition metal, and terminal alkyl. Discussion and conclusion. And the result of the present study developed novel phytochemicals marker to identify the medicinal important plant. And I highlighted the main point of the discussion because essential amino acid, that is histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine are not produced by the mammals and other animals which are given in the form of diet. And the future aspect of my study is the Nephrolepis bicereata and Nephrolepis bicereata are grown naturally without the use of synthetic fertilizer or pesticide, and also really excellent source of protein, amino acid, and natural antioxidant. And based on our finding, it can be ascertained that nutraceutical potential of Nephrolepis cordifolia and Nephrolepis bicereata will be useful in selecting plant as a nutritional supplement as well as in developing antioxidant-based drug. And further advanced spectroscopy study are required for the structural elucidation and identification of the active principal compound in the leaves of Nephrolepis cordifolia and Nephrolepis bicereata. These are the selected references, and thank you so much.
Your good name, this Bhanupriya. Your good name?
Yes, sir.
But in future presentations, you for future presentation, you need mention the voucher specimens. And every phytochemical studies or any advanced studies, before going to that, you have to prepare a voucher specimen.
Okay, sir.
And in presentation, you should have a voucher specimen should be designated.
Sir, I have submitted in the Delhi University Herbarium.
Okay, that is nice, but you have to mention in your
Yes,
presentation.
okay.
Yeah.
Thank you.
Any question from audience? Next speaker is Bhanupriya R.
Good evening. My name is Bhanupriya, coming from Sacred Heart College, Thevra. I have done my work on Lygodium, a comparative micromorpho-anatomical studies of the genus Lygodium from Kerala. Moving to the introduction, the Lygodium belongs to the family Lygodiaceae and is a terrestrial with slender stem alternately pinnate climbing by twining branches. And two species are reported in Kerala, that is Lygodium flexosum and Lygodium microphyllum. And moving to the objectives, and my objectives are to identify Lygodium species based on the leaf morphology and to study the anatomical variations, to determine the germination period of spore and pattern of gametophyte development, then to determine the spore morphology and also to approximate the sequence level variation in the tRNA sequence of the Lygodium species from Kerala. Then moving to my methodology, then I started with the morphological study of the selected species, then I moved to the anatomical study, spore analysis, gametophytic analysis and in silico analysis of Lygodium species tRNA sequence has been done. Then moving to results and discussion. And in my first objective, to identify Lygodium species based on the leaf morphology. And you can see that in Lygodium flexosum, the pinnules are lobed, and moving to the Lygodium microphyllum, the pinnules are unlobed and the midrib is not prominent. And moving to the next objective, to study the anatomical variation in Lygodium flexosum. In the rachis anatomy, you can see it is T-shaped, and whereas in Lygodium microphyllum, the vascular structure, the xylem is more prominent and in solid mass present in the center. And in petiole anatomy, you can notice a peculiar feature, that is in Lygodium flexosum, the presence of chloroplast can be seen in the central region, that is in the vascular bundles. So I don't know why it is present, and I'm still searching why the chloroplast is present in the center, and whereas that it is absent in the Lygodium microphyllum. And moving to the rhizome anatomy, in Lygodium flexosum and microphyllum shows similar structure, and where in Lygodium flexosum the epidermal hairs are prominent. And in root anatomy, in the Lygodium flexosum, in the inner cortex region, the sclerids are more prominent, which is absent in the Lygodium microphyllum. Other than in both, the vascular bundles are similar, both are diarch. And in Lygodium microphyllum, you can notice there seems like starch granules are present in the Lygodium microphyllum. And I have observed when I had taken the sections of Lygodium flexosum, when you take the sections from the base to the apex region, and we can see some slight variations in the anatomy. And moving to the stomata, in Lygodium flexosum it is anamocytic, and microphyllum it is diacytic. And moving to the third objective, the germination period, when I observed the germination, I could find that in Lygodium flexosum it is symmetrical, and in coming to Lygodium microphyllum it is asymmetrical, and it takes much time to, compared to flexosum, it takes time to germinate. And then moving to the spore analysis, so sorophore microscopic view, you can see the Lygodium flexosum it is closely arranged, whereas in Lygodium microphyllum it is raised, and pseudo-induciums are also observed. Then moving to the spore analysis, and in Lygodium flexosum the spore is trilate and isosporous, whereas in Lygodium microphyllum it is subtriangular or circular. and reticulate exposure can be seen. Then I have done the SEM analysis, where also you can see that varicate venation can be seen in the Lygodium flexosum. And in microfilm, reticulate ornamentation can be seen. And then I have done the in silico analysis in tRNA sequences. I have taken 90 samples from the NCBI, and then have made the UPGMA tree, and using Mega software, and then cluster W were used, and then 62 different nucleotide sites variations can be seen. And in coming to the future study, you can see in future study we can do the cytology, and cytological works can be done, and also in hybridization techniques of Lygodium can be analyzed, which is not done much. And so I am planning to do the cytology of Lygodium. Then conclusion, the current study fulfill all the objectives mentioned in the report, and the work also reports the anatomy of rhizome and root of Lygodium flexosum for the first time. And the current study is useful for the systematics of the genus as well as for the phylogenetic purpose. Thank you. Final results. Then anatomical studies of variations can be observed, and I have we can easily distinguish the Lygodium flexosum and microfilum on the basis of micromorpho anatomical features. Lygodium microfilm is more advanced, I think, because more sclerids are present, and it is more having biological control agent can be used. But in Kerala, Lygodium flexosum is more flourished. And Lygodium microfilm, that is Lygodium flexosum. Lygodium flexosum.
Is advanced?
That's why it's getting more flourished. Because Lygodium microfilm is preferably grown in wet regions, moist regions.
Do you find any character in anatomy? Primitiveness or advanced? Which type of steel is primitive and which one is advantage?
Both having protosteel
and which type of steel you observed in your anatomical study?
Protosteel.
Protosteel. And you have done the germination of spores.
Yes.
And whether you done in vitro condition or in vivo condition?
In vitro condition.
So can you explain that why you have chosen that species of Lygodium? There are so many species are there in that area. What is the reason for that?
Because I am interested during my PG life, I observed the saurophore. And I just started to search whether more species are there in Kerala. Then later I have identified that Lygodium flexosum and microfilum are only present. Then I just want to know why these two, what are the main anatomical features. And when I searched also, there is not much work in this species. Only Inversinus are studied in California and all.
Okay. Thank you. Any questions from audience? Sir?
Question may be a bit very critical that way. If you go back to the anatomical slides then. Well, come on, yes, this is by the way, you have the rhizome anatomy. And rhizome anatomy, if we agree with these, that two are entirely different from one another. And the left side one, what Lygodium flexosum says that so many hairs or scales they are present on rhizome is difficult to accept. Number one. From what locality it has been collected is not known really. Well, the root anatomy is quite okay. It is a dire condition. There is just one metaxylem in the center. And there are protoxylem elements, 3 or 4. Next, please. Next slide, the rachis anatomy. Come on, the next. Yes, the rachis anatomy, actually Lygodium flexosum and Lygodium microphyllum that you have got the rachis anatomy, these are not at one level of the two species. These are entirely different one.
Yes.
The rachis anatomy in Lygodium flexosum that you took is from a little higher up where the branching or the appearance of the first rachis branch comes up. And this Lygodium microphyllum, this transverse section is from just at the base of the primary rachis, or what we call the climbing rachis. So it is so different that this is rather difficult to accept that at this, these are from the same level. And this is very important. Similarly, petiole anatomy, I do not know what you want to refer to petiole. As such, there is no petiole in Lygodium. You know, the rachis, what you call, is the climbing rachis, we call rather, and then this gives away alternately the primary rachis branch and the secondary rachis branches are then formed when the primary rachis branches ceases its growth, then a pair of the lateral primary rachis secondary rachis branches, which are folia, they come out. Can you go on to the morphology that whatever you have given? Can you go on the first or second slide, the morphology is? Yes, the apical bird, this is exactly more the apical, what I want to talk you. You see the primary rachis you have written. You have written primary rachis here, but you are not talking primary rachis in your anatomy. The anatomy of primary rachis altogether that is different from that of the climbing rachis. There is no petiole as such in Lygodium species. This is very important in differentiating the Lygodium, this particularly the Lygodium, from those of the rest of the Cygiaceous members. This is very important, and it has its own significance. Okay. It has, you see the apical bud, that is not really the apical, that is the dormant bud. The dormant bud on primary rachis branch. What do you mean by stalk is not, that is the stalk really. If you go, if you follow the nomenclature of Holtam of 1959, Flora Maliciana, he has given very clear picture of the different parts of morphology of this Lygodium, rather Cygiaceae, to complete Cygiaceae, as well as the Glyciniaceae there. So you have done that way very good work as far as since you are the MSc students, or so if you want to do further work on it, and you should do, I am sure, much work is to be done particularly on the proliferation of what you call is the apical bud. That is not the apical bud really. That is a dormant bud on primary rachis branch. Its proliferation is required here. That is very important. In all the Lygodium species it is present, whether it's flexosum, japonicum, or you call microphyllum. That is important. Since you have got two live species, you can work on it. But please follow. If you don't have it, I can send you the literature from Malaysia. And I do not know whether I have noted your address from here. If you so require, you can ask me. I can send you the literature on it. Thank you.
Sure, sir. Thank you. Thank you so much.
Yes, madam. Yes, please.
You have done the sequencing of this leaf. That sequencing you have done NBCL.
Yes.
So
taken from retrieved from NCBI.
Yeah, show me that slides where you are showing that. So what percent they are matching? Is there the bootstrap map? Okay.
Yeah, Lygodium flexosum shows intermolecular and intramolecular variations.
And these are the bootstrap of those species matching. Those percent is showing the matching. Bootstrap map is, they give you the bootstrap map with the species sequencing. This is the bootstrap map.
Okay.
What percentage they are showing more closely related? It is not clear here. What percentage they are showing the similarity? It is above 80 or
76%. Is it?
Okay. Okay. That's one query. And Your anatomical work is very good, but one thing is needed for putting the bar gram.
Okay.
Because,
Thank you.
Okay, thank you very much. Next, I would like to invite Linto Paul Jacob. FSC 08.
Good afternoon, everyone. Myself, I am Linto Paul Jacob. I am pursuing my master's in plant pathology, a bit different field from pathology, but we'll talk about the importance. My topic is the phytopathological facets of pathology, the current scenario, the limitation of phytopathology and pathology, and the future. So, hope we'll start. Phytopathology is a subject where we study about the plant diseases, and we deal with the plant diseases of various families, and where we include the causes of a disease, the mechanism by the disease. The causes means disease is caused by a fungi or a bacteria or such pathogens, or the mechanism of the disease and the interaction between the disease with the plant, for say ferns, and the management of this disease. So we'll have a very quick rush into these topics in pathology. And a disease which always results in a damage of cells in plants, and the toxins and growth regulators are always produced during a disease, and the nutrients of the plants are used for the uptakes, which disturbs the metabolic activities of a plant. So when we're going a bit in the surface level of pathology, the disease triangle is proposed by Doc McPhail in 1960, which says a susceptible host and a viral pathogen and a conductive environment is always required for a plant disease to happen. The pathogen should be virulent, and the host should be susceptible, and the environment should be conductive for the infection. And confirmation of a pathogen, it is always done by Koch postulate. I'm not going deep into that. When we talk about a disease cycle, there will be an infection of a pathogen, and there will be colonization, then inoculation will be done, and incubation by the pathogen will be done, and the penetration of the pathogen to the host will be shown, and the symptoms and signs will occur, and again the infection. It's a cycle of a normal disease cycle. The inoculations will be transferred, it is always through the bacterial or fungal or spores or the mycelium. It can be transferred in many ways from a plant to another plant. Dissemination by natural as well as abiotic abiotic factors are also seen. When it comes to the bit deep into the attachment, penetration and infection of the pathogen, the fungal inoculation and the bacterial inoculation infect in a different way. The attachment of bacterial viral disease, which can be replaced by water. In case of nematodes, they directly through the plant body. The effect of pathogens in plants, the pathogens normally affect the photosynthesis, the respiration of the plant, and you can see the necrosis, and the defoliation will reduce the photosynthesis rate, and the respiration is displayed by the leaf blade will be infected by necrosis or chlorosis. And it also affects the translocation, and the translocation is affected by the pathogen in a bad way. And normally, as we are wearing a mask, plants show such defense to the pathogens. They have chemical defense as well as structural defense here. So the chemical structural difference by the walls, thorns, and the chemical difference by releasing phytotoxin, and that was A. Now we come into ferns. There are a few diseases reported, isolated and cultured from ferns. No such studies conducted in India till date in my knowledge. The pathological studies in pathology is very limited, and we'll discuss about very few diseases which was been reported. So first is bacterial blight. The pathogen is Pseudomonas gladioli, You can see the symptoms like translucent spots in leaf, and it enlarges quickly. And the management practices, in the sense these studies are done in US, and to an extent my work is currently going. I'm not going deep into that. So the management practice, in the sense, if the things is in a garden or in a laboratory method of cultivation, we can management is preferred. Then overhead waters and infected plants could be discarded. The second is graying. Graying can be due to drought or less in water in the root. The even levels in water molecules can contribute to the not droughting, and also root knot nematodes can also cause this infection. When it comes to foliar nematode will show dark spots in the base of the font, and it will be enlarged. The pathogen is nematode, and also root knot nematode are there. Then fawn lobing disease is there. It causes due to over-fertilization. Leaf-borne disease is there. The tip is died. It's also due to over-fertilization. When it comes to Phytium root rot, the plant is stunted, and the gray color is observed. It is also, the pathogen is Phytium. Then Rhizoctona blight is caused by Rhizoctona solani. It's a very famous common pathogen. The plant will be brown and webs will be seen. The management is, the practices could be done during the pathogen-free planting materials and the pasteurized potting mixtures. The limitations is that there are very less field experts in the phytopathology and pedology. As I already said, both are two vectors, and to collide it's a very different pass. Both interests should come together. That is different. So that is the difference. We have less lab facilities, and both are different. If agriculture sector and pedology sector to come together, it's a bit complicated. In the future, we know that the diseases are getting extended, the species are getting extended. So if a disease study is made, we can reduce the extension. Due to the pathological extension could be at least reduced. That is the, and the sources is Agricultural textbook, and Pennsylvania State University has conducted, they are regularly making studies on the ferns and the disease. Also the Florida University is also doing that. And thank you.
Thank you very much. So any questions from audience?
Yes. Should I go back? Rhizoctona solani was taken from the leaf. I'll go back.
But what is disappointing that you could not write the name of a fern on which it is here.
Sir,
I know you have already written somewhere that there is no combination between a pathologist and that of a teratologist. So, but it was easier for you. You should have identified this fern from anybody. You see, these are very common ferns. Similarly, you see that they are whatsoever the diseases they are, we are not aware of these. But we can identify these ferns very easily. So, otherwise, a good report, I think. This is a good report.
Thank you, sir. Thank you.
Definitely.
Sir, sir, currently I am working on Phytophthora capsicum in black pepper. So while testing I had found a few ferns. My study is currently going on. So we had collected ferns, so similar infection and isolation is going on. These Solani, that slide is of my own, which is from that we have gotten the rice field. And if we go for a study, we can see the Solani infected ferns from the rice near. Yes. No, sir. Currently, such a pointed study on pedology is not going on. I am working on pepper.
Is it naturally growing or a cultivated species you selected for your study?
My species, it was naturally growing. So, but there was a pathogenic plant during my survey. So I think I'm having the slide. The aspellinum, one of the aspellinum plant was there. So we are trying to isolate the pathogen from the fern of aspellinum. Yes, sir. Yes, please.
plant,
Yes.
especially tobacco or whatever, and see if it resembles exactly.
Sure, sir. Yes, sir. Yes, sir. Sir, it's not only Solanaceae, but every family is... yes, sir. Yes, to be frank. But currently the pathological studies in India, or not, globally it is limited to agriculture plants. Yeah. So both are two extremes, and to collate those vectors is a bit huge task. Yes. Yes, exactly, sir. Yes. Exactly. Thank you. Thank you.
I have a small question. So I have seen your abstract, you have mentioned that in India 1267 species are there. It includes 130 genera,
Yes.
of which 70 species are endemic to India. From where you have taken that information?
Sir, I think I need to... I need to refer, I think, sir. I'm sorry.
Okay.
Sir. Thank you.
I would like to invite Mr. Pradyuman. Yeah. No, 9 is not arrived. Not that 10 is not 11. FSA 11. He is going to discuss about phytodiversity of Pteris quadriata and Salvia mollusta. FSA 11.
Slide start ആവുന്നില്ല.
Good evening all. My name is Pradyumnan. My name is Pradyumnan. I am completed my PG from Calicut University. So my work is mainly based on mainly the phytotoxicity of two ferns, Pteris quadriata and Salvinia mollusta, with the help of cytotoxicity studies and GCMS analysis of these two ferns. We know pteridophytes, the vascular growers, considered as the one of the diverse and oldest groups in the plant kingdom seen in different habitats. In my study, I used Salvinia mollusta as one plant, which are under family Salviniaceae, which is a major weed that shows dominance over the other plant populations. Second plant is Pteris quadriata, under the family Pteridaceae, a terrestrial fern mesophyte, show a partial weedy nature inhabiting a larger area under affordable climatic conditions. Few reports are available concerning the bioherbicidal activities and cytotoxic activities of Pteris quadriata and Salvinia mollusta. The major aim is to investigate the cytotoxic potential of Pteris quadriata and Salvinia mollusta methanolic extract at various levels of applications. 4 major objectives are included in my work. These are extraction of methanolic extract from Pteris and Salvinia by Soxylate extraction, phytotoxic assay of fern extract, seed germination and growth using seed of cowpea and rice. Third one is cytotoxic and genotoxic assay of the fern extracts. And final one is the GCMS analysis of methanolic extract of these two ferns. The A is the first material, Salvinia mollusta, and B is the Pteris quadriata. And the experiment, first one, the experimental setup of germination bio is is, and the last one is the experimental setup of cytogenotoxic bioassay. Major methods are, first one is the extraction of the extraction. The methodic extract of selected plant species were prepared from powdered material by using soil extraction. The crude extract was kept in a hot water bath to evaporate the solvent. The dried extract were then stored in refrigerated at 4 degrees Celsius. The major steps included in my germination bioass are preparation of stock solution and series dilutions, preparation of germination plates, and final the calculation of germination indices. The bioassay is mainly based on it's a modified method of by Islam and Khatoon Nagochi 2040. Next is cytogenotoxic bioassay. I followed a method by Levan in 1938. Major steps are preparation of treatment bottles, the cytogenotoxic treatment of fern extract, and last the observation of the mitotic smears. The third objective is the GCMS analysis. A 1 ml samples were identified by comparing with the standards. Major results are the experimental were explained on the basis of 7 germination parameters. The mean value of triplets was taken as the final results of the parameters. The major parameters are germination percentage, germination index, germination energy, speed of emergence, coefficient of rate of germination, time for 50% germination, mean germination time. This picture showing the difference between the 3rd day of germination and after 8th day. The major results are an increase in T50 and mean germination time values indicate the germination effect, the inhibitory effect of the extract. The germination rate decreased when applying high dose of extract into the medium. No significant inhibition observed in rice seed germination, but showed standard growth of rice by influence of phytochemicals. Treatment with higher concentration delayed the germination process and prominent suppression of the development of cowpea seedlings. And the second objective is the cytogenotoxicity of the fern extracts on Allium cepa roots. The cytotoxicity of fern extracts were evaluated based on the aberrative indices. Almost similar values were obtained from Terris methanolic extracts and Salvinia methanolic extract treatments. The cytotoxic potential of Terris methanolic extract and Salvinia methanolic extracts increased along with the duration of experimental time. The second day of treatment with the treatment of Salvinia methanolic extract showed better cytotoxic activities. These are the major cytological aberrations observed in my work. Mainly the strap cells with the disturbed metaphase. It's an important one is the Stathmo metaphase. It is the aberrations due to the misorientation of spindle fibers. These also are the identified cytological aberrations. Ball anaphase, Stathma anaphase, coagulated anaphase, early cell plate formation, C metaphase, lollipop metaphase, and chain metaphase, etc. are the least occurring cytological aberrations. Nuclear lesions, chromosome bridges, chromosome fragments, hyperchromatia are the maximum occurring cytological aberrations. In Salvinia methanolic extract, numerous strap cells were noticed. This is the GCMS analysis of these two ferns. In lot of compounds were identified, but these compounds showing both phytochemical and not both phytochemical and cytotoxic properties. Methyl palmitate and methyl oleate are phytochemicals present in both extracts. Phytol also is present. Gamma cytosol is detected in Terris methanolic extract. Terris cordata and Salvinia mosta show similar effects on rice. Phytotoxic and cytotoxic effects of the fern extracts were showed more inhibition on cowpea than rice. And this study explained the potential activity of and the rate of toxic effects of the Terris and Salvinia to confirm the importance of these ferns as bioherbicides. Further studies were wanted to isolate the phytochemicals from the extracts and to confirm the phytotoxic potential of these ferns. These are the references. Thank you.
Any questions from audience? One or two questions.
Yes, sir.
Okay. But where are the anomalies?
So these are anomaly sets.
No, not at all.
First A is the anomaly strap cells with the disturbed metaphase, showing a metaphase.
See,
This is not a
no. This is early anaphase, the F you see, and this clumping. It is allium. It has 16 chromosomes, and the chromatids are moving towards side. So it's likely. If you take still more, then it would be properly separating.
no, sir. Two chromosomes transfers misoriented from the metaphase stage.
I am not objecting your observation. The question is, these are not abnormal ones. These are, I mean, the pictures that you show. Now, if you take the middle of the root, the cell size will be like D, F, and so. But if you take the peripheral side of the root, it would be A and B. That is the elongated cell. And it is always in the middle where the active cell division takes place. Allium, Scribum, Allium Sita, and these are the classical materials which have been used by hundreds and hundreds of papers.
Yes, sir.
And abnormalities have to be totally different. One. And secondly, when you repeat your experiment, do try to revert back by 0.5% glucose and see if they are recovered. In other words, if your observation is recovered, then that is not a damage.
Okay, sir.
But if any kind of aberration persists in the cell, that is damage.
Yes, sir.
Next time can do that. Okay.
Sure, sir.
Thank you very much, sir. Thank you very much. Thank you, Mr. Pradiyam. Thank you very much for your nice presentation. Next, I would like to invite Liju Louis. Mr. Riju Louis is going to discuss about anatomical studies of some selected species of Adiantum from Valparaiso Hills.
Separate phone answer.
These are the files handed over to you.
Mail mail. Licia 300 Licia Louis. അല്ല അയ്യാടെ എനിക്ക് അയച്ചു തന്നിട്ട് ഒരു ഞാൻ നിങ്ങൾക്ക് മെയിൽ ആക്കിട്ടെ Good evening, everyone. I am Lincia. My presentation is about anatomical study of some selective species of addendum for Valpara hills. Introduction. The evolution of land plants is a remarkable and complex history in the biosphere. Pterophytes is the vascular cryptogams. Pterophytes is the vascular cryptogamous with conducting tissues, namely xylem and phloem, are the seedless, spore-producing primitive megaprophytes. Terrestrial vascular plants are growing is mostly along the shades, streams, bank, and river banks, the evergreen forest at medium of height altitude. About addendum. Addendum is the made up of elongated evergreen and deciduous in 200 to 215 species from tropical and subtropical area of North and South America. A few from the temperature regions is Europe, Asia, Australia and New Zealand and North America. They belong in the Pterophytaceae family. The many species is grows in horticulture traits. Objectives. They take the transfer section of the type of select addendum species to understanding and anatomical characteristics of specialized species, to prepare to key based on anatomical variations in vascular bundles, to document of variations in the anatomical for 4 species of addendum. Material and methods. Objectives. To take the transfer sections to the type of the selective addendum species, to understanding the anatomical characteristics of selected species, to prepare key based on anatomical variations of vascular bundles, to document the variation in the anatomical of 4 species of addendum. Material and methods. Collection of plant material. The plant material of addendum is anatomical investigation, where collection in different part of the Valpara hills and Western Ghats is part of taxonomic study of pterophytes. Preparation of specimen. Pieces of young and mature types root of the pterophytes was preserved in Scrv bottle with formalin acetic acid alcohol. For the further studies, the taxa is studies where addendum, radianum, addendum hispilium, addendum cordatum and addendum insium. Result and discussions. Addendum radianum is thick wall and epidermis is cuticularized. Hairs are absent in epidermis. Thin wall and parenchymatous cells and thickened with tannin. Following by compactively packed in endodermis and pericycles. Xylem is V-shaped triarch in nature. Addendum hispidum. Single layer in thick-walled epidermis. Epidermis has unicellular hairs which are arranged distantly. The following by the well closely packed and parenchymatous layers. Tannin deposition was seen in occasionally. The endodermis is following by the pericycles. Scleron is complex, V-shaped and trikes. Addendum cordatums. Single layered thick-walled epidermis, 2 to 3 layers is clear and chymatous cells is compactively arranged well developed in parenchymatous cells with tannin content. Endodermis is following by the pericycles. Vascular standards is process diarch similar shaped xylems. Xylem is surrounded by phloem. Addendum insulium. The epidermis can thin-walled and single-layered. 3 to 4 layers sclerenchymatous cells is compactively arranged parenchymatous cells. Xylem is diarched and V-shaped. And key of the species is xylem V-shaped. Xylem is V-shaped triarch. V-shaped triarch with hairs, adiantum, hispadium. V-shaped triarch without hair, adiantum, radiantum. Xylem V-shaped diarch, adiantum, insilium. Xylem non-V-shaped, xylem diatom, semiluminar shaped, adiantum chordatum. Summary. Variations in the type anatomy of 4 species of adiantums is Walpare-Hills have been studied and documented. Characteristics used including the distribution and sclerenchymatous and parenchymatous cells, shape of vascular bundles, and the xylem is configurations. And in adiantum, radiantum, xylem V-shaped, triarch without hairs in epidermis. Adiantum, hispadium, xylem is V-shaped, triarch with hairs. Adiantum chordatum, xylem diarch and semiluminar shape. Adiantum, insilium, xylem V-shaped diarch. The reference. Thank you. Thank you.
Thank you. Any questions from audience?
See, V shape, so xylem is not triarch. Triarch means when you have 3 arms.
Sir, protoxylem present. Rendu metaxylem center protriarch.
This triarch is always used for xylem and metaxylem position. And this is a V shape, so this is diarch. And when there is a 3rd wing, then it's a diarch. You can correct it later on.
No, sir.
You can correct it later on.
Correct.
You can correct it.
Oh, okay, sir.
V-shape xylem cannot be triarch.
Okay, sir.
Okay, that's all. Thank you.
Okay, sir. Okay, sir. Thanks.
The terminologies that you have used are not very correct. If you look on that hispidulum, or what is the species is there? Yes, hispidulum. And hispidulum, if you track the structure of the vascular strand, it will vary right from base to the apex one, because it has to supply to the 2 lateral branch system of the lamina. So it will vary accordingly. And if you see what you call the V-shaped, if you see the terminal part of the V, it is partly inclined inward, you see. It was infolded, where the protoxylem group is quite seen prominently from here even. These protoxylem elements, they become ultimately the marginal traces for the part of the lamina one. So similarly in other one, they are not as prominent as they are seen over here. So a lot of modifications and a lot of other things are required to be done. Anyway, satisfactory.
Yes, thank you, sir.
Any question? Dr. Das, you want to ask a question?
I have a suggestion only. You can give some magnification level in these slides actually, in that anatomical point. There is no scale and no magnification, nothing. So that will be nice actually. Magnification, slides magnification, what? Scale, there is no scale in that. Bar graph.
Actually, this is not a V-shaped structure as a matter of fact. You know, these are the 2 vascular bundles. They arise from the vascular bundle of the rhizome. They unite later on, but at the very base of the step, if you look at transverse section, you will very clearly you will find that these are two vascular bundles. They unite from this proximal part, and finally they form a V shape of it. In teresvated, even including teresvated or biorhea, it becomes ultimately U shape. And further in other more advanced and more highly complex species, complex I mean in the sense of the complexity in the division of the lamina, then it will be an omega shaped. So these vary accordingly. Hispidum is there, pedatum is there. These are the species which are, as far as their lamina is concerned, that they are highly dissected lamina. So accordingly, the complexity in the vascular stent can be very easily found. So these are instead of a V, yes, you have cut from the middle of the step. If you cut at the very base, you will distinctly you will find two vascular stents, very independent vascular bundles.
You've shown that pteridophytes, all pteridophytes are megaphils. So please correct that one, because there is microphils and megaphils. If you use the terminology pteridophytes, okay, it is the fern megaphils.
Thank you.
Last one, the last slide. There is further one more slide is here, gentlemen.
Last slide.
There's one more slide, yes. On you have written only one recipe. I cannot say where it is published really. Indian Journal of Biological Sciences. There's a very good paper on anatomy, on the step anatomy of Adiantum species, some 15 or 16 species. It is investigated by Walker and Biden. The paper appeared in the Singapore Botanical Garden, Journal Singapore Bulletin Singapore Botanical Garden. Beautiful paper. 15 or 16 species, they have shown it. Like, nearly similar, they have not given any phylogenetic approach or any evolutionary approach. Similar one, right? Studies are based merely on a single cross section of the stack. That is one. You also look on to that one. If you don't have, you can ask me. I can give you.
Thank you very much.
Thank you.
So finally, I would like to conclude that session, Future Scientist Award session. So as I know that totally we are having around 7 participants for this Future Scientist Award. So according to us, as all they have given that very good presentation, because this is a Future Scientist Award, really is very good, and we are not able to find out any difference of presentation. Even though we are having some criteria, based on the criteria, we have given the mark, and I don't think there won't be any difference in that Future Scientist Award. So finally, I would like to thank the organizers and participants for thank you very much.
Thank you, judges. Now, may I invite Dr. N. Punita to give a memento to Dr. Raju Anthony. Now let us break for the tea. Kindly, we will be back at the conference hall after 30 minutes. Okay, so be back by 6 o'clock.
Department of Botany, University of Calicut, was founded in 1968 and has a long and distinguished history created by eminent faculty and researchers. It is one of the leading botany departments in India, offering MSc in Applied Plant Science and MPhil and PhD programs in botany. It has been funded by the Department of Science and Technology, Fund for Improvement of Science and Technology Infrastructure, and Kerala State Council for Science, Technology and Environment, Society for All-Round Development Programs. The university has recognized this department as a center with the potential for excellence during the Golden Jubilee year 2018. Currently, the department encompasses 8 divisions: Angiosperm Taxonomy, Cell and Molecular Biology, Cyanobacterial Diversity, Environmental Science, Fungal Diversity, Genetics and Plant Breeding, Plant Biotechnology, and Plant Physiology and Biochemistry. Many faculty members have been awarded with prestigious fellowships including Nuffield Foundation Fellowship, Smithsonian Fellowship, Alexander von Humboldt Fellowship, Janida Fellowship, and Nehru Centenary Fellowship. Several distinguished teachers have served the department in the past and contributed immensely to its growth and development. So far, more than 230 PhDs were completed from this division. The tissue culture methods developed in this department are now being widely used in the cultivation of banana, vanilla, and a variety spices. 2 genera and more than 200 species of flowering plants, 4 genera and 224 species of fungi are discovered as new to science, and 2 patent applications have been filed from the department. Our rich tradition of classical taxonomic research has brought laurels to this department in the form of coveted awards: Padma Shri, Dr. Janaki Ammal Award, Professor Puri Award, Chair of Taxonomy, Y.D. Tyagi Gold Medal, Professor P. Maheshwari Award, Dr. Vini Shivarajan Gold Medal, Excellent Scholar Award, and Jodef Stussy Gold Medal. Professor Joski Mangali, Professor M. Sabu, and Professor Santosh Thambi have been recognized by the Linnean Society of London by awarding them with its honorary fellowship. The scholars from this division have bagged 9 awards issued by International Association of Plant Taxonomy. The department is the headquarters of Indian Association for Angiosperm Taxonomy and Rigor-Mendel Foundation. The department has an internationally recognized herbarium with over 1 lakh specimens of vascular plants from India. The department is proud to have an excellent botanic garden with an area of 33.5 acres with diverse collections of zingibers, gesneriads, orchids, aquatic plants, ferns, bromeliads, spiderworts, cacti, medicinal and aromatic plants. A touch and field garden for the visually challenged people is another major attraction. Redia and Echo Chronicle are the 2 journals published from the department. The state government funded Inter-University Center for Plant Biotechnology is also associated with the department. The department has undertaken research projects in all major areas of plant science, for which it has received generous funding from the Department of Science, Science and Engineering Research Board, Ministry of Environment, Forest and Climate Change, Department of Biotechnology, the University Grants Commission, Council of Scientific and Industrial Research, National Medicinal Plants Board, and Kerala State Council for Science, Technology and Environment. The granted facilities from various funding agencies are also used for interdepartmental research. For the last 5 years, the total national and international research funding for the department was around 3 crores and 40 lakhs. More than 60 books and over 2,000 research papers in reputed journals with high impact factors were published from this department. Many faculty members have active collaborations with institutes in and outside India. Calicut University Botanical Garden, established in 1971, has developed into an excellent center for biodiversity and ex situ conservation of tropical Indian flora and exotic species. The garden, the brainchild of Professor B.K. Nair, the first head of the Department of Botany, was inaugurated in 1972 by Professor R.E. Holtom, the then director, Singapore Botanical Garden. In 1985, the Calicut University Botanical Garden achieved international status when it was recognized by the International Consortium of Botanic Garden and Center for World Conservation Strategy. Ex situ conservation of the rare, endangered and threatened plants of South India is the major thrust area of the garden. A separate section in the northern boundary of the garden is specially designed for the visually impaired. This is an initiative funded by Ministry of Environment and Forest and Climate Change. With the provision to familiarize plants by touch and feel, this section started attracting the visually impaired. About 65 species of aromatic and other curious plants and 34 seeds and fruits of diverse groups of vegetables, spices and medicinal plants are displayed here. The audio system and braille scripts provide basic information including uses of all the plants exhibited in this garden. This is one of the largest of its kind in India. The Calicut University Botanical Garden covers an area of 20 acres with a shallow basin encompassed by the sloping terrains except for a narrow gap on the southern side where it slides down to a small transitory reservoir, providing diverse habitats and niches for a variety of plants. The central shallow region is provided with a graceful placid pool and an octagonal greenhouse and avenues of royal palms and oil palms. A good collection of wild gingers are maintained in the garden in Ginger Villa, Ginger House, Spices House and in the Ginger Zone.
As short and quickly as you can.
Okay.
Good evening, one and all. Good evening. Good evening, sir. Good evening, Dr. Joel H. Nita.
Can you hear him?
Can you hear me?
Yes,
Yeah.
thanks.
Okay.
So I am very happy to introduce Dr. Joel H. Nita to this august audience. So he is his PhD in organic and evolutionary biology from Harvard University, and his dissertation is on ecology and evolution of the ferns of Muria and Tahiti in French Polynesia. And he is very interested in field biology and also in lab techniques, that is DNA extraction, PCR, Sanger DNA sequencing, etc. And he has more than 15 years of experience, research experience and teaching experience. And now he is in Department of Biological Science, Graduate School of Science, University of Tokyo as Project Research Associate. And he has several grants and awards received, like Japan Society for Promotion of Science, Kakenhi, Smithsonian Institution Barcode Initiative, Japan Society for the Promotion of Science, Kakenhi, like that, more than 10 research grants. And he has received several honors like the Young Scientist Award of Japanese Society for Plant Systematics, Best Oral Presentations, Japanese Government Scholarship, Phi Beta Kappa Society Scholarship, etc. And he has more than 25 research publications in modern technology, and he has several papers presented in national and international forums. And I greatly welcome Dr. Joel H. Nita to this august audience. Thank you. Thank you.
Thank you, Nat. Now I request H.K. Goswami chairing the session, and I'm very happy. Kindly proceed with your presentation. I request you to do that. Yes. Go ahead.
Okay, to start the screen share. Okay. Just a moment.
Yes.
Okay. Yeah, it's ready. So shall I go ahead and start my presentation then?
Yes, please
Hello?
go ahead.
Okay, yes, sorry about that. Thank you very much for the kind introduction, and thank you very much for the opportunity to present. I'm quite excited to be able to present today. So the title of my talk is DNA Barcoding of Fern Gametophytes, Past, Present, and Future. So a little bit about this talk. It's actually based on a paper that just got accepted to applications in plant sciences, and it's in press right now. So there's a preprint available at this URL, and all these slides are available online as well if you're interested at this URL, dolnita.github.io/gametobarcodeifs. And of course, you can find more information about myself at my webpage. Yeah, the paper will be out soon, so you can find out more information shortly. So the goal of this talk is to provide a practical overview of DNA barcoding in fern gametophytes, and hopefully present some useful information for you all. So first question, what is DNA barcoding? So DNA barcoding can be defined as the use of typically one DNA locus or gene for identifying species. And the term was first coined by Paul Hebert in 2003. He used it originally for studying butterflies, and since then the concept has really taken off and it's been used in many different taxonomic groups. This is just one typical example here in irises. But I would say that we need to be careful because the term barcode is a bit of a misnomer. And that's because when you think of a barcode, if you're in a store, right, a barcode is an identifier for a product, and it's exactly the same on every item of that kind of product. And it's always different between different products. But of course, no gene exists in nature that is like that. There's always going to be some amount of variation. So we should keep that in mind. It's a nice term, but it's not quite accurate. And so in the animal community, where it was first developed, they quickly decided on using the CO1 gene. It works quite well in most cases. However, it's not been as easy in plants. There's not a single marker that works well across all plants. So that's the basic answer to what is DNA barcoding. Well, why should we care about doing DNA barcoding in ferns? I would say there's two reasons. And the first of all is for taxonomy. That's because DNA sequences can provide objective evidence of species status. For example, if we look at the mean difference in the RBCL sequence between species within a genus, overall across all ferns, I've calculated this, and it's about 1.5%, plus or minus 0.78%. So it's just this is a very, very rough number, but if you sequence RBCL and you compare it with the most closely related thing, and that gives you a benchmark for if this may be a distinct species or not. That's not sufficient by itself, but it is one objective piece of information that we can use. So I would say that DNA sequences and phylogenetic analysis can really help make our species descriptions more robust and more convincing. I happen to be a subject editor at some journals, and we receive a lot of species descriptions. And of course, I'm not saying that you have to have molecular data, but if you do and you show a convincing phylogenetic study, it can really help make a species description more convincing. I'm not saying that you have to have it. I'm not saying that that alone is sufficient, but I'm saying that it can help. So that's one reason, taxonomy. But that's not the main focus of my talk today. The main focus of my talk today is using DNA barcoding to identify field-collected gametophytes. So of course we all know about the fern life cycle, alternates between sporophyte and gametophyte, but almost everything that we know about fern ecology only comes from the sporophytes, and very little is known about what the gametophytes are doing out in nature. And of course that's because fern gametophytes are morphologically cryptic, and they're really, really small. So they're hard to find in the first place, and once you do find it, we don't know what species it is based on morphology alone. Now, that's not to say that no one studied gametophytes at all. Quite the opposite. For example, the very well-known and eminent Indian turritologist, B.K. Nair, wrote a fantastic treatise on gametophytes of Homosperous ferns. I use this paper all the time. And in this paper, he says, in combination, these aspects of prothalliar morphology serve to characterize most of the larger groups of Homosperous ferns, nearly as clearly as sporophyte morphology. So he did a wonderful job of describing all of these trends and differences in morphology amongst higher groups of ferns. But the thing is, those are usually not sufficient to actually distinguish species from each other. So we need some other tools for that. And this is just an example of what I mean. So in each row, we see some of the major types of fern gametophytes that were described by Professor Nair. A and B is cordate, C and D are ribbon, and then E and F are filamentous. So these can give us some hints to the overall groups or maybe families. However, we don't know what species we have. And so I actually identified all of these gametophytes using DNA sequences, and this is sort of like a quiz and an answer. These are the answers. These are the actual specific identifications. And I mean, if you were to look at those in the field, you might think A and B, I mean, they look practically identical, but that's a tree fern and a member of Blacknaceae, so they're quite distinct, right? So those are just some examples. Now to get into a bit of the history of DNA barcoding. So as applied to fern gametophytes. The first study was actually by earlier speaker today, Harold Schneider and Eric Schupeltz. They sequenced a gametophyte that had been growing in culture, I believe at a botanic garden, as belonging to Asmunda, and by sequencing the RBCL gene. So and it was just a single individual, single gene. So that was the first documented use. And then since then, the field has grown quite a bit, and the modern, or recent, I would say, way that it's done, I've outlined here. So it typically involves two phases, and during the first phase, we assemble a reference database based off of well-known or well-identified sporophytes from a study area. So I was doing my study in French Polynesia, so that's where I tried to sample sporophytes, right? And then the second phase is you would collect gametophytes in the field, bring them back to the lab, extract their DNA, do the sequencing, and now you have this reference database so you can match it up. And that's typically done with a search algorithm called BLAST. And it'll tell you how similar those sequences are. So we can say if it matches over some given threshold, then we will identify it as that species. So it's fairly straightforward. And it's almost always, until now, been done with Sanger sequence. However, one variation on this is that you don't have to assemble that reference library. You could just blast it against an online database like GenBank or Bolt. So that's an alternative method. Or you might end up doing both. Now, when we're during that first phase, when you're putting together the reference library, you need to choose a standard marker to use. And I've already mentioned a couple times RBCL. RBCL is one of the most frequently sequenced genes in ferns and in plants generally. However, it has a fairly low rate of variation between species, and that's because it's a coding gene. It makes a protein. So it's subject to a high degree of natural selection. If it mutates too much, it's not going to survive, right? So it's oftentimes, certainly for angiosperms, it's not variable enough to be used by itself as a marker to distinguish species. So many of the plant barcode papers have advocated for using RBCL and another marker. And I would just cut to the chase here and say, give you my recommendation for ferns. I would recommend RBCL and the TERN-L, TERN-F intergenic region. So that's a spacer region, which is not subject to as much natural selection. It has more variation. And this is just an overview. In this table, we see 4 different genes or intergenic regions that have been tested and some of their qualities. And again, I would recommend RBCL and TERN-LF. Another option, however, these are all chloroplast genes. And as I'm sure most of you know, the chloroplast in ferns is maternally inherited. It only comes from the mother's side. So if we think we have a hybrid, this is not going to be very it can't tell us both parents of the hybrid because we're only using a plastid marker. However, nuclear markers are much more difficult to obtain. They're not as easy to, PCR is generally more difficult. And then since you have multiple copies, they are more difficult to analyze as well. So most barcoding studies to date have only used chloroplast genes and not worried too much about the hybrids, which tend to be fairly small in number anyways. Okay, so now I'm going to move on to show you some example studies that have assembled this DNA library. So as I mentioned earlier, this building the library or the reference database step consists of Sanger sequencing of at least one specimen per species in your given study area. I said at least one specimen per species. Well, a question arises, do we need to have multiple individuals per species? And one reason you might need to do this is to assess what's called the barcode gap. So this is an illustration of what that looks like. So in red would be the distances within species, so the variation within a species, and yellow is the variation between species. And ideally, those should be different. If they're overlapping, that's a problem, because then it becomes very difficult to tell if you have from these distances alone, if you have a member of the same species or different species. But from a practical standpoint, with RBCL anyways, this red distance within a species is almost 0 or very close to it. So there's not too much point in sampling multiple individuals per species for RBCL, because it's going to make it cost a lot more. You have to do everything 2 or 3 times, factor of 2 or 3 times more than you would otherwise. So what I have done in the past is just do 1 individual per species, except for taxonomically challenging groups or species complexes where I'm not sure if I'm not confident in their identification, then I might sample multiple individuals per species. So yeah, that would be my recommendation there. Okay, so this is one example of some of my previous research. So in the pteridophytes of Japan. And in this study, we used 2 regions, RBCL and then the TERN-HPSBA region. And actually, I would not recommend the TERN-HPSBA region because it shows really low rates of variation in many lineages of ferns. But we didn't know that at the time, so we did it. And the pteridophytes of Japan are actually quite diverse, over 700 species. So, I mean, not as many species as India, obviously, but it's a much, much smaller country, right? So I think it's, so just as a side note, Japan is a great place to study ferns because they have a high diversity for a small area. What we found here was much higher success in distinguishing species with these markers in sexual diploids, but that it was much more difficult in polyploids or epigamous taxa, as you might expect, because oftentimes the epigamous species, they might have identical RBCL or chloroplast sequences because many of them are hybrids. Okay, and another case study was research that I did in French Polynesia for my PhD project. And again, I used the same two markers. And again, I would recommend instead use Ternal F instead of Tern HBsPA. This had way fewer species, of course, because we're now in these remote, small oceanic islands. And actually observed a higher success rate. So when I say success rate, I mean the ability to distinguish between species with these markers. And I think that actually has something to do with the biogeographic history of this area, because most likely many of the species that occur in these remote oceanic islands mostly evolved elsewhere and then arrived there, they colonized it later, as opposed to continental areas, or Japan is right next to the continent, where you probably have higher rates of in situ speciation and therefore much, much less divergence between species. So barcoding isn't just for identification. It can also tell us some interesting things about the overall biogeography. Okay, now I want to move on to some examples of what we can actually do with these DNA barcodes. And one really important area of study is finding independent gametophytes. So what is an independent gametophyte? Well, because of the unique fern life cycle, we know that sporophytes and gametophytes can actually grow. They are each nutritionally independent. They can grow separate from each other. And this can actually reach some really extreme degrees where the gametophytes go completely outside of, there could be gametophyte populations that occur completely separate from sporophyte producing populations. That's what I'm showing in this diagram here. And we call those independent gametophytes. And these were originally described from North America because they belonged to mostly tropical groups that had some really unique morphologies. And so it was able to, we were able to, well, not we, that wasn't me. That was other researchers before my time, were able to identify them without DNA. But now that we can use DNA barcodes, we can actually find many more independent gametophyte populations in places where people hadn't looked previously. And one example of that is Japan. These are some recent results from Professor Noriaki Murakami. And he's been really focusing on what are called gametophyte mats. And these are these clonal colonies that form, that some ferns are able to form because, as you can see in this bottom image, at the tip of the gametophyte, it has these little dots, are these asexual tubercles called gemma. And those will fall off and start growing and create a clone, and you get this clonal mat like this. And those can be long-lived and also get to pretty big sizes that are fairly easy to find in the field, as opposed to finding one tiny chordate gametophyte. And so they've really been focusing on looking for these mats. And by doing that, they've been able to find gametophytes that are occurring completely separately from their sporophytes. And in many cases, the sporophytes are either really rare or endangered in Japan, or even completely unknown in Japan. They don't occur in the country at all. So those are some neat recent study results from Japan there. And that's an image of, you can see what the gametophyte colony looks like there in this Haplopterus mediasora. Another example is not just looking at single occurrences of species, but looking at communities of gametophytes growing together. This is a study that I was part of in Japan, where we surveyed gametophyte communities and developed sort of a new method for doing that, and that's using this garden net. So this garden net is 50 by 50 centimeters, and each of these squares is 10 by 10 centimeters. And what we do is we'll find a spot that we can see probably has some gametophytes. And the tricky thing about these gametophytes is when you're sampling in the field, you don't know what species you have. So how do you know how many to sample? It's not like you can go to a spot where sporophytes grow and say, okay, I just sampled a Criponominus minutum and a Hymenophyllum polyanthus. Okay, I'm done here. We don't know that for the gametophytes. We don't know what species they have. So in order to maintain the same sampling effort consistently across sites, we developed this method where we set down this garden net, and for each of those squares, if there is a gametophyte there, we'll select one gametophyte that's closest to the center. And then that's our plot. And we'll repeat that at different sites across the country. And so these are some of the results.
You have 5 minutes to compile. 5 minutes. You are requested to compile within 5 minutes so that we have some questions also.
Okay, sorry, I'll try to speed up a little bit. Okay, so these are some of the results showing the identities of the gametophytes there. And I also did this in Tahiti, in Moorea, and one thing that I did to improve the technique was to do things in 96-well plates. So now I was able to analyze, sequence more gametophytes at once. And one thing that was really a neat result of this is that we can see that the community structure differs between sporophytes and gametophytes. So here, the sporophytes are shown in blue, and the gametophytes are shown in red. And of course, whenever most ecological studies would only have the sporophytes. You wouldn't even see these red dots. But what we see here is that the sporophyte communities become less diverse at high elevation, and the gametophyte communities are fairly diverse across all elevations. And that's because some of those gametophytes are more widespread, especially in families like filmy ferns. Okay, another study is looking at hemiepiphytic growth. So hemiepiphytes spend part of their life on the ground and part on the tree, but it's not known necessarily whether they start in the tree or start on the ground. And so for this, I sampled gametophytes at the base of this. I found gametophytes at the base of the tree, and I used DNA barcodes to confirm that they were the same as the sporophyte. And then we know that the gametophytes start on the base of the tree and grow up. Okay, and then finally, this is a study of desiccation tolerance in filmy ferns. So desiccation tolerance could be really important in the transition when plants move from water to land. And filmy ferns are known to have desiccation tolerance in both the sporophyte and gametophyte phase, but they hadn't been compared between them. So I did a comparison, and this is showing with gametophytes identified by DNA barcoding. And surprisingly, the gametophyte stage was actually less tolerant of desiccation than the sporophyte stage. And I think what's probably going on is that instead of relying on desiccation tolerance to achieve a wide distribution, these gametophytes are actually surviving, again, via these little dispersal units called, asexual units called GEMMA, and forming these gametophyte mats that can be long-lived in protected microhabitats where they are not actually subject to drought. So those are just 5 studies that I've participated in that I can think really show the benefit of DNA barcoding. And the last set of slides I have is to show where I think the field is moving towards, and I'll try to wrap this up quickly. So basically the short version of that is that until now, all of the DNA sequencing has been done using Sanger sequencing, which tends to be kind of slow, limited number of samples, limited number of loci. But as you know, there's next generation DNA sequencing technologies are now available that could enable to do this in a high throughput way. And then maybe we could have continuous monitoring over time and space of gametophyte populations. For example, microerythrophylum PCR can really scale up the samples from PCR. Downside is that it's expensive. Another really cool upcoming technology is called the MinION DNA sequencer. And this is actually designed, it's really, really small. It's like the size of like a USB dongle. And you can take it into the field. And so this is a paper where it's not for ferns, plants, they did it on reptiles, but they took it into a rainforest site and did on-site next generation DNA sequencing and barcoding. So this could help direct other studies that if you're not able to go back to a lab and you need to stay in the field, it could be really helpful for that. And then finally, another method that's recently been developed is called tissue-direct PCR. And that is running the PCR directly on the gametophyte itself without doing any DNA extraction. So it could maybe simplify the lab work and make that go faster. And that's recently been used in Taiwan to identify independent gametophytes of Lomereopsis. And so I'll wrap it up here. So in conclusion, I think DNA barcoding can provide some unprecedented insights into fern biology. Sanger sequencing is still useful, but limited. And next generation DNA barcoding really has the potential to revolutionize fern biology again. And with that, I'd like to thank my co-author on this paper, Sally Chambers, and Botanical Garden that helped support us, Liam Ko, who shared some unpublished manuscripts, and then you all for listening to my talk and hosting me during this wonderful conference. Thank you very much. And then I have some references at the end if you want to see those.
I hope you accept a few questions.
Yes, please.
Yes. His talk is open, and if anyone is interested. In the meantime, I just want to ask, have you come across highly abnormal gametophytes in nature? I mean,
it depends what you mean by highly abnormal.
The question is, I had reported, published in 2006 in Fern Gazette, that the wild gametophyte have sex organs on the margins, Anthridia and Archegonia on the margins of a gametophyte, along with the presence of tracheids.
And the presence of what?
Tracheids, you know, xylem, which is...
Tracheids. Yeah, tracheids.
Vascular tissue is not supposed to be present in a gametophyte.
Right, yeah.
So, yes, so that was published in 2006, but I wonder if you, since you work on gametophytes, especially also from collection from wild areas, have you ever come across highly abnormal gametophyte?
Not like that, no. No tracheids.
Okay.
Yeah. I know if you cultivate gametophytes in the lab for long periods of time, oftentimes you'll see some pretty weird morphologies, but I think those are sort of aberrations, not something you would see in nature.
Maybe. Yes. Any question? Or now, do you think the gametophyte, these Gemma cups, they represent the gametophytic stage?
Yes. There's Gemma that occur in sporophytes as well. There's asexual reproduction of sporophytes as well, but it's a different thing. In sporophytes, of course, it's diploid, and gametophytes, the asexual reproduction is staying in the haploid stage. So
see, the classical botany states that gametophyte is one which has the sex organ present and rhizoids to support the nutritional effect. Gametophyte is a structure born on the plant which, when detached, gives rise to another thallus. So that cannot be compared with the prothallus. Haploid is all right. It is a gametophytic stage for that purpose, but it is not a gametophyte like prothallus which you see in other ferns or in the other bryophytes.
I'm not quite following. Sorry.
See, Gemma cup is a part of the thallus.
Oh yeah, I didn't say Gemma cup. They're just called Gemma. Yeah.
Okay. In our Markenshales we find, in coccineals we find Gemma cups, or Gemma.
Mm.
Okay.
Yeah, so the Gemma are there. If you look at the tissue, it looks almost exactly the same as the rest of the gametophyte. It's just some cells that sort of bud off to the side and then fall off.
Yeah, they fall off.
They're very simple.
Okay, that's all right. So anyway, thank you very much for a wonderful presentation. You have a question? Okay, one more question.
Yes, please.
Good evening, sir. My name is Leslie Augustin. The question is that when it comes to this NGS and barcoding, okay, this RBCL locus you are suggesting. So could you please just simplify what is the workflow of the NGS when you are using the RBCL? Could you please explain that as is for the student's perspective?
The workflow?
Just for the RBCL DNA barcoding workflow, not for the entire sequencing workflow. If suppose you are choosing any platform, is it okay? It's okay. Illumina, Solexa, or whatever it is, okay. I just want to know what is the workflow for the barcoding when it comes to the NGS.
Oh, for the NGS part.
Yeah.
Yeah, so That I think there was a similar question with Harold Schneider's talk, and I would have a similar answer that it really depends on what you're trying to do. And it's still being worked out, actually. There haven't been any studies yet using NGS for barcoding of ferns or fern gametophytes. So I can't actually say what the workflow is because no one's done it yet. We're still trying to figure that out. But I think one of the main things that you have to keep in mind is that depending on the platform, the NGS platform, they have some different constraints. For example, with how long a read they can produce. So Illumina platforms, the longest reads are 300 base pairs in one direction. Yes. So if you put that together, it could be up to 600 base pairs. But RBCL, full-length RBCL is like 1,200 or 1,300 base pairs, so you can't cover the whole thing.
Whether the Minion can do that?
Minion has longer reads,
yes.
But it also has a higher error rate.
Yeah.
So it's not clear yet how that is going to balance out for specifically with regards to barcoding of ferns. Yeah.
Thank you so much.
But those are great questions. Thank you. And I would say one other platform is PacBio, which also has long reads, but also has a higher error rate. So again, it's
because of the artificial polymers. Yes, error is there. Thank you so much. Thank you so much.
Anyone else? Okay, so you want to ask? Okay, thank you very much. Thank you very much.
Okay, thank you.
ये है, स्मिता को बुलाना है, इलियास के लिए, इलियास अमीदा, मेरे ख्याल से वही होगा, बुढ़ा हो गया होगा, बहुत ये, इलियास, अच्छा ये है।
Okay, it says it's trying to record my computer, but I need to do something.
And additionally, actually, she's now addressing the critical knowledge gaps in the Amazonian flora, focusing in inadequately sampled areas and trying to find the integrative approaches there and trying to strengthen the botanical research in the Amazon region. So, and she has some good publications in several reputed journals. So with these few words, I welcome you, madam, to
Yes,
start your
Professor.
speech.
Yes, Professor. Please continue with your presentation.
Well, good evening to everyone. Thank you so much for the invitation. I'm very happy to be here speaking to you today. Good morning from the Amazon. I'm a little bit in a different time from you. So what I'll present to you today, it's a part of my research, what my research group has been doing, focusing mainly in the using of integrative approaches in systematic and evolutionary studies. We're focusing on neotropical ferns and lycophytes, but those tools can be used to all different groups. What we're mainly concerned about is speciation, actually. We're thinking about species and we're concerned about the processes giving rise to those species. And that's because ferns and lycophytes, they are very peculiar because they have in the life cycle two independent forms. We have the sporophyte and the gametophyte. They have different sets of chromosomes. They have different forms, physiologies, and Joe Nita talked a lot about the gametophytes in his great talk. And we do have those parts of the life cycles independent. So we have selective pressures occurring in both. And we don't know if they're the same, but they're probably different. So that's peculiar among plants because that doesn't occur with bryophytes or seed plants. Also, we have the spores, which are very interesting because they make barriers, like geographical barriers as rivers, mountains, or oceans in existent ferns because they are wind-borne and they can be spread everywhere. So when you think about evolution of ferns and lycophytes, they are very, there's these peculiar points we have to consider when we are thinking about the evolution of these plants. And the other thing we have to consider are the selective pressures that will eventually cause speciation in those plants. We know some abiotic factors that are correlated with fern occurrence. For example, water availability, temperature, the soils, which are very important. We have that documented for the Amazon. And we do know mountains are sites related with higher diversity and diversification rates, but we don't know how those abiotic factors actually act as selective pressures to cause speciation in ferns. And we also have to think that those factors will act differently in the sporophytes and differently in the gametophytes. And we are not even considering the biotic factors, for example, competition, or etc. But we do know a little bit about speciation in ferns and lycophytes, and we know they are occurring mainly in allopatry because we have separate population, and that's mainly because we have a lot of dispersal occurring. But we do have a lot of sympatric speciation, which means that the populations co-occur, but they are somehow exploring different parts of the niche. So we have a kind of ecological speciation. And we do know a little bit that those two pathways of speciation are occurring differently in temperate and tropical zones. We know more about the temperate, what's happening in the temperate zones than in the tropics. Also, we do have a lot of hybridization and allopolyploidy occurring in ferns. We have several examples of speciation occurring given to hybridization and then allopolyploidy. So it's a very complex story, and we have to have that in mind when we think about the species. Also, one thing that we have to consider to define species is that the speciation is not a short time event occurring in a short time event. Speciation is a long process in an evolutionary time, given in generation times. And depending of when you're looking at that long process, you have different things. You see different things. It's a continuum. So if you look, for example, I'm not sure if you can see my point? Let's see if I can, okay. If you look at, for example, in a given moment when speciation still starting to occur, maybe it's easy to say, okay, all those populations are one lineage, one species. If you look at the other end, where the split has occurred, a lot of divergence has accumulated, you can see, oh, okay, I have two, I have three, I have several species. But mostly what we are looking at is the in-between, the gray are the species. And whenever the analysis predicted in a positive way that Microgramma baldwini was Microgramma baldwini, we had a positive prediction, we had that scored here in that cell. And you can see that the diagonal here cells are the positive predictions. Every time the analysis said, yes, based on that spectra, that would be that species. And that was correct. You have that counted here. Everything outside the diagonal are the negative predictions, the errors. So you can see, and this is our worst result. And here in this, to the right column, you can see the percentage of it, the correct predictions. So our worst result was 87% of correct predictions, which is quite good because we're just using the spectrum in the near infrared. But the errors are quite interesting too, because this is a way of identifying species. If you have, for example, a sterile specimen you can't identify, you could use that to identify. But that can also tell us a lot about the species circumscription. For example, the one with the worst prediction power here was Microgramma vaccinifolia, which is a quite difficult species, we know. And then Microgramma squamulosa, which is known for having a lot of hybrids. So we went back to the specimens, we were checking, yeah, that one actually could be a hybrid, that one, I'm not sure. So we went back to a species concept based on that result to actually see if we had problems with the species circumscription, and mostly we had problems. So it's an excellent tool for identification, but also it can be used to explore the species concepts and also identifying hybrids. The other tool we're using is morphometrics, mainly geometric morphometrics, but I have a student, a PhD student now trying the elliptic Fourier analysis, which uses the outline, as shown here from Vichi's study. And it's a very accessible tool because you can use online images to gather the data. So we just go, here we have an example from Microgramma. You use from available herbarium that have available images. You can gather the images and you just put that in R or image J, and then you can mark the landmarks and collect the data. So you don't have to have expensive field work or expensive tools to use that. Morphometrics, it's quite good, it's quite accessible. So we went back to that problem of Microgramma vaccinifolia, the one that performed not well in the near predictions. And it's a quite complex, it's a complex actually. We have these two species that are sisters, and so we have vaccinifolia, that one here, that's sister to Crispata, And you can see in the map here, their distribution overlaps, so they occur in symmetry. And they also have a morphology that's overlapping. We have here showing the proportion between fertile in red and sterile front in green. And we can see in Microgramma vaccinifolia that the fertile front can have a length like 4 times the length of the sterile front, up to fronts with the same length. And we have Microgramma crispata with only that variation in its morphology. And vaccinifolia has all this variation. And it's just not the length, the proportion. It's the shape of fronts too. So we have a lot of variation. So the question was, maybe that thing we're calling Microgramma crispata is just an extreme of the whole variation of Microgramma vaccinifolia. And because it's an extreme in a line of variation, we are just separating that and describing as a different species. So we use the morphometrics to try to see if that was the case, or if no, if it was really a good species. And the results were, well, they kind of went with what we first thought. That you can see here, it's also part of my student master thesis. We can see here in blue and pink that crispata, which is in pink, and blue, it's vaccinifolia. You can see that their distribution in the morphospace, it's quite overlapping. It's pretty much completely overlapping. So, and that happens too when we look at the near infrared results. We can see here that crispata, we have vaccinifolia that have variation outside the variation found in crispata, but they're mostly overlapping. So that indicates that Microgramma crispata is probably not a good species. We're not making any decision, taxonomic decisions now. We need more data. We are going for genetic population information to try to understand what's going on. Maybe it's the, you know, in that speciation continuum, maybe it's the starting of the process of speciation. But we have to think of how to translate that evolutionary information in a taxonomic information. And the third thing I would like to present here is the third tool we're using is the in species distribution modeling, and not just the models, but also the environmental information we can extract from the same data sets we use to build the models. So we have the ability to predict from the known distribution of a species, the known occurrence of a species, and a set of different kinds of climatic soils and terrain, topographic data sets. We can describe and we can predict what is the ecological, what we would, some people don't like calling niche, let's call it niche. What would be the niche that species is occupying? It's not really the niche because we don't have the biotic factors, which would be the abiotic niche, for example, climatic niche, if you prefer. So we can have that, and we can use that to understand both the evolution or speciation process occurring, and even to test the deletation of a species. So what we're doing now with 2 different groups, I'm showing here what we're doing with Microgramma, but we're also working with Amauropelta, genus Intellicturaceae. We are trying to compare the niche in a phylogenetic context. So this is the phylogeny of Microgramma I published last year. So we are working in clades to compare if closely related species, if they occupy distinct or similar environmental spaces or niche, if they have the same niche or if the niche is divergent. Because if the niches are close and they come from the same ancestor, that means that they're probably competing for the resources in the niche. So we are expecting that closely related species will occupy distinct niches, meaning that they are speciating by ecological speciation. So that's something that's ongoing in our studies here. And another research line that we're starting now is to think a little bit broader, using that too, and the other ones I showed before, using the NIR, the spectral data, the morphometric data to characterize what we would call the morphological space that one species is occupying, and using the environmental data to define the environmental space, not the geographic space, but in a n-dimensional space. And what we want to understand now, if there is a correlation between those two, if a given species occupying a niche has a given morphology, and that's reflected in their morphology, in a phylogenetic context. And we also want to understand if closely related species will have divergent phenotypes, and that would be mainly given to, again, ecological speciation, because if they are occupying the same environmental space, if they have very close related morphology, they are competing. So if they have divergent phenotypes, if it's not similar, if it's very different, it's because they are trying to avoid that competition. And then we could have speciation due to these competitions, so ecological speciation. Or if they have similar phenotype, or similar occupation in the environmental space, that could be, I don't know, maybe given to stabilizing selection, maybe it's just morphological stasis, maybe it's an evolutionary constraint. So this is what we're trying to understand now, not just to define species, but trying to understand a little bit about the process behind what's going on in the speciation in these models we're using. And what I would like to, I hope you followed me, because it's a very short time to explain everything. But the message I would like to leave here today is that looks can be deceiving, meaning that using just macromorphology to define species can lead us to just building artificial species, especially in ferns and lycophytes, that we have so few characteristics, macromorphological characteristics to use. And we don't have flowers, we don't have reproductive flower phenology, pollinators, etc. And we're just using mainly the sporophytes. We're not even using the gametophytes to define species in the traditional macromorphological approaches. So I'll just join my voice with Jonita. You don't need genetic data, but if you have other sources of information to help circumscribe and define a species that correspond to a lineage, that it will be very, very helpful.
Excuse
me,
you have 5 minutes more, and then we want to have questions.
Okay, I just finished.
Yes.
I would just like to thank you for the attention and show here all my students. Without them, I wouldn't be able to be presenting these results here. And I leave my email here. If you have questions, if you want any literature, you can write me and I'll be happy to share.
Okay.
And again, thank you very much for the invitation.
Her talk is open for questions, any kind of comments or... Yeah. You should have.
Okay. So, madam, can you explain or can you elaborate that how you are using that NIR data to discriminate the species? How the way you are using?
Okay. So what we use the NIR data we use to test a species concept, because I tell the analysis what each species are. So I'm telling these 5 the data corresponding to these 5 specimens are Microgramma baldwini, for example. So what we do, we test our premises, the names we already put in the model. So what we did in this analysis, we wanted to know if data from the different sizes of the frond would be different. And we wanted to know if both would be okay, because you can read from one side and then the other, or you can use both. Because what we use in the analysis, the whole spectrum. So every peak, every wavelength peak is a data, is a information. We wanted to know if more information would be more helpful. So what we do, what this confusion matrix they do, they use the set of spectra for a given species, and they randomly pick one species, one specimen, and try to compare that spectra from that specimen with what we told the model is the spectra of the species. We read several different specimens, and then we sum that and say, this is the spectra of this species. And then we just pick up a specimen and we try to assign with that data if that fits that spectra. Is that clear?
Can you assign the data with a specific type of macromolecules?
No, we don't know what they are. I've read in the literature that it's mainly carbohydrates, maybe celluloses and some proteins. Every different part of the wavelength, it will be the signature of a different type of molecules. But we are not inferring that. We are not looking into that. We are just assuming that the whole wavelength reflects, it's like a fingerprint. It reflects what's in that leaf. And that would be somehow a reflect of the genomic information of that species, because, you know, it's a metabolomics information. But we're not inferring what it is. We don't know, actually, the specific macromolecules that are. We're just using it as a fingerprint, as a proxy.
So can you correlate these NIR data with your niche concept?
I'm sorry, can you repeat the question?
Can you correlate these NIR data with your niche modeling or niche concept?
Yes, that's what we are going to do now. We are using that as a proxy, that and the morphometrics we use in the n-dimensional space to try to correlate. So that's what we are going to do, which is the last thing I showed. We are trying to understand, we are trying to understand, one, if the spectra is correlated with the morphology, the morphometrics, and then we are going to see if that is correlated to the niche. And then we are, in a phylogenetic context, we are going to try to compare in, closely related, distant related species that we're starting to do that now. We just got the grant to do that.
Okay, thank you, ma'am.
Thank you.
No more question. Thank you very much, Professor. Thank you very much.
Thank you. Here.
during the Indian Science Congress. Do you remember that?
Of course.
Yeah, and perhaps you remember me also, because...
Absolutely. You didn't change.
So, today again we are meeting. That was you could say a hard copy meeting. Today is a soft copy meeting because in the computer sense we can use that. Well, for those of us over here, Dr. Maltrata, he is going to speak on his very pet subject, fern-insect interactions. He received his doctorate degree of natural sciences in 1994 at University, Germany, for performing field studies of high mountain ferns in Costa Rica. Since 1998, his research, he is researching and is a professor at the Institute of Ecology in Zalpa, Mexico. His broad research interest covers numerous aspects of fern ecology, ranging from the study of diversity and abundance, conservation, seasonality of growth and reproduction, functional morphoanatomy, nutrient ecology, and whatnot. So it's a long list. He teaches courses on tropical plant ecology, database management, and scientific writing, and was advisor of 23 graduate and postgraduate students from Mexico, Brazil, Bolivia, and Peru. He has authored over 80 scientific publications, co-edited in 2010, the book Fern Ecology, along with, it was Joan Sharp and others. You have that book on fern ecology. And that was published by the Cambridge University Press. It was compiled from 2011 to 2016. The annual review of teratological research is under him, and he acts as a referee for over 60 international journals. Since 2013, he is an associate editor of the Fern Gazette and the American Fern Journal. In 22, he was awarded the highest level within the Mexican research system. That's Klaus Maltrata, and today we'll have an idea of what he has been doing with insects on ferns and ferns on insects, their relationship, etc. So, Klaus, the entire stage is yours. Continue with your work.
Yes, please go ahead, Dr. Klaus.
Thank you very much for the introduction, Dr. Kuller. Of course, I remember you. You have to tell me the secret how to stay young for so long time. Good to see you all together. And yes, thanks a lot for the organizers for inviting me to give a talk at this conference. Ladies and gentlemen, dear Fern colleagues, in this talk, I would like to present you my current view of fern-insect interactions. If we put the ferns into the center of all interactions, it will like something like this. Ferns interact practically with nearly every other kind of organisms on Earth, and that is not surprising. We'll see in a moment why. But in this talk, I'll focus just on the interactions of ferns with insects. And if we think about this kind of interactions, it is obvious that there are antagonistic interactions. That means herbivorous insects feed on ferns, they damage the ferns. There are some mutualistic interactions. In other words, for example, some ants defend ferns, so there is some benefit for the ants and the ferns. And there might be neutral interactions if you think, for example, on saprophytic fungi, so there is no damage to ferns, and the ferns do not necessarily receive a benefit from such an interaction. But in the rest of the talk, I just will focus on the interactions of ferns with insects. So, there were many historical hypotheses of fern-insect interactions, and I want to just tell you about some of them. There was the perception that ferns are less attacked by arthropods than angiosperms. This goes perhaps back to Bruce 1920, who stated that only an extremely small, almost negligible portion of herbivorous insects subsist on ferns. As a consequence, numerous other authors were assuming what are the reasons for such observations. So some were stating that ferns are just of a lower nutritional value, so insects just would not care to feed on them. Others were assuming that ferns just lack many morphological structures that are interested for feeders, for herbivores. Of course, they lack flowers, fruits, and seeds. They also lack probably wood. So there are numerous insects that wouldn't be interested to feed on ferns. And then there was also the perception that ferns have just fewer biochemical defense strategies, like a diversity a lot smaller in biochemical compounds. But wait a moment. That would be an argument that would be going into the opposite direction. Imagine if ferns would have fewer defense strategies, you would expect that more species would be able to feed them. So there was a bit like a strange vision that ferns are just not interesting to study in their biochemistry, in their interaction with insects, and perhaps just because there aren't too many ferns that are useful as an angiosperm. Of course, there is an alternative hypothesis as always. And in this case, the alternative hypothesis is relatively simple. It is just we don't know. If we didn't look enough into interactions of ferns and insects, we won't know about them. And so we would underestimate this kind of interactions. So it's the same thing with the biochemistry. If we do not make any biochemical analysis of ferns, we would not know about all their components. As a consequence, we really have to think about how much of our current knowledge is just extremely biased. So let's take a look at these hypotheses we had historically. The statement that ferns are less damaged, we were looking about no more than 15 years ago into this question, and the leaf damage on many species we're looking at are ranging between 5 and 15%. And that's the percentage of leaf damage also in angiosperms. There's nothing different about it. So this hypothesis is apparently wrong. The lower nutritional value, the supposed lower nutritional value, we can look at it in a different way. And that was published by Hummel et al. In 2008. They were just questioning how can you have such an idea that ferns wouldn't be of lower nutritional value if they were the main food source of dinosaurs, the largest organisms that lived on Earth. with the exception of the marine ones, of course. So with time, most of these hypotheses were just identified to be wrong. Of course, there are some questions we can't exclude. For example, the lack of morphological features that ferns have no flowers, fruit, seeds, wood, that is not surprising, and that would restrict a good number of insects that are specialized on these kinds of tissues. But anyway, if you also talk about biochemical defense strategies, there will be a huge difference between ferns and angiosperms, but that's just a numbers game. It is not surprising that about 10,600 known fern species would have nearly for sure a lower diversity of biochemical compounds than about nearly 300,000 flowering plants. So we may just ask in general, why should ferns have fewer herbivores than angiosperms? Especially if we take a look at their distribution. The geographical distribution also indicates that ferns are nearly everywhere, perhaps just with the exception of some desert areas. And of course, ferns also have an enormous diversity of leaves, which would be not just in forms, in shape, it's also in their composition, in their tissues. So there is no reason wherever you look that ferns should have fewer interactions. So what we did with my PhD student, we're just looking into the numbers games. Let's assume for a moment that each plant species, angiosperms, gymnosperms, and ferns, would have the same number of herbivores, just assigned at random. In this case, you will make a list of herbivorous insects, here by order. Sorry. So, for example, in Coleoptera, there are about 122,000 herbivorous insect species, and then you do the same thing for the butterflies, for flies, and so on. You would end up, depending on the source, with about 300 to 400,000 insect species that are plant feeders. And on the other hand, you can look at how many insect species were reported to feed on ferns. That was in 1980, 465 species. And in angiosperms, just everything else. As a consequence, in 1980, in this article by Hendricks, there was a difference factor of nearly 30 more times insects feeding on angiosperms than on ferns. But of course, the question is how much of that is a bias? So what we did, we got into the literature using our restrictions during COVID to do just bibliographic work. And we found nearly twice as many fern-feeding insects that were reported before. So here it's about the 809 fern-feeding insects on the right. And those were reported to feed on about 382 fern species as food plots. What you see here is the result of a network analysis. And here it's like a chi-square statistic. What you do is on the left, you have this narrow row of observed interactions, in this case of ferns, fern families. So what you see is the size of each square is the number of species that serve as food plots. And in the middle, it would be the large gray column, would be the expected range. And you see, obviously, that there's a huge number of expected species that have never been reported. Remember that this analysis assumes that all insect species would be randomly feeding on just whatever plant species. The point about it is that you can see the biases in this way. For example, in Dextetiaceae, we have a huge number of reported insects, and that is just a consequence that bracken, which is Pteridium aquilinum, and all the other currently divided species of Pteridium that would be within the same genus, have been studied very much because it is a problematic species, a weedy species, and a lot of researchers were looking for biological control organisms. So we know a lot more about insects on this plant group than on other ferns. But there are other groups that are astonishingly heavily attacked. In blue here, the Equisetaceae, for example, or the Lygodiaceae, and so on. On the other hand, the really large fern families here in red means that they have fewer insect species identified to feed on them than we would expect. Now, we can do the same analysis, in this case now in insects. So this would be the columns on the right. So here you see the insect orders, and you can see that, for example, Coleoptera are biased in the opposite way. So there are fewer Coleoptera than we would expect to feed on ferns. On the other hand, we have a larger number of butterflies and moths, because here you can see that in blue. In other words, this kind of analysis gives us an idea that first, there are a lot more species than we expected. Just in the last 30 years, there are enough studies to show that the numbers doubled. And we can identify research biases to focus specifically on some groups that would be interesting to investigate in the future. So if you do these kind of interaction studies, you look into the networks, the relationships between insects and ferns, and here it's just like the three figures on three insect orders, like Hemiptera, Coleoptera, and Lepidoptera. And the only thing I want to show you here is like it seems a very chaotic structure. In other words, there is no specific order. If we would assume, for example, as it was done in the past, that only insects of very old age would interact with these supposedly old groups of ferns, you would have lines that would run just from the top group in insects to the top group in ferns, because both groups here are organized in phylogenetic order. But it's not the case. In other words, what you see here is a proof that there is a lot of host plant switching going on. In other words, that ferns are attacked by many insect groups. Really looks like random. But let's take a deeper look. Before we go into many examples of fern feeders, I just want to talk a moment about the defense strategies of ferns. If ferns wouldn't be attacked by insects, they would not possess defense strategies, and especially not very elaborate defense strategies. But if we just look in such a list here, you can see that there is all kinds of defense strategies, of compensation, of resistance, of structural, of biochemical, or mutualistic defenses. And the only question that is still not clear if there are also induced defense mechanisms. In other words, it is assumed that the highest kind of defense for plants would be one that is not always produced in the plant, but only once the herbivore appears. Once the herbivore attacks the plant, this would trigger the defense mechanism, and it's unclear if this happens in ferns. But with this panorama, it becomes clear that there is a lot of things going on in ferns we just ignored for quite a while. So, why would organisms feed on ferns? Let's start with the most opportunistic and weird kind of interactions. For example, here, a bird on the Azores Island. And this one feeds on sporangia of ferns, but only if there is no other food around. So if there are scarce food sources available, it's like an emergency case. That would be a reason to switch a host plant or food plant. In other cases, also very strange, we became aware of such interactions because Sugita et al. Was looking at some flying foxes in Japan, and they were feeding on this Asplenium species and were interested to find out if the spores, once they pass the digestive tract, would be still viable. And indeed, there were. So there is even something going on of a possible endothoracic dispersal. But when I was started to look more and more on interactions, ferns with herbivores, I was looking for kinds of indicators of specialization. And there's a number of things you can just imagine. If there is a specialization going on, then you would find multitrophic interactions. You would find mimicry, gall formers. You would find specific feeders on just one tissue of fern. You would find mutualisms, and you would find coevolution. So let's go that quickly through for some examples. In 2009, this wonderful contribution by Morse showed that here one moth was feeding on two different fern species in the US. And this moth was attacked by a parasitoid wasp. And in addition, this parasitoid was even once more attacked by a hyperparasitoid. So it was a 4-level trophic interaction. But the weirdest thing about it was that the hyperparasitoid on top of these interactions had a different success in producing offspring depending on which fern species the moth was feeding on. This means that there is a lot going on in evolution that tells us that there is a high degree of specialization, and that means that these kind of interactions had quite some time to develop. But look at these examples. Mimicry. If a species is feeding on specific ferns, they try to hide, to camouflage on these so that they won't be discovered by any predators. And here it seems that this moth on the left side is really hiding in what looks like just a natural saurus. But what it is, it's a cocoon of this moth larvae, and they just put on top of this cocoon the trash of all the sporangia they were feeding on. So they are hiding there. Or another example of a moth, that is hiding here, producing scales that look similar to the scales of this polystichum they were feeding on. Our authors were just reporting things like background matching. It's another kind of mimicry in some way. They just hide on the surface they are feeding on. This means that there is specialization going on. Or here, an observation I made years ago, the eggs of some butterflies look indeed very, very similar to the sporangia. Is there something like a Saurus mimicry? Another kind of very specialized insects are the gall formers. Because gall formers have to communicate chemically with their hosts so that these would produce the galls. This means that there is a very specific communication going on, and that means that this kind of specialization also is quite old to develop. And finally, mutualisms. If a fern is attacked by some insects, they may also attract these same ferns some defenders. You could imagine that this would start first. Sorry, I don't know why the slides are always changing on their own. And to offer some niche where the ants would live. But this would be just a living space. In this case, this Lechinopterus species has solid rhizomes. There is no hollow space to live in. But other species, like in this microgramma, there are hollow rhizomes. And if there are specific openings, they can be colonized by the ants. And even better, the ants bring in trash and waste materials that the fern can absorb with the roots that are reaching into this rhizome. So they benefit from what the ant is bringing into the rhizome. But if we make a theoretical pathway of evolution, a possible pathway, then you would come up with more or less such a conclusion. Because I was always wondering, how do these incredible interactions develop? So first you have the attack of a herbivorous insect that damages ferns. You would have ferns that attract ants by offering them just housing, like Domatia. These would develop from solid rhizomes to hollow rhizomes. Then the ferns would even be able to possibly benefit from the nutrients the ants bring into these rhizomes. And finally, if you attracted the right ants, those would be aggressive and even defend you. So there are even more interesting things going on in ferns. Perhaps you have never heard about nectaries in ferns, but if you have heard about it, you might also think it's a very rare issue. It is, but not as rare as we thought. In 2013, a checklist of extrafloral, of course extrafloral in ferns, but extrafloral nectaries reported 39 species of ferns from 4 fern families to possess nectaries. And those supposedly attract ants. Of course, that is nothing compared to the more than 4,000 species in flowering plants. But I did some more research on the nectaries, and we found about 100 species, once more about more than twice as they were reported, because they were just, they're difficult to find, and here you can see the reason why. In this case, for example, on the figure B, you just see the droplets. There's nothing strange about the tissue there. In some species, they are translucent, so you can see a difference between the nectar tissue and the regular leaf tissue. But if this is not the case, it looks like droplets. And if you don't know what it is, you would think it's just a water droplet. But of course, you can check for that. You can make a proof of it, just checking if it contains sugar, if it's sweet. If the droplets are sweet, you can even taste that with your finger, and you already know, there might be something going on. And another way to find out nectaries and ferns is just look out for ants, because those are a lot better in finding nectaries than we are. And finally, there's some proof of coevolution of ferns and insects. They are very scarce so far, but I think they are just rare because, few people look at it. And often it's not the fern people, it's not the fern researchers, but some entomologists. And in this case, for example, this was published in 2015 from Russia. They found that there are some bug species that developed on ferns. If you see here, all the species in this tree in green are fern feeders, and they specialized on those. So there was in some moment a host plant switching, and then they were switching from angiosperms to ferns. And that also indicates that there is a lot of interest for insect species to feed on fern species as well.
Dr. Klaus, please summarize so that we can go for questions.
Yep, I'm nearly ready. Thank you. I wanted to finish here just with one further question. If we do research in fern-insect interactions, you would also ask what it's good for, just to learn about biogeography of species, taxonomy, interactions. There are many interests that I just took out two I could imagine. One thing is, imagine like we produce a lot of ornamental or otherwise useful ferns, and we would be interested to know which species we have to deal with, insect species that may trouble us during their cultivation. Or the other way around, there are really species we want to control, as here in this picture on Lygodium in Florida. So there are also applied reasons why we should do more fern-insect interaction research.
Oh yes, that's very good. Thank you very much. Some questions, please. Any question or comment or anything? Yes, please. Dr. Khullar has some question.
Hello. Dr. Maltrata, do you think there is any correlationship between the insect damage on ferns occurring in tropical areas versus ferns occurring in temperate areas? Because ferns occurring in tropical areas have a very soft sort of a texture compared to ferns occurring in temperate areas that have a more thick, leathery texture. Is there any sort of a correlation, that means, of the weather and conditions of growth versus the insect-fern relationship? What do you think about that?
Yeah, Honestly, I think we cannot respond to that question now because we just still have not enough data on that. The average is nearly always somewhere between the 5 and 15%. There are outliers, and interestingly, one of these outliers is a polysticum, and you know the polysticum is already a bit of a hard tissue, so it's not necessarily related just hard versus a soft tissue. If you think about whatever toxic substances might be there, more important than just the texture of the tissue. But all this kind of questions we should really investigate. The question, for example, if epiphytes and terrestrial species are different, if tropical or temperate species are different, and just one more idea there, of course that in the tropics you can also have very hard tissued ferns. In Mexico, for example, this happens in the dry areas. The drought-adapted species have very tough tissue as well, and also in the high mountain areas. In the Paramo areas, for example, with a high ultraviolet radiation, with strong winds, you also have this kind of very tough tissue. And then you have to open up even more and to imagine what happens to the insects. And of course, at high elevation, you have fewer insects. So it's also to think about where do you have more insect species, and that would be clearly in the tropics.
Thank you very much.
Thank you.
Yes, please.
Thank you, Dr. Claus. It was really very enlightening and a very informative lecture. I just had one question. If the insects prefer the taste of the ferns, do they have any bias towards gametophyte over sporophyte? A gametophyte looks more juicy, but I didn't see any of the ant attacking in gametophyte. Why do they like tough adult plants?
Yeah, that's a really good question. I think we don't know nearly nothing about feeding on gametophytes. I think you also cultivate gametophytes, and sometimes you will see some damage anyway. I think the real problem is that the small gametophytes, if they are predated, perhaps they just disappear completely, and then it's difficult to quantify damage. But yeah, you're right. The point about gametophytes and sporophytes is always interesting because we have to think about that they have the same genetic information, they have the same potential, but they don't use it. And that's something really interesting about ferns. If you think about whatever study you perform, you have to ask, why does how does a gametophyte know that he's a gametophyte? Yeah, and especially in epigamic species where they have the same number of chromosomes. So they just do not use that information and develop into a sporophyte. But yeah, I don't know. Perhaps we should do also some trials and offer insect species, gametophytes and sporophytes to find out if they find something more juicy than others.
Not a question, but just a comment. I have read many papers in Paleontographica by D.D. Pant and his associates. Even in the fossils, they have found the insects. I cannot exactly remember the genus, but it is of Cretaceous to other parts, maybe even Mesozoic. They have found in fossils the insects, when they macerate the tissue, they found the insects. So it's quite old. Yeah.
You're right. There's a lot of more information out there. It is just very scattered. And often I think the problem is, as I was telling you, that entomologists and teratologists rarely collaborate, and I think that's exactly the message here. We need a stronger collaboration. If you look into the data when we did this research, it is just strange to imagine that entomologists were reporting very detailed their insects, and then as a food plant, they were just reporting, oh, it was just a fern, I don't know which one. Yeah. And on the other hand, the pteridologists were very similar in some way, like they're very careful about identifying the fern species, and then they reported it was just a bug. So, like, yeah, we have to do just more of collaborative research to get a deeper understanding of these interactions.
You have some question?
So, my name is Leslie. I have I don't know whether it is a foolish question, but I just want to ask the question. Why does insects has a limitation towards ferns? Why they are not preferring lycophytes? And the second question is that more than Lepidopteran members, is there any weevils are attacking these ferns? Is that kind of interactions has been observed in the field? Two questions.
Okay, thank you, Leslie. There are no foolish questions. Never think about that. I think the best questions are often the supposedly simple ones, and then we are in trouble because we don't have a response for them. But I can tell you, just like here I was focusing mainly on the interaction with ferns. With lycophytes, we have a lot less information. There are some reports in Hupersia and Selaginellus as well. So there is not a restriction there. Lycophytes are also attacked. I think the point there is very clear that lycophytes just are not very abundant in any ecosystem. As a consequence, yeah, they don't have so much biomass to offer. And for an insect, it's very important to have enough food. So if whatever group you're investigating is not offering a lot of biomass, then it's not the most interesting food to look for. And the other question about the cotyledonids, indeed, it is the best represented group in the Coleoptera that feeds on ferns, and I think there will be many more. When we were hunting for these fern insects, I can just tell you I was amazed about the numbers, and it's just a question because many of those are so tiny, so small, that you would nearly never see them if you do not hunt them specifically. It's like you won't even recognize them in the field, like walking around on the ferns, as you would see that with larger insects. As a consequence, I think it's, as always, it's a lot of dedication. We have just to do our job, look in detail what's going on, and we'll find a lot more insects on ferns, and a lot of really interesting results will show up in the future. Thank you.
Thank you very much, Professor, for a wonderful and highly enlightening talk. Thank you very much.
Thank you very much, and good luck with the Congress.
Thank you very much.
Yep.
So I request Soma to please just give a brief, quick review.
Good evening. Here in this technical session 6, we have 3 presenters. Here one is Joel A. Nitter. He is talking about DNA barcoding of fern gametophytes, past, present and future. And he reviewed on the barcoding, and especially he recommended RBCL, tRNL, tRNF barcoding recommended. And apart from that, only for for Sanger sequencing, he preferred for next-gen DNA sequencing for potential fern biology. And then the next was the using integrative approaches in systematic and evolutionary studies on neotropical ferns and lycophytes by Almeida. And she gave an integrative approach including both all the morphometric data and also the near infrared spectral data along with the environmental data and the DNA sequencing. And then he compiled using all those data in bioradar.com analysis and tried to give the phylogenetic evolution as well as the speciation of the species she has studied. And the last one was a panorama of fern-insect interactions by Klaus Maltreter. And here beautifully we have enriched with the information of the insect-fern antagonistic interactions as well as the mutualism and how the ants help to defend the ferns apart from their several defense mechanisms in ferns. That's all. Thank you.
And he very rightly suggested that you have to hunt to find insects. Insects will not come and tell you. You have now tried to hunt them. Okay, thank you.
Thank you, Dr. H.K. Goswami, for chairing the session. Thank you, Dr. Soma Sukul. Now I request Professor H.K. Goswami to present a memento as a token of our love to Dr. Soma Sukul. Please, sir. Thank you, sir. Thank you, ma'am. So all the technical sessions of the day has come to an end. I thank all the participants for your cooperation and enthusiastic participation. Tomorrow's session will start by 9 a.m. The breakfast will be served from 8 a.m. Now all are kindly requested to move to the University Park immediately for dinner. Vehicles are arranged outside the seminar hall. Thank you. Good night and see you tomorrow.